multimodal data sets and discovery. And then, as we uncover the questions and support, multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can together stand the If we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end and cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics. we could anticipate the future and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new or predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. With expansive multimodal data sets and AI-powered discovery, with the highest, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. There, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the in this at the nexus of innovation and with expansive multimodal data sets and AI-powered discovery, with the highest quality insight and cost, we can accelerate impact and improve human health together. What we could anticipate is and transcriptomics, proteomics. Question, innovation, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this time, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve health together. What if we could anticipate the future of disease and prepare for it today? Genomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. And explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together. What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of where personalized medicine within reach. As we uncover the questions and explore the solutions together, stakes of our search. Your question is the innovation. Your innovation is the insight, is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate health together. What if we could anticipate the future of disease and prepare for it today?
Good morning, everyone. Please stay tuned. We'll soon start. It's okay.
Disease and prepare for it today. Genomics, epigenomics, transcriptomics, proteomics. Question. What if we could anticipate the future of disease and prepare for it today. Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity or predictive. What if we could anticipate the future of disease and prepare for it today? Genomics, as we uncover the epigenomics, transcriptomics, proteomics. Question, innovation. Questions and explore the solutions together, we come to understand the promise. Ecosystem at the nexus of innovation, solution, and community. With expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your project, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabytes of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistics team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budgets. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genlab.ng or cgl_ont@genlab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question:
Hello, excuse me. Those online cannot hear you. Hello, please unmute. Those online on Zoom cannot hear you, please. Please unmute the Zoom. You are muted. You are muted.
It's my rundown. Okay.
Hello?
Okay. After Professor Chimeze Jesin, the Dean of Faculty of Science, has given his welcome remarks, the next person will be the Vice Chancellor, University of Port Harcourt, Professor Owunari Abraham Georgewill. He will declare this event officially open. After that, Professor Boabid Badawi, Chair, African BioGenome Project, will give us a talk on African BioGenome Project, AfricaBP, Open Institute for Genomics and Bioinformatics. Then after that, we'll take some comments and questions. And then we go for a tea break. Thank you. And I call on Professor Julian Osuji to come and give us his preamble.
Thank you, my digital vice chancellor, Professor Georgewill, able Deputy Vice Chancellor, Administration and Advancement, Professor Nyaso, my Dean of Science, Professor Ogugwe, distinguished professors, members, gentlemen of the press, ladies and gentlemen. I welcome all participants to this first of the 2026 series of the African BioGenome Project Open Institute Regional Hybrid Workshop and the inaugural African Congress on Digital Sequence Information, Infrastructure Development, and policy. The University of Port Harcourt hosted the inaugural edition of this workshop on 15th and 16th of February 2023. Its success led to the foundation of 3 subsequent workshops held in Southern Africa, Northern Africa, and East and Central Africa. Africa's diverse ecology is richly endowed with biodiversity with genetic resources that remains largely underexplored. To address this, AfricaBP aims to sequence 100,000 endemic species over 10 years, as outlined in the 2022 white paper, a publication in Nature Journal. This initiative emphasizes capacity building through workshops and fellowships, encouraging engagement from omics-based scientists worldwide to enhance know-how and funding for the project. At the 2023 West Africa Regional Workshop, I highlighted the urgent need for capacity in African biodiversity genomics and bioinformatics, as noted in our Nature Biotechnology publication. This aligns with AfricaBP's mission to train and develop local expertise to sequence over 80% of African biodiversity genomes within the continent. The workshop focused on capacity building, supports the collaboration among industry, academia, and entrepreneurship, leading to training fellowships at institutions like the African Genome Center, Morocco, and the University of Illinois. This resulted in on advancing Africa's bioeconomy and biodiversity conservation in another Nature journal. The AfricaBP 2025 workshop series focused on the theme biodiversity genomics and One Health, specifically emphasizing the West African sub-theme of biodiversity genomics and livelihoods, showcasing and livelihood sustainability in West Africa. This series aimed to dive into the correlation between health and wealth, showcasing how AfricaBP's initiatives in genomics and bioinformatics contribute to conservation, capacity building, and health sustainability across the continent. A key highlight of the workshop is the anticipated outcomes from the 2025 regional workshop titled Contributions of African Biodiversity Genomics and Bioinformatics to the One Health Nexus, which in preprint review. The 2026 workshop series aimed at enhancing Africa's positioning in the digital sequence economy. This initiative seeks to capitalize on the recent advancements by AfricaBP in leveraging digital sequence technology. Key topics for discussion include the generation, storage, and accessibility of digital sequence data obtained through omics technology. The workshop will explore grassroots-friendly strategies for material acquisition and implementation of relevant policies that ensure equitable access and benefits for all stakeholders. Additionally, the Congress will focus on bridging the gaps between biological samples, traditional knowledge holders, digital information, and end users, and are all encouraged. You are all encouraged to engage with these critical issues as you partake in this exciting 2026 event. Thank you. You're welcome.
Thank you, Professor Julian Osuji, for your well-crafted preamble. I now call on Professor Chimezie Jesin Ogugwe, Faculty of Science, Dean, Faculty of Science. Please, sir, come over and give us your welcome remarks.
Yeah, thank you. The Vice Chancellor, sir, Professor Owunari Abraham Jojo, the Digital Vice Chancellor. The Deputy Vice Chancellor, Administration and Advancement, Professor Chukwudi Onyaso. The Director, ICTC, Professor Mary Wondo. Conference participants, both here present and online, gentlemen of the press, distinguished ladies and gentlemen, good morning. This is a welcome address by the Dean Faculty of Science at this epoch ceremony. It is truly a pleasure and privilege to welcome you all to the University of Port Harcourt for the first African Congress on Digital Sequence Information, DSI, Infrastructure Development and Policy. This Congress, with its thought-provoking theme, Powering African DSI Infrastructure Through Deeper Policy Discourse, could not have come at a more important time. It is especially significant that we are gathered here within the wider context of the African BioGenome Project Open Institute Regional Workshops, yet with a clear and dedicated focus on the issues surrounding digital sequence information in Africa. Across the world, digital sequence information is rapidly shaping the future of science. It is influencing how we understand biodiversity, respond to health challenges, and develop sustainable solutions for our environment. However, as a continent, we are still working to strengthen the structures, policies, and systems that will allow us to fully harness these opportunities in ways that are equitable and beneficial to our people. This is why this Congress matters. It brings together not just scientists, but also policymakers, institutional leaders, and partners who all have a role to play in shaping the future of DSI in Africa. It creates a space for honest conversations about where we are, what gaps exist, and how we can move forward together. At the Faculty of Science, University of Port Harcourt, we believe strongly that science must speak to society, research must inform policy, innovation. We believe strongly in that. Hosting this Congress reflects our commitment to supporting conversations and collaborations that drive meaningful change, not just within our institution, but across Nigeria and the African continent. Over the course of this Congress, I encourage us all to engage openly, think boldly, and focus on practical outcomes. Beyond discussions, we must begin to shape ideas into frameworks and frameworks into actions that will strengthen Africans' capacity to generate, manage, and govern digital biological data responsibly and effectively. We also hope that this gathering will inspire a new generation of science policy champions who can bridge the gap between knowledge and decision-making, and who will continue to push forward the development of strong and inclusive DSI infrastructure across Africa. And I'm happy today to see a lot of young people here, participating in this conference. Once again, I warmly welcome you to the University of Port Harcourt. I wish you a productive, insightful, and rewarding Congress experience. Thank you, and God bless.
Thank you, Professor Chimezie Jesin Ogugwe. Thank you for that wonderful remarks. May I call on the Vice Chancellor, University of Port Harcourt, Professor Owunari Abraham Georgeville, to declare this event officially open.
Thank you. Good morning, everyone. My respected Deputy Vice Chancellor, Admin and Advancement, you're welcome. The Dean of Faculty of Sciences, you're welcome. The convener of this Congress, you are welcome. Director of ICTC, you are welcome. Other distinguished guests and participants, those in persons and those online, I welcome all of you. And I thought that students will have taken this opportunity to be here more massively, because most times you spend so much money traveling for conferences outside your station, abroad, and so on. But today, this kind of wonderful conference is brought to your doorstep. And I thought students in this area, in these thematic discussions, will have found it so as an opportunity to be present. To some, it will be the first time they may be participating in such a high-level conference. I know that university workers on strike, but that does not affect students. Are students on strike too? Students, are you on strike? They are not on strike. Okay. Welcome to the unique University of Port Harcourt for the annual African BioGenome Project event. This is my fourth time initiating this annual program. The 2026 Open Institute Regional Workshop brings together experts in genomics, digital sequence information, and artificial intelligence. This week, discussion will focus on advancement in genomics and bioinformatics and relevant policies, emphasizing the significance of digital sequence data in understanding life processes. The potential for misuse of this knowledge necessitates education on international policies like the Convention of Biological Diversity and the Nagoya Protocol. The African BioGenome Project aims to empower Africa. The University of Port Harcourt proudly supports these initiatives and celebrates the consortium's achievements, including 6 publications in the Nature magazine or journal. I appreciate the great job done by the local organizers, sponsors, and speakers for their hard work and dedication towards success of this outing. I welcome the delegates, all of you, to the University of Port Harcourt. Your in-person participation and engagement make this workshop truly special. I also welcome online participants who could not be here in person. I acknowledge that your participation makes this hybrid event very robust. Today's Twins event focus on biodiversity, genomics, and digital information infrastructure development. That is scaling West African biodiversity, genomics, and bioinformatics insights in the age of artificial intelligence. And the first African Congress on digital sequence information, infrastructure development, and policy. We believe that is very timely. Having sincerely welcomed you all to this event as the 9th Vice Chancellor of the University of Port Harcourt, and on behalf of the Governing Council, the management, the Senate, the staff and students of this very unique University of Port Harcourt, I have the privilege and honor to hereby declare this twin event open and wish all of us, a memorable participation experience. Thank you. God bless us all. Thank you. Thank you.
Thank you, Professor Georgeville, for declaring this event officially open. Thank you for doing justice to this, your presentation, sir. We are very grateful to you, sir. Next will be Professor Boabid Badawi. He will be presenting online. He's the chair, African BioGenome Project. He will give us a talk on African BioGenome Project, that is AfricaBP, Open Institute for Genomics and Bioinformatics.
Thank you very much. I will try to share and activate the camera. 1 second, please.
Please, Professor Boabid Badawi,
Yeah, he's
can
online.
you unmute yourself?
Yes, yes. Can you hear me?
Can you hear him on site?
Can you hear me?
He's speaking online. Can you hear him on site?
Please, you can speak.
But can you hear me? Because I'm not sure.
Can you hear us online? Can you hear us on site? Boabid, you can carry on, please.
Yes, I will try to share. Thank God. Can you see the presentation?
Yes, please. But we can see the speaker's slide, speaker's note or something. So you need to switch it. You need to switch.
Okay.
So you can just maybe you can present the way you have it previously, Boabid.
No, don't worry. I will fix it. Just I have to, because I have two screens. I will share the correct screen.
All right. Okay.
Yeah. But just I'm not sure that they are hearing us. Thank God. That's the problem.
Yeah, I think it's more of the IT on the ground. So, Bob, I think you can progress and present while they fix it.
Okay, good.
We can hear you.
Okay, great. Okay, just 1 moment, please, to fix the presentation, and they will come in.
Okay.
So, Genevieve, can you hear us from Zoom?
Yes, I can hear you.
Okay, all right. Okay, I want to be sure that the on-site participant can hear us on Zoom. So, Boabid is trying to fix his presentation.
Yes.
So, Boabid, do you want to put it on Google Drive, or then you use Google Slide to present, or how do you want to do that?
I will just tell me if now you can see the complete presentation.
Yes. Yes.
You can see it? Thank
Yes,
God?
carry on, please.
Okay, good. So I'll just activate my camera so we can. It's okay?
Yes, please carry on.
Okay. Hello, everyone. So first, I would like to thank all the people of the University of Port Harcourt who contributed in the organization of this important event. So we will be unpacking framework so ambitious. I think that it's actively rewriting the rules of science on the African continent. I'm speaking about the African BioGenome or AfricaBP. So over the next few minutes, we will see exactly how genomics could be utilized in the best way possible for conservation, bioeconomy, and biological sovereignty. But before we even get into the biological of it, I really want you to consider the massive macroeconomic stakes of biological data. I mean, just to ask questions, I mean, can sequencing DNA actually act as a shield for continent future? Or say it in another way, could it literally safeguard an entire continent economy? Just keep this in mind because we will have some hints in this sense to try to answer this question. So just think about the massive scale of nature capital we are talking about here. I mean, from the Mediterranean basin to the Guinean forest of West Africa, all the way to the Horn of Africa and the Cape Floristic region. I mean, this is a fantastic shared global heritage. But as we know, we are staring there down a 6-month mass extinction, and this isn't some hypothetical far off threats. I mean, it's real that climate and human pressures are actively eroding the very ecosystem that sustain both local lives and national economies right now. So we can understand that to turn the tide, the solution has to be equally massive. I mean, the goal of AfricaBP is to sequence the foundational DNA of 1,500 endemic species across the continent. I mean, it's just, we are not just talking about saving few animals or plants. This is a massive infrastructure project designed to map the entire ecological web. So here is how we start to trace the path from crisis to self-sustaining sovereignty. I mean, after covering the heritage and impending threats, we will speak about the AfricaBP grand vision, the safeguarding of biology, and the unlocking of genomic bioeconomy, the activating of health nexus, and finally, the sovereignty and the flywheel. So let me speak about the AfricaBP grand vision. So this vision, let's say it started from situation of isolation to coordination. This evolution happened surprisingly fast. Back in 2013 to 2015, you had this scattered, completely isolated plant and animal genomics communities slowly starting to talk to each other. But then by 2019, portable sequencing came along and totally changed the game. It literally put advanced genomics directly into the hand of local scientists. So that technological leap birthed the unified framework by 2021, which is AfricaBP, creating a single cohesive continental umbrella. We believe that this massive paradigm shift, we are moving completely away from the old fragmented model of outsourcing sequencing, because what happened historically is that the old approach we were using, or most of the African countries use, just drained data and capacity right out of the continent. And we believe that the AfricaBP approach now is flipping that entirely. It's a unified network built in Africa, especially for Africa. And it's powered entirely by distributed governance. The core pipeline of this whole operation really boils down to 3 interlocking actions. First, initiate sequencing of those 1,500 endemic species. Second, coordinate efforts by acting as the central unifying form access across all the continent. And third, which is very important, deploying the result data to regional biobanks like the Africa DCDC. And this, of course, to ensure that this data actually drives real-world continental policy and scales on-the-ground capacity. Of course, a project of such magnitude needs a scientific rigor, and we believe it has it. Because this project, AfricaBP, is driven by a distributed network of 1,900 scientists, and crucially, 87 of them are operating right on the ground, working with or alongside 22 integrated organizations. So together what they try to ensure global scientific standards and protect and prioritize the regional economic interest, either in the north or south or east or west or central Africa. Which bring us to section 3, safeguarding biology and capacity. So this is pillar 1. It's all about generating vital genomic references of 1,500 species. But we can ask why this is absolutely crucial for everyday life. Well, because the key to food security, because we think that those genomic references are the key to food security, because it gives geneticists, either plant or animal breeders, the exact genetic information they need to build a resilient, climate-adapted food system. Furthermore, it aligns perfectly with the global biodiversity target to maintain 90% of genetic diversity by 2030. But to actually generate that massive amount of data, they have to bridge a major capacity gap. We know that building capacity is a problem in Africa. But through the hub and spoke model, the Open Institute is decentralizing bioinformatics infrastructure. Is organizing workshop in any region. And just an example here, we scaled from 21 hands-on scientists in 2022 to an estimated to more than 500 by 2025. Of course, this is very important because it's completely bypassed traditional visa cost and travel barriers that generally hinder the movement of the African scientists. Let's dive now into the financial data in sector 4. Unlocking the genomic bioeconomy. So let's see a simulation that we did for Morocco and later on generalize this to other African countries. So the number you can see here, 3.29, it's very indicative. What I mean by that is that for every single dollar invested in domestic genome sequencing, 3.29 is generated in downstream economic value. This is what we name the benefit cost ratio. And it proves once and for all that genomics isn't just a scientific endeavor, it's a profound economic multiplier. And this is very important because in Africa, we are looking not just to science as a science, but we are looking to build economies. We are looking to produce more. We are looking more to the comfort of the African citizen. Looking at this at the macroeconomic level, we did the same thing for the other African countries. And we found that the foundation $20 million investment is projected to steadily grow in 79 million in Morocco. And this is thanks to what the economists, they call sectoral spillover. What I mean by that is that when you make an investment, it sparks massively downstream all the economy, including agriculture, research and development, innovation, and localized bioindustries. So as I said before, zooming out for the other African countries, we can see that the economic models predict a literal genomic shock expanding this kind of capacity, which restructure the capital and the output. Let's take an example as we are in Nigeria, where the event is organized. In this country alone, models show an extraordinary 22.14 benefit cost ratio. We are talking about triggering up to 26% gains in capital income. That's really a very promising thing to expect if we invest in sequencing and genomics during the next 10 years in Nigeria. Of course, economic momentum means nothing without public health, which brings us to this section. So I mean the health nexus. Because here is the thing, you literally cannot protect human health without the intervention, in vacuum. This One Health approach proves that human medical frameworks are entirely incomplete without mapping the genetic diversity of plants, animal, and the environment. Because we need to connect all this data to track the disease spillover between wildlife and the humans. And we have many examples. I think one had been done even in UM6P University, if I remember well. It utilized the language model to monitor antimicrobial resistance and really secure long-term ecological stability. So finally, let's wrap up with this section that I named Sovereignty and Flywheel, and see how this could become self-sustaining ecosystem. Before this engine that I name Flywheel can wane sustainably, as you can see in this figure, in this slide, Pillar 6 has to establish the rules of engagement. This rules include structuring a roadmap to create a robust framework to ensure the digital sequence information respect traditional knowledge of indigenous people. It creates national policies that activate and protect African intellectual property. But of course, still, it has to have the capacity to foster a really dynamic environment of open innovation. And this take us or lead us to the ultimate goal, which I name here the flywheel. And the main message of this is that AfricaBP, we don't consider it as just one of sequencing projects, because we want it to be a pilot project for all the economic system in Africa and of all the possible collaboration within the African countries. And we want it to be a designed and self-sustaining continental ecosystem. As you can see in the figures, it works like this. The vision should drive the capacity unlock the rapid genomics discovery, which could in itself triggers massive economic prosperity. The prosperity then informs sovereign policy, and this sovereign policy reinvest right back into the initial vision. It could be, if well done, permanent, continuous loop of empowerment. Of course, we can ask how we think that we can keep this flywheel spinning. The action plan relies on 4 clearly steps that we mentioned before. First, we have to support the sequencing. We have a big vision of 1,500 endemic species. Second, we should continue capacity building, empowering local experts through the Open Institute. Third, we have in the midterm invest heavily in the resulting bioeconomy and using the information that we get from genomics to sustain the African economy. And above all, guaranteeing data equity at every single stage, because this is what happens often, that the indigenous and the local people are forgotten. So for that, this specific objective or directive, ensuring absolute African genomic sovereignty and data equity, it sits at the heart of the AfricaBP framework. It's not just participating in global science. It's about guaranteeing and taking uncompromised ownership of the biological data, ensuring that the people of Africa are the primary beneficiaries of their own natural heritage. So to close, I think that we have the blueprint for sovereign thriving, self-sustaining genomic ecosystem, African genomic system. We started to work on it with the collaboration of many scientists. I take the opportunity to thank all of them. The only question now is how quickly we can build that together. And I think to reach our goal, we need more collaboration and more African people to join the coordination of this event. Thank you very much for your attention, and I will be happy to take questions if there are any. Thank you.
Thank you for that wonderful presentation, Professor Boabid Badawi. Thank you for doing justice to that topic. You have really enlightened us on so many things that we may not have known before now. Please, we want to take some comments and questions from the background. Okay. Yes. Pardon me? Somebody should move and take. Okay, put another microphone.
Yeah, Asimi.
Yes. Thank you so much for this opportunity. The Komi-Montreal Global Biodiversity Framework have 23 targets, and the theme of this workshop is on target 13, increase the sharing benefits from genetic resources, digital sequence information, and traditional knowledge. So my question is, how can we achieve this target without the implementation of the different MBSAPs in different African countries? Because I've worked on the review and implementation of Nigeria, which is still hanging. It has not been implemented or submitted on the MBSAP platform. So 13, how is it possible without, because what I know is in order to take a genome or whatever, you're working on maybe a particular species or a vital keystone species. For example, if you want to take that, the sequence to Germany or any other country to sequence that genome, you have to, there are a lot of policies you have to pass through to do that. So without implementation of the MBSAPs of these various countries in Africa, how can we be able to achieve target 13 of the Komi-Montreal Global Biodiversity Framework?
Yeah, thank you very much for your question. So maybe Genevieve, if I may take this. So I think we're kind of your question is early because tomorrow we are dedicating the whole of tomorrow on DSI and the Convention on Biological Diversity and WIPO and BBNG and all of those things. So, and maybe perhaps if we center the question specifically on the discussion that Boabid presented, mainly high level, then that would allow us time to kind of dive deeper into it tomorrow so that we don't discuss what we're going to discuss tomorrow today. If that's okay. That's to the gentleman that asked the question. Yeah, so I would leave that tomorrow. So we'll pick it up tomorrow because we have experts in the room tomorrow from the convention, so from the UN. We'll have experts also from CGIAR and also so many of my colleagues here. And we also have someone that is an expert in Nigerian ABS as well. If that's okay, yeah. let's pick that up tomorrow.
That is okay. Okay, so we'll leave it for tomorrow. Thank you. But please put it down. Okay. More questions? Should we go ahead and take more questions? Or we'll leave it for tomorrow. If any, yes, yes.
Thank you very much, Mr. Vice Chancellor, DVC, my Dean, ladies and gentlemen. The structure of this program is bipartisan. Today is the science, the technology involved in biodiversity, genomics, bioinformatics, and all that. Tomorrow, we'll go into the policies. Asime is a resource person for IUCN, and I think that's why he's interested in that. So Asime, don't worry, tomorrow we are going to KMGBF and all the frameworks that are associated with biodiversity genomics. And there are actually people who are here because of that. So today you are going to see the science, the technology, and all of that. Tomorrow we'll be seeing the policies. We have, like Dr. Tango that said, there are so many policy experts coming from America, Europe, Africa, Asia, and all that. So that's it. Thank you very much.
Tea break. Photograph before tea break. Please stay tuned. Please, while we wait for the tea break, we want to call on the keynote presenter for the next session. I want to call on Dr. Shane Murray, the lead genomics coordinator, Diplomics South Africa, to give us a talk on decoding South Africa's biodiversity. Please unmute yourself.
Yeah, so I think I can see Shane online. Shane, are you online? I can see, okay, okay, she started a video. Go ahead, please.
30 minutes to speak.
Shane, are you online? Could you please start your slide?
Please unmute yourself.
And unmute. Shane, so I think if Shane is not online, we can move to the next speaker. If the next speaker is on site online. So
the next speaker is Dr. Innocent Honor, Chief Natural Resources Officer, African Natural Resources Management and Investment Center, African Development Bank, Abidjan, Ivory Coast. The talk will be on how can the digital sequencing information, DSI economy, contribute to financing development in Africa?
yeah, I don't also think, because we are a few minutes early, Innocent is not online yet. So let's see if who is online? Nikki Adams. So
Dr. Nikki ready?
Yeah,
Nikki Adams ready.
So yeah, we can. So
okay,
we can make Nikki Adams a co-host, and Nikki Adams, please make your 10 minutes presentation.
Okay. The talk will be from big dreams to big possibilities.
Good morning, everybody.
Dr. Nikki Adams.
Please carry on, Nikki Adams.
Good morning, everybody.
Yeah, please carry on. We can hear you.
Perfect. Okay, I'll start sharing my screen now. Just give me 2 seconds. Perfect. Can you see my screen okay?
Yes, please, we can.
Perfect. Okay, so good morning, everyone. My name is Nikki Adams, and I'm a field application scientist for Oxford Nanopore Technologies. I'm based in Cape Town, South Africa. I will be giving you an overview of how Oxford Nanopore Technologies has blended creativity, action, and resilience to become a one-stop multi-omic solution. Before I begin, I would like to declare that I have no conflicts of interest with regards to the content of this presentation, and please note that Oxford Nanopore Technology products are currently for research use only. Since 2014, Oxford Nanopore has consistently delivered innovation by introducing new sequencing devices to address specific needs with enhanced platform performance through chemistry and software improvements. Our technology is one platform that adapts to your needs, no matter the scale or biology of interest. Whether you're working with DNA, RNA, or perhaps other molecules in the future, this technology is continuously evolving to explore biology without limits. What ties it all together is a single, flexible approach that makes discovery more connected, streamlined, and scalable. At the heart of our technology is the nanopore. These animations illustrate how our core chemistry, which is made up of nanopores embedded in a membrane and motor enzymes attached to nucleic acids, allows direct sequencing of DNA or RNA molecules as it moves through the nanopore. The regulated passage of nucleic acid through the nanopore causes disruptions in an ionic current, which can be decoded into sequencing data rich in epigenetic information. Unlike other platforms, we do not use enzymes to amplify nucleic acids on the flow cell, and therefore data is not lost due to amplification bias. Even though we are known for our long reads, I would like to remind everyone that our platform is read length agnostic and can sequence reads as short as 20 bases. Our powerful software enables real-time analysis of data as it is being generated, allowing immediate access to information for quicker decision-making. Our bespoke electronics are low cost and scalable to support low or high throughput projects. Hello?
No, carry on, please.
Oh, okay. We have two flow cell types which house the magic of nanopore sequencing. These flow cells use the same chemistry and only differ in the number of pores they have available. Our min-ion flow cells are primarily used for smaller genomes or targeted applications, whereas the PromethION flow cell is geared towards larger genome sequencing. Our devices have been designed to fit a range of research needs, from low-throughput mobile sequencing with the min-ion Mark 1D to high-throughput benchtop sequences which have integrated compute, such as the GridION or PromethION 24. Our technology allows you to access the full biology from a molecule of any read length, be it single nucleotide polymorphisms, methylation, or structural variants, without having to use another device or a different technology. Read lengths of up to 4 million bases enable easy assembly of any genome and is particularly useful for specific organisms like bacteria who have extra chromosomal DNA. Over the past decade, our team has persevered to continuously improve the performance of our platforms, enabling multi-omic data to be generated in a single run. The hard work is paying off as our technology currently provides the most complete biology from one sequencing run. Everyone knows how difficult it is to capture all variant types, but this slide illustrates all the dimensions that Oxford Nanopore data can cover. It spans from high performance in mapping to variant detection, which includes base modifications. Modification information, together with long reads, enables state-of-the-art phasing and genome assembly. These quality metrics showcase the platform's high performance across multiple dimensions, such as single nucleotide polymorphisms, structural variant detection, RNA and DNA modification accuracy, and telomere-to-telomere chromosome assembly, enabling robust interpretation at every layer of biology. With the ability to recover gap-free genomes, phase genes end-to-end, and detect dozens of modifications, the platform delivers the depth and resolution needed for advanced genomics, transcriptomics, and metagenomics, all at scale. In order to unravel this complex multidimensional data, we require software that is easy to use. Our teams have been working on a tool called Dorado that has moved beyond base calling and provides the basis for optimized analytical insight. For ease of use, the latest versions of Dorado are gradually integrated into the Minnow device software and our secondary analysis software platform, Epitome. We continue to deliver performance enhancements, especially for our optimized high-accuracy base calling models, which is recommended for most applications, as it is compatible with real-time base calling and alignment on all of our devices. We are obtaining raw read accuracy gains of over Q22 and improved SNP and indel performance. The latest release of Dorado includes a variant calling tool, but it is still at its early stages of implementation and not yet integrated into Minnow or Epitome. Claire 3 is still our recommended variant caller, and our teams are working on developing a tool for somatic variant calling, aiming at specific variant detection of less than 5% variant allele frequency. Traditionally, we use single nucleotide polymorphisms to phase reads into haplotypes. However, in regions of high homozygosity, this becomes really difficult due to the lack of informative variants. Because different haplotypes often exhibit distinct methylation patterns, we can leverage this epigenetic information to improve phasing continuity across these challenging regions using the POMFRET tool. What you see here is the improvement from classical variant phasing on the top left to methylation-assisted phasing on the bottom left, which helps extend phase blocks. Our scalable platforms with easy-to-use workflows facilitates the needs of customers, enabling low to high throughput projects across the application spectrum from small to large genomes. From a genomics perspective, we have multiple workflows for targeted whole genome or telomere to telomere applications. The benefits for all of these workflows is the access to multidimensional data, cost-effective approaches, and fast turnaround times on scalable platforms, allowing the delivery on the needs of a wide variety of customers with no batch penalty. We have 3 enrichment options, from focused to flexible, all on one platform. All enrichment options enable the detection of single nucleotide variants and structural variants, with adaptive sampling having the added benefit of providing methylation information. One can choose the best tool based on the enrichment depth, region size, and data types that they require. For high-throughput whole genome sequencing projects, our team is currently working on a high-throughput barcoding kit and protocol to ensure consistent 30x coverage for all samples. Multiple barcoded samples are pooled and sequenced across several flow cells, which guarantees that even if one flow cell underperforms, the sample is not lost, and the combined data from all of those flow cells achieves the necessary coverage. As I mentioned, the protocol is still currently in development, but it will provide detailed guidance on automating extraction and library preparation using liquid handlers, and it includes plate-based shearing and limb tracking. This methodology can also lower the cost per genome, as it has the potential to enable more than one genome per flow cell, and this will improve the cost. We have 2 transcriptomic approaches which are continuously being developed to improve output and usability. We have recently released a new and improved cDNA protocol, which allows the capture of more full-length transcripts with double the read length and up to 150 gigabases of output per PromethION flow cell. This allows improved expression analysis accuracy, as transcript coverage is significantly enhanced at the 5' end. Oxford Nanopore Direct RNA sequencing delivers high output of exceptional accuracy. This includes RNA modification information, which is critical for understanding various biological processes. As our platform is read-length agnostic, we can capture both full-length transcripts and short RNAs in a single streamlined workflow. The next big dream is enabling proteomics capabilities on our platforms, and our team is working hard to make this a real possibility in the future. Thank you so much for your attention.
Okay. Thank you, Nikki Adams. So I think you might just hang on the online, because there will be a Q&A session. It's going to be a panel session. Do we have on-site participants? Genevieve, do we want to call the next presenter? I think Shane should be ready now.
Okay.
Yeah, so do you want to call her? Because she said she'll be ready in 10 minutes, well, which is now,
Okay.
actually. Yeah.
Okay. Dr. Shane Ray, can we take your presentation?
Shane, could
Decoding
you please...
South Africa's biodiversity.
Shane, please unmute and start your presentation.
Shane is ready.
Shane, could you please unmute then? Okay, I think we would go to the next presenter. We have, do we have Vilches from Illumina? Vilches? Yeah, we have Vilches. So Vilches, could you please present? You have a co-host right now.
Hello, can you hear me?
Yes, please, carry on. You have 10 minutes to, yeah?
Yeah, I will share my screen.
Yeah.
Can you please confirm to me you can see my screen?
It's coming up. Yes, now we can see. Please carry on.
Perfect. Thank you very much for the invitation. And to introduce myself, my name is Gaëlle Vilches, and I am an informatics sales specialist for Illumina, covering several territories within Europe and including Africa. So very nice to meet you all. Before really starting my presentation, for a short introduction, I would like to say that it's very well known, Illumina for the sequencing part. And nowadays we can sequence, the more and more faster, the more and more cheaper. That's really great. That's interesting, and that's opened a lot of different opportunities for all the projects all around the world. But with talking with all the different users all around the world, we've seen that now informatics is the real bottleneck. And this is really why Illumina invested in this field for the last 7 years. So here it's really like a short overview, of all the informatic portfolio Illumina have now. So just for a little story, Illumina have now more than 500, R&D people working every day just for informatics. So this is just to show to you how important this field is for us. So very briefly here in this slide, you can see that we can start even on the LIMS part, to carry, small amount, but also very, very large amount of samples, within the lab and to help to handle very large projects. We also have some tools, like BasePace that can help to, scale large projects to, like very easy to use interface, enable users to launch analysis very efficiently and quickly. We have Dragon, more and more well known now for the secondary analysis part, and we'll talk a little bit more about this. We can also have complete environment with Illumina Connected Analytics that allows users to analyze Illumina data, but it's a complete data agnostic platform, so you can analyze all the kind of data you want, create your own database, even do data science with this. And we have all the tools, more specialized like imaging for rare disease, Illumina Connected Insights for oncology. And we have now a tool called Illumina Connected Multiomics that allow users to analyze data from, RNA, proteomics, methylation, even RNA spatial data, single cell, that allows all users to have all this kind of different multiomics data. So with this, we have now what we call Illumina Connected Software that can help users, from end to end to analyze all the data. And as you can see here, we have a lot of AI tools that are more and more present within our different solutions. Talking about AI, we invest in this field too, now we have 41 people working full day, all around the world on this field. And we took, very great members within the team. To give you, little statistics here, close to 50% of the team, are first author in some papers like Nature, Science, or even Cell. Here we can see a slides, on the different big project that trust us, that trust our software, that trust our tools to handle their data. So you can see all around the world, all of this project work with us and trust us. Our tools are very well known for human, but just to mention that this is not just human-centric. You can use our tools for all the kind of species. And to give you some example, Dragon, the tool that I mentioned to analyze in the secondary data, can also handle close to every species you want, like it can be mammal, it can be plants, you have some germline pipeline, RNA pipeline, and even imputation pipeline, very well known for, low path studies. And here are some, examples on some customers that have trust us, like Texas M&A AgriLife. We also have Kazusa DNA Research Institute, working on very large plants, and also University of Sydney, that worked with us for a koala study. When we talk about this kind of big projects, we talk about cohorts, of course, and this is here a virtual circle we have, like you have your sample, you sequence them, you analyze them, you do the interpretation, you find new insights, for this sample, and like this, you have them in your population study that can help you, to find even new insights, etc., etc. Like this is thanks to that we have a virtual circle. And this is how we design our tools, like the tool like Illumina Connected Analytics, to handle this very large number of samples that can help you to either, sequence individual samples, analyze them individually and find very accurate results from them, but also analyze very, very large cohorts. Well, this is thanks to all of, this great advance that we have now, tools, Illumina connected software and different application that can be used, for all your different research. It could be for small labs, it could be for large labs, it could be for small studies or very large studies, and from all around the world. It's a platform, where you can have some collaboration, you can have some different people from different institutes, different places all around the world, that can work together and find new insights for your projects. And because data security and privacy are very important, just to mention very quickly, we have a lot of different accreditations, of course, available. We have our website, trust.illumina.com, that I invite you to have a look, to have all the details about this topic. And to finish here, I would like to invite you to scan this QR code. It is just a little form, if you want to be in contact with us, have more documentation, like we're more than happy to talk with you, with your project, how we can work together and how we can help you. So thank you very much.
Thank you, Vishals. So I think you can also just hang on the call, because we're going to just have just a short panel Q&A, one question each. Maybe Genevieve, do you want to call on Shane if she's ready now? You guys are on mute. Shane should be on the call now. Shane, are you ready?
Yes, I'm ready.
Okay, Shane is ready. So Shane is ready. Genevieve, you want to call her, then she can present.
Oh, okay. Yeah, Dr. Shane, we are ready. Tell you, make your presentation.
Okay, I just want to make sure you can see it.
Yes.
Okay. I'm going to switch my camera off while I present, but thank you very much for the invitation to present at today's meeting. It's a great honor to be able to share some of our experience in South Africa with the broader AfricaBP community. So today I'm going to be talking about the 1K SA project, and our byline is Decoding South Africa's Biodiversity. My name is Shane Murray, and I am the lead genomics coordinator for Diplomics. Diplomics stands for Distributed Platform in Omics. It has nothing to do with diplomatic relations at all. And we are in the omics area, so we're a research infrastructure that is supported by the South Africa's Department of Science, Technology and Innovation through the South African Research Infrastructure Roadmap project. And Diplomics is the only research infrastructure in the omics or molecular biology field. All the other research infrastructure supported by SAREA are in the environmental monitoring sector. We are hosted by the Center for Proteomic and Genomic Research, which is a not-for-profit research services company based in Cape Town. And CPGR itself is also supported by the DSTI through a separate instrument called the Technology Innovation Agency. So, just waiting to go to my next slide. I don't know why it's not going to the next slide. Okay, there we go. There are three different domains to diplomics. These are genomics, proteomics, and metabolomics, and also bioinformatics. And one of the mechanisms that we use within diplomics to enable access of researchers in South Africa to this high-end omics infrastructure that we help the different labs across our network maintain is a mechanism that we call a high visibility, high impact project. And there are four main objectives that we want to achieve out of a high visibility, high impact project. This is, we want to develop a community of users. We want to make sure that there is a high element of training that's part of the project in order to enable students, in particular postgrad students, to have access to the infrastructure. There's also a method development component. So we want to enable labs that are part of the project to be able to develop new methods that they can go on to offer as research services to other scientists. And then we also want to incorporate aspects of open science into the project. We are funded by the DSTI, who have an open science policy for instruments that they support. And so it's very important that we consider open science in our objectives. And so 2 years ago, the 1KSA project was set up as a high visibility, high impact project. The aim here is to sequence 1,000 biodiversity genomes over the next 3 to 5 years. And 6 of our partner labs elected to be part of this project. There are 11 genomics partners overall. For the project, we purchased Oxford Nanopore sequences, so specifically a P24, a bigger machine, which was placed at the diplomics training lab. So this was for training and for method development, but also for 1KSA. And then 4 P2 solos, which are the smaller Oxford Nanopore high-end sequencing instruments, which can take 2 PromethION flow cells each, 1 PromethION flow cell being the equivalent of sequencing a human genome at about 30 times coverage. And our partner labs in the 1KSA project are listed here on the slide. So the Institute for Microbial Biotechnology and Metagenomics at the University of the Western Cape. They are a bacterial sequencing specialist lab, and they sequence in collaboration with one of the biodiversity biobanks of South Africa biobanks. Sengen is a plant-specific genomics lab, and so they focused on plant genome sequencing. The University of Pretoria Genomics Lab is in the north of the country, and their focus is more on a wide variety of different organisms that's relevant to the community in that area. And then in the eastern side of the country, we have SIAB, which is the South African Institute for Aquatic Biodiversity, where their focus is on aquatic genome sequencing, both of fish and marine invertebrates. And then the SAMRC Genomics platform is also a partner on the project. Apologies, my slides are taking a while to go to the next one.
Yeah, I think it's frozen, Shane. We're just seeing the high visibility slide.
Okay. Do you see the next one?
Yes, please carry on.
Yes. Okay. So why did we elect to use Oxford Nanopore specifically for this project? So long read sequencing is ideally suited for de novo draft genome assembly. And this slide shows sort of a very high-end description of how Oxford Nanopore sequencing works. So simply put, DNA double strand is separated into a single strand, and then guided through a nanopore in a membrane. And as that DNA strand passes through that pore, it changes the current that is across that membrane. And this change in the current intensity is then recorded as a squiggle plot, which is on the right. And that is then base called into the base sequence of that particular DNA strand. And so if you think of the analogy of building a puzzle, if you have very big puzzle pieces, so very long sequencing reads, it is easier to build that puzzle together than using lots and lots of very small puzzle pieces. And especially when you don't have the picture on the puzzle box of how to build that puzzle. So we are going in blind. We don't have a reference genome. We are actually, in effect, creating a reference genome. And so that is why these big puzzle pieces, these long sequencing strands, are useful for the project. So what is 1KSA? It's a number of different things. It is biodiversity sequencing, as I've explained already. So this takes place in South Africa by South Africans from start to finish. So sampling, DNA extraction, library prep, sequencing, and data handling all happens in South Africa. And unfortunately, there's been a tradition up until now where South African scientists collaborate with overseas researchers, where they get sequencing done at a discounted price as part of a collaboration, but none of the know-how is transferred to those scientists. So we're trying to break that paradigm through 1KSA. 1KSA is also a framework for training and outreach, and I'll explain a little bit more about that later. It's a distributed infrastructure, so it's not just a single lab sequencing on behalf of South Africa. There's 6 different labs involved. We use a lot of method development, and we deploy open science in terms of sharing what we are doing in the project. We have draft genome assemblies, which are created, which can then be used by the scientists that nominate the species for sequencing for further research. And then also 1KSA is a centralized collection of sequencing files that can be accessed by other scientists as well. So this is the landing page for 1KSA. This is the update as of last week. On the project, we update our website on a monthly basis following our steering committee meeting. And these are some of the key milestones that we report back on. So I mentioned it's a community project, so we ask scientists to nominate species for sequencing. And to date, we've received nearly 600 eukaryotic species for sequencing. The bacteria are handled slightly differently through the biobanks, but we've sequenced, well, the UWC, our bacterial sequencing partner, has sequenced over 550 bacterial genomes. We've received about half of these samples, so that might seem like a low number, but we ask our sample contributors to contribute DNA. And so that comes with its own set of challenges in terms of getting sufficient DNA of good enough quality for long-read sequencing. And we also ask the sample contributors to make sure that they have all the necessary permits and ethics in place as well. To date, we've sequenced 212 of these eukaryote samples. And out of those, 181 have gone through the draft genome assembly, de novo genome assembly pipeline that we set up for 1KSA. And we run this in order to create some data that we publish on a species card. And it's also just really a check to make sure that there is sufficient data that is usable for the sample contributor to take further. And the species cards, we've published 165 of those to date. And you can have a look at those on our website. And the website link is listed here. So this is an overview of the project. So it starts and kind of ends also on the webpage, where scientists can fill in a form motivating their species of interest, which is then considered by the 1KSA steering committee meeting on a monthly basis. And once that sample is selected, the sample contributor is then sent an electronic sample submission form, DNA submission form. And this is, we use the electronic database REDCap for all of our project management purposes in the project. Then the sample contributor can choose which lab they would like to send their DNA to. The sample is then delivered to that lab, where it undergoes quality control and library prep. And then whole genome sequencing takes place after that, using the instruments that I described earlier. All of the metrics along the way are recorded on REDCap so that we can keep track of each sample. And following the sequencing step, the data, the base call data, is then transferred using Globus Connect to the CHPC. So the high performance computer, the central high performance computer that is also supported by the DSTI. And there we have a draft genome assembly. We have a GitHub link describing the steps we use in the draft genome assembly. We also make use of Zenodo to record all our different methods. We have a Zenodo community, a 1KSA Zenodo community, and the link for this community is available from the 1KSA website. Following the draft genome assembly step, the data is then transferred to DARISA, another local integrated infrastructure for cold storage, and the data is then shared with the sample contributor, and it is also shared with anybody who requests access to that data and is given permission through the Data Access Committee. Yeah, so that's in a nutshell the different steps of the project. If you want to find out more about any one of these steps, there is more information on the website. A big part of our project is visibility and awareness. And so the species cards help us with that. These are three examples from the early phase of the project. So the Bruder's whale on the left-hand side was sequenced by the University of Pretoria. And it is the only whale that is considered to be an inshore whale in South African waters. So the other whales that come to South African waters migrate across the oceans, whereas the Bruder's whale remains relatively close to the shore. Then the one in the middle is speckboom, which is an iconic plant in South Africa, become very popular in gardens and so on in the last few years. Yeah, there's a lot of sort of interest in this particular plant. And this was sequenced by Sengen. And then our national fish, the galjoen, which was sequenced by SciAb, is the example on the right-hand side. We also make videos about some of the different species, and these stories can be found on the website along with stickers. So these are fun representations of the different organisms, and you can see here the Bruder's whale, the speckboom, and the galjoen sticker. And these are always very, very popular at conferences and help spread the word about the project as well. So we have a community of scientists who have nominated and are still nominating species to 1KSA for genome sequencing. And this slide represents the wide diversity of organizations that these scientists work for. To date, we've had 129 scientists from all these different organizations submitting their nominations to 1KSA. So in terms of nominating a species, this is the link on the website where scientists can nominate their species of interest. There are a couple of criteria that are required. So first of all, the DNA needs to meet the quality standards. And this is a minimum of a microgram of double-stranded DNA as measured by a qubit. But ideally, we'd like to have more than that. But we do understand that in some cases that we've got to work with what's available. The purity of the DNA has to be really high, so the nanodrop ratios have to be above 2. And then the DNA also needs to be intact. So typically we wouldn't accept DNA with an average fragment size less than about 10 kb. But again, the higher that kb, the better. We also require that the organism is indigenous to South Africa. It doesn't have to be endemic to South Africa, but it needs to be indigenous to South Africa and collected within the South Africa's borders. I mentioned already that the scientists have to have permits and ethics clearance for the specimen before it's considered for 1KSA. And so typically that would be, as I mentioned, ethics. Sampling varies depending on the organism. And also depending on the application of the genome, we also require that either the discovery phase notification needs to be completed. So this is a letter that is written or a form that is sent to the Department of Forestry, Fisheries and the Environment. If commercialization is considered, then a bioprospecting permit is also required. We haven't had too many examples of that yet, though. And that permit requires that prior informed consent and a material transfer agreement is concluded with the indigenous knowledge holders of any prior art around that species. Our terms and conditions, which I'll get onto now, require an open science component to it. And so we also ask that the sample contributors develop a data management plan, or a DMP, which will describe how they will back up that data, how they will use that data, and so on. We also, at the time of submission, request metadata around the sample, where was it collected. We also request, where possible, that a voucher sample from that organism is submitted to the biobanks. And this is so that in future, the genome sequence linked to these voucher specimens can be used more widely. The terms of use are listed on the website, and we employ the saying as open as possible, as closed as necessary. And in terms of data use and publication, we have started to think about our samples as being either conservation samples or bioeconomy samples. So in the case of conservation samples, this is where having the genetic data would be really, really useful, especially for endangered species, in terms of developing tools for population management. And so here we ask that the data is uploaded onto the European Nucleotide Archive within 3 years of sequencing. We said within 3 years of sequencing in order to enable South African students. Typical period of time for a PhD study would be 3 years, to give them an opportunity to finish their sequencing and their analysis and their PhD first, before the data is made is available more openly. For bioeconomy species, especially species where there's indigenous knowledge associated with that, we have recommended that the Data Access Committee applies an embargo on the release of that data onto ENA until there is a full evaluation of any IP and indigenous knowledge about that species. And we've also started a conversation with the African Bioinformatics Institute about potentially creating a data hub for 1KSA in the long term, for the long-term use of the data that could potentially reside on an African INDSC. And I'm bringing that up now in light of the theme of today's meeting, which is powering African DSI infrastructure through deeper policy discourse. So that is probably something that will be discussed in the course of the next 2 days. We have framework and policy around the Data Access Committee and how you can apply for that, and the data transfer agreement. We've got a standard template that we use. We also request that scientists use the acknowledgement statement on our website at the time of publication, and also that 1KSA sample nominators are happy to take part in any publicity and reporting requirements that we have. We've also asked the sample contributors to track any intellectual property that might be developed, and in the very long term, any benefit sharing would go to South Africa and to the indigenous knowledge holders, but also we would request that a very small portion, still to be determined, would come into an education trust that we would like to establish for future biodiversity education, biodiversity genomics education purposes. But that's all sort of longer term plans at this stage. How am I doing for time? Okay, I'm not going to go into this in a lot of detail, but I just wanted to highlight that in terms of lab method development, there are 3 components to it. The DNA quality control, or the QC requirements met. What is the optimal average fragment size for doing library prep? For library prep, we've done a lot of method development around multiplexing, so smaller genomes that couldn't be multiplexed on one flow cell. And then also for sequencing, we've done a lot of optimization about washing and reloading in order to get the optimal data output from a particular flow cell. And that is an ongoing process, and those protocols are constantly being updated. In terms of the bioinformatics workflow, I mentioned it's on GitHub. We initially developed workflow in Nextflow, using Fly as the assembler and then using Racon as the polishing step. But we found that we weren't able to assemble within the requirements on the CHPC, genomes bigger than about 3 gigabases in size. And then last year, about this time last year, Hi-Fism was modified for use with Oxford Nanopore sequencing, and that has been a game changer for this project because it's allowed us to assemble increasingly larger genomes, particularly plant genomes. And that's really allowed us to push through, and we've had about 80 genomes now that have been assembled using Hi-Fism. So we also have done method development around which sort of assembler to use. And just from a very high level perspective, the first QC step that we carry out after looking at the past base called reads is to do a KMO peak analysis. So this is looking at the frequency of a particular KMO in the data. And if you get a peak in the graph, that's an indication that there's sufficient data for carrying out the assembly. There's a lot of filtering and trimming that one can do to try and get that KMO peak. But after all those have been explored and exhausted, then we have to go back to the lab and do more sequencing in order to get that KMO peak. So that's been a useful tool from a QC perspective. Then we look at the size of the predicted genome, and we use the GOAT genome on a tree tool from EBI for that. And if the expected genome size is bigger than 3 gigabases, then we will immediately go to Hi-Fism and use that tool. If it's less than 3 gigabases, and if the sequencing in 50, which I'll discuss in the next slide, is less than 10 gigabases, then we'll use we'll still use the Fly tool with Racon polishing. Okay, so I'm going to just quickly discuss some of the results. And the key things that we look at overall is the amount of data produced. So this is one of the metrics that we look at is the read N50. So this is the size of the smallest read where that particular read and all of the larger reads make up at least 50% of all of the sequencing reads. We also look at contiguity of the draft genome assembly. So the contig in 50 is one of the metrics we look at. So that's the length of the contig where all the contigs of that length and longer cover at least half of the assembly. And we set ourselves in the beginning sort of a lowish bar of about 30 kilobases as being the minimum contig in 50. But these days we rarely see that. We're always getting contig in 50s in the megabase region. We also look at completeness, and here we use the BUSCO or Benchmarking Universal Single Copy Orthologs tool. And here we are looking for a BUSCO score of greater than 95%. So here I'm presenting the first 100 species of 1KSA. So we're now up to 180, more than 180 that have been assembled. So this represents the beginning of the project. And in the slide on the left-hand side are the sequencing N50 counts. So these are the number of species with a read N50 in the bins indicated on the x-axis. And you'll see that the read N50s are sort of probably the average is just less than 10 in this slide. I don't have a graph, but increasingly we are getting read N50s sort of more towards 25 and higher. In terms of the contig N50, so this is from the draft assembly. We also, as I mentioned, getting most of our assemblies on the megabase range. But we are now getting more and more with Hi-Fism coming on board that are about 100 megabases or more. And on that note, one of our recent species that we carried out the assembly for is the Clan William cedar. This is endemic to the Cedarburg region in the Western Cape. So this is a region about 3 to 4 hours north of Nassau. And this was such an important tree that this whole region, this whole mountainous region was named after the tree. But unfortunately, the populations have suffered due to, in the early days of colonialism, being used for shipbuilding, particularly masts of ships. And now with climate change and also with the increased number of fires that come through the region have really severely impacted the populations of this tree. So having a reference genome, having a long read sequencing data set is really an important tool for population genetics of this tree. And so we were very pleased that we were able to do the sequencing to generate a really good sequencing in 50, just over 18 kilobases. We used Hi-Fism for the genome assembly. The genome length was what we expected, so just less than 10 gigabases in size with a very good BUSCO score. And it's our best assembly in 50 to date, so over 300 megabases, which we are very happy with a very low contact number. So this particular tree, unfortunately, was caught in a fire in December last year, and there's only about 10% of the tree left over. And everyone is hoping that it's going to be able to rejuvenate and to grow back. This tree is estimated to be over 490 years old as well. So it really is, it's quite, yeah, it's going to be a very useful tool for analysis of this species going forward. Okay, so just quickly about in terms of the BUSCO scores. So this graph shows our different groups of organisms that we've looked at. And in most cases, you'll see our BUSCO scores, when we count the single and the duplicated genes together, are over 95%. The only one group, the amphibian group, we've only done one species to date, and that's the western leopard toad. And there's the sticker picture over there of the western leopard toad. Here we really struggled with this. There was lots of filtering and back and forth, but eventually the best we could do in terms of the BUSCO score was 88%. But we did start off with not great DNA. This is a highly endangered toad species, which is found in the Cape region. So to go on to training, so in Diplomics, we use a train-the-trainer model. So back in 2023, we held a training workshop for our Oxford Nanopore labs. And these labs, the people who came for the training then went back to their home institutes. And to date, they've run over 14 workshops on Oxford Nanopore library prep and sequencing. And this has reached more than 140 participants. We've also supported AfricaBP's Open Institute Southern African workshops over the last 3 years, the first two in Pretoria and the last one last year in 2025 in Cape Town. And last year, we also added our 1KSA genome assembly workshops. We ran 2 last year, and actually next week, we're going to be running our first, what we're calling 1KSA Academy, which is library prep, sequencing, and the genome assembly workshop back-to-back in over 1 week. Not only do we provide training for the community of labs, and also the community of scientists bringing their samples to 1KSA for sequencing, we also are upskilling our training labs, and we're planning for this August a PORE-C workshop. So PORE-C is the high C equivalent for Oxford Nanopore, whereby the labs, the 1KSA labs, will be trained in running PORE-C workflows, which will enable them to then offer PORE-C as a service to 1KSA sample contributors who might want to take their genomes further in terms of getting a chromosome level genome assembly, depending on their research needs going forward. A few points about open science. So as I mentioned, we employ the saying as open as possible, as closed as necessary. And so when we were designing the 1KSA project, we tried to align it to a number of the different UNESCO recommendations as possible. So in terms of open methods, we have used the website to highlight access to the methods along with the 1KSA REDCap records and protocols in our Zenodo community. In terms of open resources, we enable access to sequencing through the Data Access Committee and through sample nominations. We enable access to the national infrastructure, computing infrastructure, the CHPC and DARISA through the project. We have a data management plan, which is on our Zenodo community, and you can access that through the website and through the community. And we also promote training to these different resources. We use open source through demonstrating the 1KSA workflow on GitHub. We have open data. We have a data management plan. We have a data access committee. We create a visibility to the data through our species cards and videos. And ultimately, we want the data to be on an open repository such as ENA after 3 years. And then finally, open access. We also promote publication of 1KSA genome assemblies in open access journals. So that brings me to the end of my talk. Thank you very much for your attention. I would just like to thank the labs that are involved in the 1KSA project. They're listed here. Thank you to them for all the hard work and all the method development and all the troubleshooting that's gone into this project to date. And then if you would like to find out more about Diplomics or about 1KSA, please go to our website, either the Diplomics website or the 1KSA website. And also, we're very active on LinkedIn. We advertise a lot of our training on LinkedIn, so please go to LinkedIn as well and follow us there. Thank you very much for your attention.
Right. Thank you so much, Shane, for the extensive presentation. So maybe Genevieve, I don't know if we have, in a sense, Shane, could you please just hang on the call so we can be able to have like collect all the Q&A at once?
Sure.
Genevieve, I don't think we have Innocent yet on the call, and we have Nkaba.
Hello,
Olu.
thank God.
Yes.
Can you hear me?
Yeah, I can hear you.
Yeah, okay. Please, because people have been here for quite some time and it's very hot, we want to take some people out for tea break. People have been seated there as well. Yes, so we just want.
So can we spend just 5 minutes to collect the question and answer and then round up, if that's okay, so that this session will be closed, and then when we return, we start a new session. Is that okay?
Yes, that's fine.
Okay. All right. Okay, I think
Yes,
we're going to skip Innocent because I don't think he's here. Maybe Genevieve, is there any question for Nikki, Boybit, Vilches, and Shane on site before I start asking those online? Can you check with those on site if there are questions?
Please, if you have your questions, we want to take them. Questions?
Start with those online first. We have a few questions here. Obona, actually I have Riyadh. So Riyadh, can you ask your question? You've been asked to unmute. Riyadh Bofra, can you please ask your question? You can unmute now.
Hello, everyone.
Yes, please go ahead.
In the context of initiatives such as the Africa BioGenome Project, and in line with our vision in Algeria, genomic data sovereignty is a central concept. So could you please clarify what genomic data is physically stored? I'm speaking to Gael. What genomic data is physically stored, Illumina cloud, third-party cloud, or on-premise?
Yeah.
And also whether a fully on-premise solution is available without any external data transfer, and how you ensure data confidentiality and full national control over sensitive genomic information. Thank you.
Yeah. Okay, so I think Riyadh, you asked more of the questions that will be discussed tomorrow. So around sovereignty, access and benefit sharing and everything. So, but I don't think this is more for Vicious and Nikki Adams. This is not for you, right? So I'm gonna take this if that's okay.
Well, I think it.
Does it align with your? Okay, you can go ahead then.
Yeah, I'll try to be as much concise as possible,
Yeah.
because that's a big one.
I
So thank
know.
you first for this question. So for the different solution we have within Illumina, well, we want to be flexible. So there is fully in cloud solution. You can be hybrid. You can be on-prem. Of course, some complete environments, and some project that it's managing very large codes, of course, it's more cloud. We have several options that can be possible here too. So we have mostly of our tool based on AWS, and you can select after the different location physically where you want your data to be. And we have, like I said, 20 of different accreditation, like the 27001, et cetera. You have how the data encrypted, the single sign-on, like we have absolutely everything to make sure that your data are secured and that we are compliant. If you have a very specific project, you want to check for a specific accreditation whatsoever, we are more than open to talk with you with your project. Do not hesitate to come back to us, and we can be pretty flexible on this one.
Thank you,
Thank you very much.
Nikki, do you
Thank
have
you.
any response to that as well?
So yeah, thanks for your question. From an Oxford Nanopore point of view, the data is fully owned by the customer. No data is streamed at all to any of our cloud-based servers or anything like that. So it is completely owned by the customer, and the customer then would, have complete control of where and how that data is analyzed. So yeah. Even our cloud services as well, if we do have any cloud services available for any analysis, that is also, there's a 2-week, period where that data then gets taken off. But also we don't do any human type analysis in the cloud system either. So it's mostly for bacterial genomes or any other sort of genomes that happens in the cloud. So yeah, so it's completely owned by the customer.
Thank you, Nikki.
Thank
Shane,
you.
do you want to go for it? Then before I ask you guys the more specific questions we have online here.
Sure. So within the 1KSA project, we haven't used any cloud services at all. So the data is, as Nikki said, it's the data comes from the different labs. We move it to the central computing infrastructure using Globus, but it's all password protected, and then it's backed up cold storage on Derisa. And
just to add that Derisa is the national.
Yes.
Just because there are people here who don't know what Derisa is.
Yeah. Sorry. Yeah.
The
So
national
Derisa
infrastructure in South Africa. Yeah.
Yeah. It is the national sort of computing infrastructure for South Africa. So NICUS is the high level name, National Infrastructure for Computing in South Africa, NICUS. So the CHPC is our central high performance computer where we do the analysis. We run the pipeline for doing 1KSA. So we bring the data onto the CHPC, we run it. Typically that takes about a 2-week period, depending on the size of the genome and how many times we have to push the wall time out. And then it gets transferred from there onto Derisa and backed up. We also keep a backup locally on the servers here at the CPGR as well. So we have 2 copies of the data as well, just in case there are any issues with one of the copies.
Okay, so I'm going to ask you, there's a lot of questions on the chat, so during your free time, please feel free to respond to them, but just 1 or 2 questions so we can try to kind of have a tea break time.
Thank you very much.
So thank you, Riyad. I'm going to just ask some questions that are being posted here. The first question mainly is to Nikki Adams. I think I can't find it again. That's a question by Sally, I presume. Sally is asking, Oxford Nanopore has been expanding access to sequencing technology across the continent, but access and capability are not the same thing. Where do you see the most significant gaps between having the technology and having the conditions that make that technology meaningful, and what distinguishes the context where adoption actually leads to lasting institutional capacity? So I think this is kind of a long one, if you can remember everything.
Yeah, that is quite a long question, and I actually can't even see it on the chat at the moment.
It's at 11:13. So if you scroll up to 11:13 by Sally, Katie.
Okay.
Otherwise, I can ask Sally to Sally, I can give you access to just ask so you can provide more context. You can unmute Sally. You can unmute Sally if you want. Okay, you can, otherwise you can go to
Oh, okay.
Okay,
I've been
Sally is here.
able to unmute. Thanks so much.
Hi
Thank God.
there.
Hi, Nikki. And sorry for the long question.
That's okay.
I've just been listening to everyone's presentations, and I've just been thinking about, access and capability. And as we all know, they're not the same thing. And so what I was thinking about is pretty much, or what I'm asking is, where do you see the most significant gaps between actually having the technology and having the conditions that make this technology meaningful? And in the longer context, what I'd be interested in hearing is what really distinguishes adoption that actually leads to lasting institutional capacity, because there are instances where you do see the technology available in certain countries, but there's no capacity to uptake it. So that's what I'm trying to get at. Thanks.
Okay. Yeah. So yeah, accessing the technology, I think one of the, from what I, am aware of, is actually being able to get to some of our, third-party reagents that is needed to actually implement our workflows. So from a logistical point of view, Oxford Nanopore is actively trying to get more channel partners in Africa to get access to these third-party reagents needed for the workflows to actually be, implemented at site. So that's the one thing. So from a practical point of view, doing the work, it's sort of logistical, I would say, issues. Our actual workflows themselves are really simple and easy to implement. So once they have those, the agents, etc., training, I can even train online, etc. We've got a lot of support at the moment, as I said, channel partners, including myself and one of our other colleagues in Tanzania. We are available to actually then implement those workflows for the customers. Coming to the sort of dry lab things, or dry lab part of things, we do generate a lot of data. So, you get a lot of rich data from Oxford Nanopore Technologies, and, having the sort of correct sort of server capacity, as Shane said, they need to sort of be able to move this data around, being able to actually store that data, back up their data. That can also sometimes be a little bit of a sort of, problem with regards to accessing and actually using the data. But from an analysis point of view, we do have the Epitome platform that allows even wet lab scientists to do the analysis. So I think the sort of hindrance is more of actually being able to manage the amount of data that gets generated from our technology. That I think can be one of the sort of, part of the puzzle that we need to maybe, make sure the customers are aware of when taking on, this data sets that we can actually generate. I hope that answers your question.
Thank you,
May
Nikki.
Sorry, I can't find the put up my hand button. May I add to that?
Yeah.
Thank God.
Yeah.
I don't use Zoom very much anymore. Hi, Sally, how are you? I just wanted to add to that. So with my Diplomics hat on, not with my 1KSA hat on. So we see in the South African context, there is a huge appetite for new sequencing instruments, and people buy them and they put them in their lab and everything looks great for the first year. And then the wheels fall off, and then the sustainability becomes an issue, sustainability of continuing the sequencing with that instrument. And so that's partly why the mechanism, this high visibility, high impact project mechanism, was instituted, to try and break that. But it is a huge challenge. It's a huge challenge to make sure that that sequencing infrastructure is sustainable, not just from a training and an initial sequencing perspective, but long term. So, and it's multifaceted, as Nikki explained. It's, it's about what to do with the data, where to transfer the data. And one aspect that a lot of scientists overlook is also the requirement for maintenance of the machine and warranties, so on, going forward, which is not cheap. And so getting the sequencing to be sustainable, to generate some kind of research services, is the only kind of way to go. So there's some kind of income coming in to pay those warranties and so on going forward. So it's a multifaceted approach that's required, and all of these things need to be considered, I would say, at the start of a new sequencing initiative.
Thank you, Shane. So I think the next question, I have a question for Illumina. Maybe Virtues, you can also make some comments on the previous question asked to Nikki, and then you can now also answer this question. So one is asking if Illumina offer cloud service, and if so, how does that work essentially? So you can blend those questions together and respond to them.
A lot of things to talk about. Well, we have some cloud services, yes, and they can be, like I mentioned before, in several kind of levels. You can have a very simple push button solution, or you can have a very more complex environment when you can close to do everything. And I fully agree that, and I'm bioinformatician, like from studies, like this is really my field. And we have seen, especially nowadays, like pricing for a component is going crazy. We have to find solution, how we can help users to analyze data. And for example, the different challenges we have is, for example, and talking about very large project, is the analysis time. And this is also why, we have solutions like Dragon, to just give you an idea of how Dragon is fast with just one server. You can analyze a whole genome sequencing 30x alignment plus all the kind of variant colors you want, small and large, in less than 30 minutes, which is pretty rare record. So for example, with this kind of technology, we try to help users like generate information from the sequencing with Dragon, and after have this very large environment where you can find insights from your cohorts, from all the samples you had, and for your project, basically. I think that's
yeah. Thank you, Gail. Then I think one final question. This was asked to Gail Illumina, but I think we're going to spread it out and everyone will have a chance to respond, and then we can wrap up the session. So there are lots of questions on the chat. So Shane, Nikki, and Gail, please feel free to respond to them. We can't ask all of them at the moment. So someone is asking, beyond sequencing, the real challenge is scaling bioinformatics across uneven African institutions. How can systems be designed to remain interoperable and sustainable when electricity, bandwidth, expertise vary so widely, without deepening inequities in genomic access? And to add my own response to that, my own question to that, is that in a nutshell, again, is how do we also see, can we ever get to a point where I can be walking on the road or in the field and take my device and sequence a genome by taking a picture, and then the genome is sequenced? So this is more like I don't know if you guys see that happening. I think that is particularly to Illumina and also to ONT, where you can sequence a genome by skipping all the wet lab steps, all the library prep, all the pre-analytical step. So do you see that possible? And I think, in my view, that is one way to solve this kind of problem long term that Davis Marabada asked. Putting it in context is, many years ago, when you take pictures, you go to a photo lab and then you take pictures. It used to be very sophisticated, but as cameras and phone became the norm, you could go on a trip and then you take photos. You don't really need to have that kind of. So, I mean, it'd be nice to respond to that, to see. I know it's like a fiction, but can we ever get to that point? And if you can put that in the context of Davis Marabada's question, that would also be helpful. Maybe I'll start with you, Vishels, and then we go to Oxford Nanopore and then to Shane.
All right, that's a lot of questions. All right. So first, about system where electricity bandwidth, can be a challenge. So first, we have some tools, of course, to check, that data is transferred, in the right way. Like, of course, we're already working in this since a very long time, with customers that can have some bandwidth issues. So we have a lot of different tools to check, of course, the integrity of the data, and we have the tools talking about security, blah, 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 to encrypt the data correctly. After, for a word where you can do the sequencing without any library prep and this kind of things, well, I do not know if you heard about our new technology, TruePath, when you can actually now on the NovAseq X, just put the DNA like this, and it will sequence your data without any library prep. So how we can make the life, easier of all the different labs, before and after the sequencing, that's the goal that we are trying to achieve today.
Yeah. And I think, is that also the same technology where you don't need to store things in -80 or cold temperature or liquid, something like that? I know there's this type where you don't need to store any of the thing in a cold chain or something like that. I don't know if that's the same machine, the Illumina machine as well.
We have more and more of our, core consumable that are now room temperature.
Okay, all right, perfect.
Because, well, there's a lot of things, there is also the carbon footprint and blah, 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 everything to consider. So, yeah.
Yeah, I mean, in that case, then we're probably getting closer to the point where we can sequence by just taking a picture of the plant on the field and the genome is sequenced.
The dream.
Over to you, Nikki Adams.
Thanks so much. So I'm gonna sort of touch on one of your comments about sort of, with the electricity, the internet issues that we have, a lot of the Oxford Nanopore technology workflows right now, we've always moved towards wanting to be sort of a mobile sequencing system. So we've done that. We've been out in the field without electricity, using power packs, etc., to actually sequence things at the site of, epidemics, etc. So I think from a ONT point of view, we've already ticked some of those boxes. A lot of our epitome workflows, we can download them before we go off. We have all the databases online on your local computer, and you do all your analysis locally. So definitely, I think from a ONT point of view, we want to go that way. We want to make it easily and accessible for everybody. That is our motto. But from a sequencing from a picture, I would definitely have to speak to developers about that, but we definitely want to make sure that we can provide the easiest workflows as possible for our customers for implementation. We've got some of the quickest library prep workflows that there are out there to actually generate, libraries within an hour with minimal additional components, etc., that is needed. So for sure, sequencing anything by anyone, anywhere, that is what we continuously strive to do. And we already doing that. But yeah, let me see how far we can get with just popping something into a tube. And yeah, but yeah, definitely ONTs on that stream as well. Over to you, Shane.
Shane, do you have any comment on this? Thank you, Nikki.
Yes, yeah. So just to sort of expand on what Nikki said, I think already, depending on what you want to do the sequencing for, so it's things like eDNA analysis, barcoding, you can do that in the field already. You just need a little lab in a bag, or something like a bento lab, which is a portable structure for the DNA extraction steps, which are increasingly getting easier and easier. You can use a little dipstick method now, which we trialed actually in the training lab here, and it works really well on water analysis. But you do need to do that PCR step for barcoding specifically. So you do need some kind of infrastructure, but solo solutions, there are lots of opportunities around that. So definitely you can do field-based targeted analysis. That's already happening. And that's where projects like 1KSA, like AfricaBP, where the reference genome is created, that becomes tools for doing targeted design, primer design for barcoding, for example. And so, it's the bigger infrastructure of the project that will enable this kind of more targeted analysis. Another really exciting area that we'd love to explore is forensic genomics analysis. So in cases of poaching, and a lot of that happens in South Africa, of all different types of organisms, increasingly more of plants as well, and then being able to do, analysis at border posts and that kind of thing on DNA to show that it's this particular species that has been poached and found.
Thank you, Nikki. Sharon?
Yeah.
I think hopefully in a few years' time when we do assemble again, we can kind of reassess whether we're able to get to the point of the picture as well. Given that all of these bits and pieces are there, is perhaps to kind of put them all together. And I'm sure 20 years ago, if someone said there would be a phone, you can take a picture and then you can upload it to wherever cloud, then someone might have said it wouldn't be possible. But if you look at the fact that we have the ability that's for us to predict, like ChatGPT, OpenAI, and all of those things, is because of the amount of data from but from the point where you have the mobile systems, particularly. So I'm hopefully we will get there at some point. In the interest of time, I am not going to read the rest of the questions, Sheniki girl. So please feel free to go to the chat and comment on them, and we'll just take a short break and then reconvene for the next session.
Okay.
Thank you so much.
Thank you very much.
Thank
Bye-bye.
you.
See you all in about
Enjoy your
30
tea
minutes
break.
time.
Okay.
Yeah.
Bye.
Hello, all. I've just been told that the tea break is for 15 minutes, so we'll be back in 15 minutes time.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has three varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we move on to the Promethean. Promethean offers about 7 terabyte of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you to do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budget. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genlab.ng or cgl_ont@genlab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal datasets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has three varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabytes of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistics team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budget. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genlab.ng or cgl_ont@genlab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
International vibes.
Let me when you move like that on me. Let me be, don't stop, don't talk, just feel, let me yeah, right there, move, no stress, no rules, you're always talking, I'm cool, low lights, one dream, your body moving on beat, slow,
Say you feel it,
I feel it,
say you're ready,
I'm ready,
let me be,
let me be, let me
be,
when you grind that slow on me,
let me be,
let me be, tonight we free, no sleep, no love talk, just motion, love. Let me be, when you move like that on me. Let me, don't stop, don't talk, just feel. Don't stop.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, Innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has three varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabytes of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budgets. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genelab.ng or tgl_ont@genelab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, We're here with you every step of the way. Vega by PacBio. Hi.
Dr. Tango, are you there?
Yes, I'm here. Thanks. So I think
Okay, Presidency, are you there too?
with Densi. So let me make him a co-host. All right. Professor Dunsi, you're a co-host now. You can start your video as well.
Yes, as we start this session, I wish to recognize the presence of the 7th Vice Chancellor of the University of Port Harcourt, Professor Joseph Atubokiki Ajenka, who's a very great Vice Chancellor in this university and is a lover of science. He promoted science in this university to a scale that is quite appreciable, and we all love him. We want Professor Ajenka to give us a few words of advice, few words of encouragement.
Well, good evening. It's almost afternoon now in Nigeria. Good afternoon, or good morning, or good evening, wherever you are. This African BioGenome Project, Open Institute for Genomics and Bioinformatics, West Africa Regional Workshop, is something which I consider very important in the life of this university and indeed other universities. And we'd like to congratulate Professor Julian Osuji for driving this initiative. The way the world is developing, we must pay attention to biogenome, genomics in general, in such a way that we can develop a lot of things out of it. Because it's an open field where a lot can be developed. And Africa is a virgin field for this. So we'd like to encourage all participants on this conference to contribute their knowledge and experience in such a way that this field of specialization is properly developed for the benefit of society. So once again, I thank all participants for attending and congratulate the keynote speaker in particular. I met a part of that keynote. It was well presented. We'd like to encourage everybody, all the presenters that will yet come on stream. We look forward to your papers and your presentations. I know my friend, Professor Vincent Demio is also going to speak this afternoon. So once again, you're all welcome to this important workshop and conference. Thank you very much.
Yes, can we switch on over to the next session? I guess it's time for it. For those ones, I welcome you, please, to this wonderful workshop and conference. For those ones who will be presenting their presentation on the poster presentation and oral, I hope you will be there. You remember that you only have 5 minutes to do the presentation, and then if the 5 minutes elapses, we'll have to cut you so that we'll be able to meet our timing for the keynote speakers after this session, please. That we'll not have issues with time. And so if Dr. Peter Chukura is around, can he come up with his 5 minutes oral presentation, please?
Yeah, Dr. Peter Chukura, are you on site or online? If you're online, please, I can't see you online, but if you're on site, then please go to the podium. Professor Suji, do we have, is Peter on site?
I have checked the the participants are not thinking he is there, and he is the only author to the paper. So I don't know, you don't have any colleague that will present the paper for him. So we can skip him and move to the next person. Yeah.
Are you here, Peter Chukura?
I'm not thinking so that he's there.
I don't think
Dr. Peter Chukura, are you here? Okay.
Yeah.
Can we just person?
Yeah, can we switch on to Paul A.S. Soremi? Are you there, please? Either on ground or in person or online, so that you can do your presentation for 5 minutes, please.
Yeah, so Paul, if you are here, please put on the chat box. I can't see Paul.
Dr. Paul Soremi. Dr. Paul Soremi, are you here?
Okay, it looks like Paul. It looks
he's
like
not
Paul
there also.
It looks like he's online. He's on site.
He's on site.
We've not seen him. We'll check the next person.
Yeah,
Fatima.
Elagi.
Okay.
Fatima Elagi, are you ready?
Yeah, so maybe I think if we don't get Fatima, we have Nkaba here. I think we try to call earlier on before the break. So maybe we can take Nkaba and then switch into the oral presentation if that's okay.
Okay,
right.
but yeah, Dr. Angon, maybe what we'll also do is that for those ones who are either on ground or they are online, they can just put on chat that they are here so that we don't waste our time calling their names and with their so that the Nkaba will be presented while we start sort out for those who are presenting. Yeah.
Nkaba just messaged me that they are on site, actually.
It's okay.
We called them earlier. Okay, maybe Olarewaju. Is Olarewaju on site then? We can get the Nkaba presentation for 10 minutes.
Yeah, Olarewaju is on site.
Okay, please let him speak. Yeah.
Okay.
And Janto is online. So Professor Odense, once Olarewaju finishes and finishes Q&A, we can go to Janto Ndalo, who is online.
Okay, no worries.
So we can't see the on-site podium or something. So could you please, we're just seeing the poster.
Okay, good afternoon, everyone. Distinguished Professor, the Vice Chancellor, please allow me to stand on the already established protocol. It's a pleasure to be here. I'm from AfricaBP, a proud partner of African BP initiative, and I'm here to give a very short presentation on the importance of the partnership between academia and industry for sequencing breakthrough in Africa, because my presentation would actually look at things from the industry perspective. But before I go into the presentation, please allow me to quickly give a short introduction about who we are as a company. So we are Africa's foremost company, and for over 2 decades now, we've been, serving the life science community across sub-Saharan Africa with quality laboratory products and services. We have our headquarters in South Africa, Pretoria to be precise, and over the years we spread across the continent with additional 8 physical offices in Uganda, Nigeria here, Cameroon, Kenya, and Tanzania. And beyond, distribution of equipment, we also offer laboratory services such as Sanger DNA sequencing and next generation sequencing using the PacBio Hi-Fi long-range sequencing technology. So getting right into the presentation, so I would like to take us through the memory lane of how the genomic revolution all started. Certain human genome project, like I said earlier, on my perspective will come from the history. So the Human Genome Project, was funded by the U.S. National Institute of Health and the Department of Energy. It started in 1990 to be completed within a space of 15 years. And it was budgeted to be completed with approximately $3 billion USD using the Sanger DNA sequencing technology. But along the line, a key major player in the project, Craig Venter, actually, pulled out of the project because he wasn't quite impressed with the pace at which the project was unfolding. So he started up his own company called Celera Genomics, privately funded, obviously. And it was also aimed, to compete with the Human Genome Project, with a plan to sell subscription, access human genome data, which along the line didn't really go well. But a very key important thing from that was the fact that, is it up? Okay, sorry, we have a little bit of glitch here. It's not showing still. Maybe you can use duplicates. Yeah, it's not showing on the screen yet. Yeah. Can you give us? Okay, I just send it to you. Okay, let me see if I can do that. Yeah, if you have a flash drive, maybe you can just copy it out. You don't have it. You don't have it. No, I don't have a flash with me yet. It's detecting it. I'm sorry, there's a little bit of technical glitch here. Is it showing
but
now?
I think to be able to help Olarewaju, are you able to join on Zoom via your laptop? Is that possible?
Oh, okay, yeah.
So log into your Zoom and then share your laptop. They'll share it. I think that's the way to do it, while the technical IT team resolve the issue.
Okay,
So
I'll just do that.
Yeah.
Okay. So I'm trying to join on my computer here. Oh, yes, I can. Okay. So I'm going to connect. You don't have a flash, right?
So, Professor Udonsi, I think there would be, I understand there will be a bit of, because they are trying to get the laptop and the stuff on. So maybe, okay, I can see he's now sharing.
Okay.
All right, okay, all right, go ahead.
Okay, it's all right.
Yeah, I think. Olarewaju? Okay, please carry on. We can see your screen. Please carry on and start talking. Can the IT person unmute? IT, please unmute.
So as I was saying earlier on, before we had the little technical glitch, I want to just take a quick look through the genomic revolution and the impact that the industry played in terms of growth. That's good. So it all started, setting the stage was the Human Genome Project, which was, publicly funded by the U.S. National Institute of Health and the Department of Energy. It started in 1990 with a aim to actually complete it within 15 years, of approximately $3 billion USD. And the major technology to be used then was the Sanger DNA sequencing technology. And a major key player, the pulled out of that project, all because it wasn't really pleased about the pace that the project was unfolding, of course, due to public bureaucracy. So he started the company called Celera Genomics, privately funded, with the aim to also, compete with the Human Project, and a plan to actually sell subscription access to human genome data, which, like I said earlier on, didn't really go down well. But a key thing out of Celera Genomics was the fact that they developed whole genome shotgun sequencing technology, which was actually quite effective and faster than the DNA sequencing technology. And because this technology makes use of the massive parallel sequencing approach. And then down the line, there was, let me try and get rid of this. Yes, so there was some sort of, competition between the publicly led initiative and the private led initiative. But what came out of that was the fact that it was a healthy competition that led to the completion of the human genome way ahead of schedule, approximately around 13 years. And even the spending was even way lesser than what they budgeted, which was approximately about $1 billion USD. And in 2000, both sectors came together, combined the data sets from the Human Genome Project and the Cellular Genomics-led Human Genome Project to come up with a near complete human genome of about 92% coverage. And through this data, the researchers were able to answer questions that were previously difficult to answer. And this also even led to new research questions in the field of omics, such as what we now know as the transcriptomics, the epigenome, metagenomics, just to mention a few. And importantly, this genomic field has even contributed. A recent publication from Science noted that the field has actually contributed from inception of about close to $1 trillion USD to the US economy. Now, recently, a lot of innovative approach has been developed by the industry in order to make the Human Genome Project complete. In the sense that initially the reference genome was about 92% coverage, but with the T2T consortium, we now have the complete human genome, which was released in 2002, with the use of several long-read sequencing technology to sequence the remaining 8% that was missing in the initial reference genome. And according to the Science publication, it was really noted there that the PacBio Hi-Fi reads were used in resolving complex structural variation and gaps in initial reference human genome, and it was of all the long-read sequence technology that was used, a major star of the whole project was the PacBio Smart technology, making it the core technology for reference quality genome. So if you're looking into sequencing different species to get a reference quality human genome, then PacBio Smart technology is actually the go-to technology for that. Now, in the context of Africa, a lot of people, as you're all aware, that Africa is the heart of genetic diversity. In fact, African population harbors more genetic variation than all non-African population combined. And this is simply explained by our longer population history. The continent is known as the origin of human being. And this also, has given rise to more mutations and diversification to occur. But unfortunately, if you look at the global, genomic data set landscape, the continent as a whole is grossly underrepresented. Less than about 2% of the human genome data we have out there were actually derived from the African population. And this genetic diversity doesn't only occur. Scientists are not just saying this in the human African population alone, but it cuts across other biological species as well, such as economically important crops and even in animal and livestock. But why is this so important? So this high level and rich biodiversity is actually very important in the sense that it serves as a gold mine for potentials to fuel innovations and growth in both healthcare and precision medicine, agriculture and food security, conservation of our genetic resources, and more importantly, our bioeconomy. Now, looking at the African data in action, of course, despite the fact that we are underrepresented, we've actually been able to, the African data has actually been used for so many things in the space of healthcare, human health, in agriculture and conservation efforts. A very good example, so I'll just be giving, just because of the limited time I have, I'll just be giving a few examples of success stories where African data set have actually been used, to translate to products with, impact in our society. The first one is the H3 Africa SNP genotyping array, which was, developed as a result of a collaboration between the H3 Africa Initiative and the industry partner. Before this array, the SNP genotyping array that was in the market were actually developed and designed using data sets from mostly Caucasian population. But with this array now, researchers are able to associate genomic variants with various diseases peculiar to African and African ancestry population, and also helps to understand the risk factors for these diseases. Beyond this research use, this panel actually has the potential, to help us develop, diagnostic tools, therapeutics, and even cure for diseases that are, endemic in African population. In the space of agriculture, I would agree with most people here that, in fact, a lot of our data sets from, our rich biodiversity in terms of plants and animals actually being used to develop improved crop varieties and livestock breeds, not only in the African context, but even used elsewhere in another continent. And this has been made happen by the collaboration between industry partners such as seed companies and international agricultural research institutions to generate genetic data, technology transfer, and even commercialize some of these products. Now, looking back at what all these success stories and what exactly are the enablers of successful partnership between academia, the industry, or the public and private partnership. The first one is data sharing frameworks. And I think this is like the main theme of this conference. And I really, really look forward to a lot of, diverse perspective from speakers that will definitely talk on this. But just to highlight that there's a need for clear agreement on DSI ownership and access that align with global frameworks, such as the Nagoya Protocol. Because according to a CGIAR 2024 report, current multilateral benefit sharing scheme that require tracking and tracing of DSI being used in particular product do not fully reflect the contribution of all the upstream DSI. So what this simply means that as rich as our biodiversity is in Africa, we just do not want to keep churning out, genetic data for developed countries to, reap the value from those data. But also, let's come together whereby we can, both private-public partnership and even the public, the policymaker and the public sector come together to position Africa as not as a data source, as well as contribute to the value that we can actually generate from this data we have in our genetic resources. The next is shared infrastructure. You will all agree with me that the global funding purse is getting smaller and shrinking by the day. So this is actually very, very important. There's a need for the public sector, the academia, and the industry to come together to collaborate and share resources. A very good example of that is the collaboration between the University of South Africa and AfricaBP BioTech, to jointly acquire a third generation sequencing equipment called the PacBio Review. And with this platform here in Africa, we've been able to sequence a lot of our endemic plant and animal species. I also use this opportunity to share with everyone that we will also be deploying similar technology to the West African region. In the coming months, we'll be having the PacBio Vega here in Nigeria, which will serve as an hub for researchers across the West African region to gain access to this state-of-the-art facility. So if you have interesting projects in the area of genomics, epigenomics, metagenomics, just to name a few, please, we'll be very much happy to collaborate with you and help you achieve your research goal. The next one is the capacity building and talent mobility. I mean, during the previous presentations, someone made something a very striking note that in Africa, and actually that we're actually generating a lot of genetic data, even way more at the pace that is faster than the capacity that we have to actually utilize this data to translate them to meaningful products in our society. And for us to actually do that, we need to, as much as we are investing in this infrastructure, we're also building, our human resources capacity, especially our bioinformaticians. We need to, invest in them, train them, build their capacity so that they are not just only generating data, but they are also making, mining those data using the power of AI to translate them to something beneficial, not just to the scientific community, but, to the society at large. The last key enablers that I will touch upon here is the co-development funding model. This is also very important. We need to come together, share resources, financial resources together, come up with joint grants for sequencing African genetic resources. A very good example was the collaboration between AfricaBP, IITA, AfricaBP, and the Malibu Foundation, where resources were put together, to do a chromosome scale genome assembly annotation of the white star apple, popularly known as Agbalumo, in the West African region. So with this, the data is publicly available for researchers and breeders, to conserve these fruits and also make the livelihood of smallholder farmers that make a living out of this. So to end my presentation, I would like to leave with this note that tomorrow's Africa genomic discovery starts today. And for us to, fully utilize the potential of a diverse genetic resources, there has to be a strong collaboration between the academia, the industry, the private and the public sector. Thank you very much for your time.
So
thank God, yes.
Yeah, so I think maybe we can just take questions for Olarewaju and then slide into the oral presentation.
Yeah.
So yeah. So if there are any questions.
Any questions?
So I think one question, Olarewaju. I have one. If I don't know if there are any questions on site. Genevieve, are there questions on site?
Yeah.
Okay. You take the on-site question and then let me know. Then I can ask the online questions.
Yeah. Okay. Thank you for the presentation. My question is going to be practical in the sense that African biodiversity and genomic data, and you talked about data sharing. I would like to ask in practical terms, how has this data sharing helped improve our agriculture sector and healthcare? Now we're talking about personalized medicine. What is the fate of Africa and specifically Nigeria in meeting up with this trend in the medical sciences? Thank you. Thank you very much, Prof. Yeah, so in the scientific space, I mean, for us to make groundbreaking discovery, we need to work with a lot of data. So that's why it's important that we share data with the scientific community so that people can see what's going on in our lab and also use whatever data we have in our lab to also, make meaningful inferences that could lead to something tangible from their own lab as well. So I think data sharing is very important. We cannot remove that out of the important is the fact that data sovereignty. We need to be sure that the benefit, the value that these data are translating into countries or continents that are actually, contributing to this data, also benefiting from it, the value from those value. It could be monetary, it could be non-monetary. So I think that's something that we need to sit down. I think what will be also discussed because of this conference. So data sharing is important, but what is equally important is the equitable benefit sharing across all the stakeholders. Yeah, thank you.
Good afternoon, everyone. Thank you very much for your presentation. So I wanted to talk about the H3 African P genotyping array. So this is the first time I'm actually hearing about it. So please, could you further explain or use for African, the disease is unique to the African population?
That's a very good question. So the H3 SNP genotyping array was, developed as a result of the collaboration. It's one of the many projects that was, carried out by the H3 Africa initiative. And this was actually done in collaboration with the industry partner. So before that array was designed or developed, like I said, we used to have SNP arrays that we had developed using data from the Caucasians, the white population. Do you get? And when you use such data in our population, oftentimes we meet out pathogenic variants that were not covered in those populations. So with this SNP genotyping specifically designed for the African population, we researchers are able, to do some sort of association studies, like the SNPs that are associated with diseases that are peculiar in our whole population. And from that, we can also understand the risk factors of those SNPs eventually. And this could lead to diagnostic tools and even cure for some of these diseases. I hope I've been able to answer your question. Good. Thank you.
Okay. Yes, thank you. You can just ask on a question on the chat box.
Online.
So that's
yeah. Okay. Okay. So Olorewaju, I think this one is from me, basically, so that we can wrap up in the interest of time. So could you please expand more? Olorewaju, are you there?
Yes, I'm with you.
Yeah. So could you please expand more what you and your organizations are doing in terms of because one of the things we're looking at is kind of having long reads and especially in the West Africa region, because that will be very helpful in increasing the sequencing throughput from the region. And ultimately, we know that not all countries can have sequencer, but at least if each region have their own sequencer to get us to chromosome scale, that is one of the optimum things we're looking at. So in the next few minutes, if you can expand more about that and what the future is looking like in terms of that direction in the context of the long read and PacBio and all of those things, that will also be very helpful.
Yeah, thank you very much. Thank God. So for us, I think about tech. Like I said earlier, we are deeply rooted in the African continent. We have spread across sub-Saharan Africa, and in fact, we, one way or the other, pioneering sequencing our genetic biodiversity right on the continent. So beyond centralizing most of our sequencing efforts in South Africa, we also want to expand into every other subsidiaries that we have. Like I said earlier on, we have locally here in Nigeria. Through that, we'll be able to, to attend the needs of the researchers across the West African region, within them, better pricing points, and even bigger capacity. So researchers across the region can come to our facility, organize workshops and training to build them, to gain access to this technology. So those are the things we're doing. And sincere, when it comes to sequencing our genetic resources, there were quite a number of limitations with the traditional short-read sequencing platform, especially when it comes to sequencing regions that have repetitive sequences or regions with high GC content. So, but with this technology, those kind of limitations are not there. We have more coverage of genomes, regardless of the species you're working with. You're able to have better coverage. In fact, the quality metrics are very great. We have better Q-score. We have better coverage depth when using this type of technology. So these are like state-of-the-art technology that we are bringing closer to scientists across the sub-Saharan region with any of the researchers who want to make use of this technology.
Yeah, thank you, Olarewaju. So, and if there are no more questions on site, and we would like to move to the oral presentation, and thank you, Olarewaju. I think we'll also see more of you during the course of the week as well. Over to you, Udensi. I believe we have some presenters. We have one of the presenter is online.
Yes,
Ntando.
Janto is around.
Yeah, okay, over to you, Janto. Please share your screen.
We still have one more person that wants to ask a question. Please, can we take the question?
That's fine. That's fine.
Good afternoon, everyone. Mr. Oluwafobi, please, from your presentation, having your topic as from genomic data to discovery, let me say my suggestion, because of where you're coming from. You know that Nigeria has a lot of plant biodiversity. We have a rich biodiversity of plants, and in our societies today, you see wherever you go, you see some herbal stores and thereabouts, all those things. And you know that in health is wealth, and the world is going back to trado medicine, So I will suggest and I will ask that use your position in your capacity, as you guys are bringing in some of those things for sequencing in Nigeria as very, very soon. For people to as well gather some of these trado medicine practitioners. Let us have a lot of genomic data on herbal medicine, to preserve them and to use it ourselves, so that some of this petroleum, what do I say? Some of this medicine that are gotten from that synthetic products, we reduce, so that we make use of our natural plant products. We train more of phyto doctors, train them in phytomedicine, so that we will live long. That's my suggestion. Please put it in your data when you get there. you tell them. Thank you very much.
That's just a suggestion, not really a question. It's a suggestion. So I guess another word will take it over to his industry and then discuss that, isn't it?
Yeah. Professor Odense, I just sent you the names of people who are online. So in that order, that is the timeline they are supposed to present. So if you can call them in the order.
That on the chat?
Private chat. Yeah, on the private chat, yeah. So I think the first person is Babangida Sanusi, then followed by Vandada.
Okay, let me just check.
They are all online.
Yeah, he got, okay. Okay, so Babangida can come and present his presentation, please. Babangida, are you there?
Babangida Sanusi, please unmute. Please unmute Babangida Sanusi. You have the co-host rights. Otherwise, we'll skip you and then move to the next speaker. Okay, I think he has unmuted.
Vandana, if he's there.
No, Babangida has unmuted. So Babangida, do you want to just make 5-minute presentation of your oral?
Okay, yes, let me share my slides.
Please carry on, Babangida.
Yes, I'm waiting.
Please start presenting. You have 5 minutes.
You have 5 minutes, please.
Okay. Okay. Good morning, everyone. My name is Babangida Sanusi from the Department of Biochemistry at Ahmadu University's area. I will be presenting my work on in silico transcriptome-based identification of African ancestry diagnostic survey. In silico transcriptome-based identification of African ancestry-specific breast cancer biomarker. This work was carried out in collaboration with my colleagues from Department of Biochemistry, Biotechnology, Pharmacology, Institute of Fundamental Medicine, and Biology, Transform Federal University, and Sinka Bioscience. Briefly, this presentation will cover introduction, objectives, research questions, study design, result and discussion, and then finally, I will conclude. By the way of introduction, breast cancer is one of the most common cancer in women globally, and the hidden cause of death. In Nigeria, it accounts for about 35.2% of all cancer cases, with increasing incidence. The major challenge is late diagnosis, and this is partly due to lack of population-specific biomarkers. This highlights the need for an African ancestry-focused molecular markers that would be used to galvanize Africans. The objective of the work is to identify a differentially expressed genes between the tumor and normal tissue among the African cohorts, and then to evaluate their biomarker potential. Then to explore the biological functions and the pathways associated to the identified differentially expressed genes or possible biomarkers. To study design, what we did is we first of all identify a transcriptome data obtained from gene expression omnivores, some published databases. We collected the informations and then utilized Xena browser to carry out differential gene expression analysis, where we identify differentially expressed genes. The differentially expressed genes, we identify the genes that are significantly associated with the patient survival. That is from among the differentially expressed genes, we identify those genes that were significantly associated to the patient survival based on Kaplan-Meier survival analysis. And then we also went ahead to identify from the significantly differentially expressed biomarkers. We went ahead to identify possible biomarkers, and as well as those genes that have gene product associated to membrane protein. From the identified genes, we went ahead to identify the possible protein-protein interactions. And, from there, we also went ahead to identify the pathways in which these identified biomarkers were related to. So what we did is we obtained data from gene expression omnibus and some published articles where we carried out differential gene expression analysis using a Genome browser. Then we identified the differentially expressed genes from the different published paper, as well as from the gene expression data from the gene expression omnibus. After identifying the differentially expressed genes, we collected the ones that are most significantly expressed. The most significantly expressed genes were collected, and then we determined the survival analysis of how these genes or expression of those genes are related to the survival of the patients. We were able to identify quite a number of genes. See, some of them include CC, DC 163, RNF1, 35, NIN, G1, quite a number of them. And then we predict the biomarker potentials of those genes, out of which we identify 5 genes, which include BIRC7, PLK1, CDK6, HLGA. Most significantly, we identify BIRC7 and PLK1 to be the most highly potential candidate marker genes, and some of those genes were also linked to membrane proteins, and that gave them the upper hand to be potentially considered as biomarkers in cancer. So we went ahead to identify the interactions analysis using interaction study, which shows how these dysregulated genes in cancer interact at protein level. So we use a cytoscape to identify the hop genes, those ones that are central in the disease progression, and possibly these genes will potentially be utilized as biomarkers or a critical therapeutic target. And of course, we were able to identify some of those genes that were effectively to have the potential biomarker, activity like KI-EE1328 and VLP1 as indicated. And then the pathway enrichment analysis shows that these genes were involved in quite a number of molecular functions, cellular component, and biological processes that are linked to cancer progression, cell regulation mechanisms. And then we have the integrated pathway, which shows all the interacting genes in the comprehensive pathway network. And this provides us a system-level understanding of brain cancer biology in African population, highlighting the key pathway driving tumor development. In conclusion, we were able to identify 3 key genes, which include BIRC7PLK1, KIA1328, were identified as promising biomarkers which has strong potential to distinguish tumor from normal tissues and are significantly associated with breast cancer patient survival, suggesting their usefulness in advancing precision medicine for African breast cancer population. Thank you.
Thank you, Babangida, for that presentation. But you just hang on, okay, for questions when other colleagues have already presented, please, okay? Just gather yourself together for the question they will ask you, right? So can we invite Vandana, please, if he or she is on site or online?
Vandana is online. So
okay, please.
let me, Vandana, you can unmute now. So who is sharing?
Hi, everyone. Can you hear me?
Yes.
Yes, we can hear you. Go ahead.
Yeah, let me share my screen. I hope it's visible.
Yeah. So start. Yeah, you can start sharing now.
Yeah. So hi everyone, I'm Vandana. I'm a PhD scholar from Manipal University, Jaipur, India, and I'm working under the guidance of Professor Prashant Silva.
I can't see the screen.
I'm sorry?
I can't see the screen. Have you shared?
Yes, it is shared.
So Vandana, just can you share your screen now?
Yeah.
So
yeah, perfect. You can go ahead, please.
Yeah, it's visible, right?
Yeah, for sure.
Yes, please. Yeah, go ahead.
Yeah, so today I'll be presenting a recent work
So
on
is there a chance there's a background noise, Vandana? Do you just want to move out a bit from where the noise is, please?
Yeah. Is it okay now?
Okay, go
yes,
ahead,
it's
please.
perfect. Carry on.
Yeah, so today I'll be talking about our recent research on distinct genetic variants from whole exome sequencing of syndromic ARMs. Anorectal malformations are congenital anomalies that affects the normal development of anus and rectum, and this mainly arises due to the defects in hindgut development. So the global incidence of this disease is 1 in 5,000 live births. And there are two types of ARM mainly, that is the syndromic ARMs and the non-syndromic ones. The syndromic ARMs will have associated anomalies, which is mentioned in the figure, and the non-syndromic will have no associated anomalies and it will be isolated defects. And it is reported that in India, around 15% of all the 15% of the surgeries that is happening in the pediatric department is from anorectal malformation. Even though this is clinically relevant and many surgical improvements are being done, genetic studies are still essential to understand the variants which are causing this disease. This happens mainly due to lack of genomic data, especially in countries like India. So for this, we have 8 samples, which are syndromic ARM samples with high ARM cases, and the phenotypes are mentioned here. So what we did is we have patients with syndromic ARMs, and we have collected the samples with the consent, and the DNA is extracted and the whole exome sequencing is done. And then we used our in-house pipeline called Convex to identify the variants, which uses 4 variant callers, and the consensus variance is taken. And we will validate with the ClinVar and with some filtration steps. At the same time, we use 2 AI-based technologies, the GenAssure and the ION, where the VCF files from the Convex is uploaded with the HPO terms of the disease, and they will keep the variants associated with the disease. From all the 3, we'll get a final list of variants, and then we did prioritized variants validated through IgE.
So these are the 20 variants that we got after the filtration and prioritization from all the 8 samples. So I'll be explaining them in the next slides in brief. So we get a set of variants which affects the embryonic patterning and signaling pathways. So this mainly affects the sonic head shock signaling pathway and the notch signaling, which are essential for cloacal formation and which the defects can lead to anorectal malformations. Next is the epithelial morphogenesis and developmental communication genes. These regulates the cytoskeletal dynamics and epithelial morphogenesis, and we also got some variants in the GJB2 and CCD-C50, which are mainly affecting the hearing loss. But in most of the ARM patients, we can see hearing loss also. Then we also found some genes in the metabolic integrity and cytokine signaling. This affects the mitochondrial function and can lead to complex phenotypes. So we also have some cilia-mediated signaling and tissue remodeling variants in the genes PKHD1 and PKD1. These cilia-dependent pathways are mainly important for SHH pathways, which will lead to cloacal formation. So defects in this can lead to abnormal hindgut and cloacal development. We have also done some comparative analysis of the variants in multiple data sets. So out of the 20 variants which we got, as I mentioned, we used a AI-based technology called ION. So 9 variants were prioritized by ION because some of the variants we got from this 20 were listed as VUS or conflicting interpretations from ClinVar. So, but ION was able to classify them as benign or likely pathogenic or pathogenic one. We also tried to compare our results with the Korean dataset, which used.
Vandana, please, just to inform you that we have just 1 minute to wrap up, please.
Yeah. So we couldn't find any common variants between the Korean study and the Jiva study, which was done on ARM. So this was the gene interaction network that we got, and this PKHD1, PKD1, and PKD2. These 3 genes were most important ones, and these are linked to the SHH notch and ciliary pathways, and these are mainly important for the cloacal and midline formation. We also identified 170 CNVs across all the 8 samples, and we found that one CNV which is in the chromosome 22 was having a variant from the MIF2, which is relevant for mitochondrial function, which may suggest that mitochondrial dysfunction can also lead to ARM. So in conclusion, we have prioritized 20 variants from 8 samples, and we used AI-based technologies to classify the variants which was not classified by ClinVar, and we are now trying to validate these results using functional assays. So I thank my guide and all the lab members and the collaborators. Thank you.
Oh, thank you, Vandana, for that presentation. Like I said, you just hang on, okay, for questions after we have wrapped up with the other presenters, please. Can we have a faker that is Sihamet? Yes. Okay, to the podium. If you are online, please. Is he there? Hello. Is it El Faka? Is it?
Yeah, Sihame is online. So Sihame, could you please join and unmute and share your screen, please? You now have the co-host right. Unmute. Sihame Elfaka. Elfakri. Could you please unmute Sihame? Yeah, I think he's not responding. So
Okay, so we can
let's move to the next.
Janto, please.
Yeah.
Janto, are you there?
Yes, I'm here.
Okay, please, you can share your screen, please.
Okay. Are you able to see my screen?
For sure, yeah. Just go ahead, please.
Okay.
Please.
Very good afternoon to you all. Thank you so much for this opportunity. My name is Jen Tandalo, and I work with World Agroforestry Center, specifically with the African Orphan Crop Consortium Lab and Molecular Lab, where we sequence, assemble, and annotate African orphan crops together with agroforestry tree species. So far, we've been able to publish reference genomes for about 7 species already. As you can see on the screen. We also have a sequence data for almost 60 plus tree and crop species. Among the crops we've worked on is Moringa oleifera, which is a multi-purpose fast-growing crop that is native to the Himalayas, but now widespread across the tropics. Moringa is known for its multiple benefits. Actually, most of its parts are useful either as a source of nutrition or agroforestry plants, a potential source of bioenergy, and also a source of medicine. Now, in our analysis, the gap was that there is not so much enough information, genetic diversity information of Moringa, and for that purpose, we intended to genotype the 19 Moringa accession, 19 Moringa provenances, and these were obtained from 3 regions: Africa, Southeast Asia, and Caribbean, and from 8 countries. That is from 8 countries. And in this assessing, we were able to find out the level of genetic diversity existing within the 19 Moringa oleifera provenances, and also to elucidate the Moringa oleifera genetic structuring. And this information then helped us to identify Moringa co-collection and for conservation and distribution. Now, these are the main collection sites of the Moringa tree, Moringa samples. Now, in our methodology, we actually used isolate 2 plant DNA isolation kit as indicated on the screen. And then later on, we sequenced using DATSEQ technology, which is a genotyping by sequencing platform. Now, in our results, we could, through phylogenetic analysis, you can clearly see that the Southeastern Asia provenances were so distinct from the rest of the population and had the highest relative genetic distance. And now looking at the African population, Malawi population had the highest relative genetic distance from the rest of the populations. Therefore, this one was clearly indicating that Malawi must have a unique introduction at a given time point, and it involved multiple gene pools introduced. Here we can see a population tree and an individual sample tree. Now, next, we also explored principal coordinate analysis, and the output was corroborating with what we got from the principal coordinate from the phylogenetic analysis, where the Malawi samples kept on distinguishing itself from the rest of the African populations, and also Pangasinan and Kokalokun Philippine population being so distinct, that being as a reason of proximity to the center of origin of Moringa, which I've said earlier on. Now, looking at genetic structuring of Moringa, we can clearly see that if all these populations were divided into 2 main populations, which was our best case, and then we'll have Moringa, Malawi population segregating itself from the rest of the populations. If we were to extrapolate and look at K equals 3 and K equals to 4, then we clearly see that Malawi is still distincting itself from the rest of the population. So that means that Malawi population is very diverse and actually very different from the rest of the samples that we worked with. Looking at the analysis of molecular variants, we can see that there was a higher gene flow within population as compared to among population. That is can be explained because of cross-pollination. Now, in conclusion, we actually discovered that Malawi population is the most distinct and diverse population in the African collection, and therefore it becomes our center of focus in terms of seed collection, conservation, and distribution. Apart from that, we also realized that the high throughput platform DATSEQ is very good in the studying of the African orphan crops. We look forward to doing a functional genomics through genetic-wide associated studies and also marker-associated studies. Something that came out unique is the clustering of the West African and Caribbean population together, which is a unique finding, and we need to look into it even further. I would like to appreciate World Agroforestry Center team that are working with me, African BioGenome Project, for giving me this opportunity, and all the co-authors. Thank you so much.
Yeah, thank you, Gento, for that presentation. Like we just said, that you just hang on, okay, for questions. For me, I have questions to ask you, but not now, right? I will invite Musari Kahil.
Professor Dente, I think someone said the last speaker said he has just a battery that's going to last for 30 minutes. So is there a chance we can consider that speaker, then we'll move to the next? So that will be Roland Efoli on behalf of Peter. So Roland, do you want to, because his battery is going to run down. So Roland, you have the co-host right. Please unmute and speak.
Yes, Roland?
Yeah, I'm trying to pull up my, all right. Thank you very much. Good afternoon, everyone. I'm grateful for the opportunity to be here to make this presentation. My name is Roland Efoli. On behalf of the co-authors, I'll be making this presentation on genome-wide characterization of AP2 gene family in Dioscura species and its expression dynamics under abiotic stress. The AP2 gene family plays an essential role in plant growth and development. Further research has shown that this gene family is known to regulate plant tolerance to stress responses. These are some of the AP2 subfamily members, and we'll be seeing some of these abbreviations as we go on. As we well know, yam is a staple crop in West Africa, vulnerable to biotic and abiotic stresses. The AP2 transcription factors remain uncharacterized in the Dioscura species, and this study has the potential to provide molecular insights into breeding stress-resilient yam varieties. And so this research was done with at this basic level with these objectives to identify and characterize AP2 subfamily genes in the genomes of various Dioscura species to infer evolutionary relationships among the identified Dioscura AP2 proteins and to analyze the relative expression patterns of the AP2 subfamily genes in tissues of Dioscura species under drought stress. So we began this research by using the appropriate bioinformatic tools to identify, as we can see, 47 sequences made it through the criteria, one of which is a 70 to 100% identity to the AP2 subfamily genes already characterized in Arabidopsis thaliana. And as we can observe, we can see the distribution of the AP2 subfamily genes that we identified in the Dioscura species. The next thing we did was we had to check for all of the 47 sequences to ensure that these sequences contain the characteristic AP2 dual domain, because the AP2 subfamily of the AP2 ERF superfamily contains the dual AP2 domain. And as we can see, all the 47 sequences contained this, characteristic dual domain. We proceeded to check for the motif analysis. And as is observed, we can see that motifs 1 and 2 were present in all of the Dioscura species and all of the sequences that we identified. And below we can see the amino sequence, the amino acid sequences, that represents the motifs. we proceeded to do a phylogenetic analysis to infer the evolutionary relationships between in this gene family among the genes. And we found that, as we can see, the WRI gene clustered to form a clade, and we had the AIL, the CRL5 closely clustered together. After this, we did move on to do a promoter region analysis, and we identified some elements that are known to be found in that implicates, stress resilience. And as you can see, it's Asterix. We identified ABRE, GBox, amongst others. Afterwards, we did an expression profile analysis of the AP2 genes in the scoria tissues under drought stress.
Please, Roland, please know that you have just 1 minute to round up, please.
Thank you, Prof. The drought was induced by completely withdrawing water for 21 days. And as we observed from the results we have, we observed an upregulation in the leaves of alata across the three genes that we used for this analysis. And we noticed a downregulation in D rotundata leaves that were used in this analysis. So to quickly give a summary, 47 AP2 members were identified and every other thing else that has been said. And for the want of time, it's important to know this is a research that is still ongoing, and we intend to analyze the expression profiles across different drought time points, and the candidate genes that would emerge from this will further undergo a functional validation. Thank you so much.
Yeah, thank you, Roland, for that presentation. Can we invite Musari Kahid for his presentation, please? Musari, are you there?
Yeah, let me check. I think Musari was here earlier on. Just let me give him. Yeah, Musari Kahid. I need to give him the speaking rights. Yeah, Musari is here. Musari, you have co-host speaking right now. You can put on your video and unmute. Khalid, yeah, go ahead.
Hello, sorry, I'm having battery challenge. So I would like to present at the end.
Okay, at the end. Okay, that's fine. That's fine.
So can Onyeka Kingsley Omosu, if he is there, whether on ground or online?
So is Onyeka online on site? I saw him earlier online, but he's no longer online. So Genevieve, is Onyeka online on site rather?
Can you hear us from the end, please?
Can you hear me?
Yeah.
Can you hear me?
Yes.
Okay, that's fine. Onyeka is not physically here. He wishes to present his paper virtually. And
yeah,
well,
yes. Tango, please, could you enable him present, please? Hello, Dr. Tango, please.
Onyeka, are you online?
He's not online. So let's move to the next speaker. I can't find him online.
Try to find
Fakadu, are you there? Is Fakadu on this call?
Yeah, Fakadu is here, so he's gonna speak now.
Enable him to present, please.
Fakadu, please unmute yourself.
Is he there?
Yeah, hello.
Okay, go ahead, please. Thank you.
Okay, thank you very much.
Please. Yep.
Good afternoon, everyone. I am Fakadu Gamechu from Ethiopia, working on public health entomology research at Ethiopian Public Health Institute, Addis Ababa, and a PhD student at Addis Ababa University. So thank you for the invitation. Now I'm going to present my research finding on genomic analysis revealed selection pressure and the emergence of molecular insecticide resistance in malaria vector Anopheles gambiae from Western Ethiopia. So, is that visible? Sorry.
Yes, please carry on.
Yeah, thank you very much. So, as a background,
Is it
central
possible
to malaria
better?
transmission.
It's okay. Yeah.
Okay, very good. Anopheles gambiae species complex, a group of morphologically similar species, exhibits remarkable genetic diversity and ecological adaptability. So nowadays, recent advances in genomic technologies have enabled deeper insight into population structure and evolutionary dynamics. So due to many factors, mosquitoes just change from time to time. For example, mosquitoes are exposed to high levels of insecticides in many ways, and the changing of insecticides from time to time exerted high strengths of selection pressure on mosquito species. And so we plan to explore the single nucleotide polymorphism mutations and population structure for Anopheles gambiae sensu stricto from Western Ethiopia, and to discover genes or any novel under positive selection. And the study sites were the western part of Ethiopia, Gambela and Benishangul Gumuz. And initially, the major malaria vector is known in Ethiopia. Yeah. Are you hearing me? Sorry.
Okay.
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 So, Anopheles arabiensis was the major malaria vector until today. But we just, after whole genome sequence conducted at Sanger sequencing, we just uncovered that this other malaria vector, Anopheles gambiae sensu stricto, confirmed to be present in Ethiopia. So we did this by taxon designation, by ancestral informative marker, comparing this Anopheles gambiae with neighboring countries' species. That, like, we compared with Peony molecular form and Anopheles gambiae, Kilusi, and others in neighboring countries. For example, here dominantly we compared with Kenyans. So we did principal component analysis and never joining tree analysis for population structure and compared this taxon with these neighboring countries. And finally, we confirmed that this is gambiae or Anopheles gambiae sensu stricto in Ethiopia. So the other one is we just conducted target site resistance, and we just analyzed and got many findings. And within our findings, we got candidate genes and known gene variants. For example, L995F, which is known as KDR West, and L995S KDR is reported here, and this mosquito species having very high frequencies in these regions. And we conducted this in two cohorts, major season and minor seasons. So, and the other one is the metabolic gene resistance. And for this, we analyzed copy number variants for many gene families. For example, cytochrome P455 gene families, carboxyl monooxygenase, and glutathione S transferase class. And finally, we got with excellent findings, as it was reported in some Eastern Central African countries as well. And we did all genome selection for haplotype selection within all chromosome contigs. And here, for instance, we got on 2 other chromosome contigs for all cohorts, that is with a strong selection haplotypes under these localities. And also for the same is true for chromosome arm to L. So in conclusion, we confirmed the presence of this species in Western Ethiopia, having KDR West and KDR East, that is ranged moderate to high frequencies within these localities. But within our analysis, there was no treat for resistance to dieldrin and acetylcholinesterase 1 that we haven't identified or investigated the resistant variants that have been yet registered and reported in other African countries. So there are also, this study identifies a newly emerging signal of selection across multiple loci spanning new gene loci, and within which acetyl-CoA is one within this, but which is not experimentally and operationally valid. Because due time, I'll finalize on this, and I would like to acknowledge all these authors. Professor Lemu Golasa from Addis Ababa University is one of my major advisors, like Kelly Bennett, Dr. Chris Carson, Alistair Mile, Perseid Ugas, and others are all those authors that I would like to acknowledge. Thank you so much.
Yeah, thank you, Musari Kahil, for that presentation. I'm thinking that Onyeka is not around, because if he's not around, we go for the quick discussion.
I think he just joined. So
Onyeka.
All right. Can you
in the next 5 minutes, please?
Yes, I'm here now.
Yeah, in the next 5 minutes, please.
Could you please make a 5 minutes oral presentation? You have the co-host right to
Yes,
share.
please. I shared my presentation. I wanted it to be shared from that end so that I can present. I shared with Professor Suji. I don't know if he shared it.
Can you share with me then? Is it a Google slide or an attachment?
Yes, it's on slide.
Okay, can you send me send me a chat now with the slide so I can, but I don't have the URL, the Google slide URL.
Okay, let me see. Let me see if I can share from my side.
Okay. Okay. Yeah, that's fine. I can see it. Yeah. Please carry on. 5 minutes.
Is it coming up?
Yeah, we can see it quite clearly. Are you the one presenting World Agroforestry?
No.
No, it's not. It's not me.
Okay, just one moment.
So it's actually shared to you on WhatsApp. Can I have your WhatsApp number?
On the chat. So I just sent you a message on Zoom. Who is sharing then?
No, that was
because I'm finding it difficult to share for me.
Who shared this?
Okay, can you stop your screen? Can you stop the screen so that I can share mine?
Okay, I'm just trying to find the person who is sharing their screen. So Janto, could you please stop sharing your screen, please? Okay, I think I can. All right. Okay, carry on. You can share now.
All right. So, okay, I'm finding it difficult. Let me share with you so that I can share from your side.
Okay, that's fine. So I just sent you a Zoom chat now. So if it's a Google, if it's a URL, then you can just send it. I sent you a private chat too.
All right. So you want me to share it to you on this chat?
I sent you a private URL, a private message, so you can just reply to me. I'm the one with AfricaBP STMEC. So I just sent you. Just check your messages.
So I have it on a PowerPoint, PDF on PowerPoint.
Oh, it's on PowerPoint. Okay, can you send it to my email then?
Yes, please.
You have my email?
No.
I'm just sending it to you on the private chat now. So because I can't. Okay, I just sent it to you, so you can now forward it there.
Is it possible that we can be taking a little question while you sort that out?
Yeah. Yeah, I think that's fine. We can do that.
Yeah.
We can do that.
Yeah.
Yeah.
I don't know if I don't think I have not seen any question on the chat box. So I don't know if there's any person who has a question online or on site.
Yeah, I think for online there are some questions. There's a question from Kaoki. So maybe the presenters that presented can please have their videos on, and because we can just have the panel Q&A now and then much later. So someone is asking, Adedapo is asking, is Moringa native to Africa? If no, has the species been sequenced from its area of origin, and how is it different from your data? So, and I think this is to Janto. So Janto, do you want to answer this?
But Janto has answered the question on that chat room.
Okay, maybe she has a platform to answer again. So.
Yeah, she has answered that question on the chat room and gave reference to what the answer. Yeah.
All right. So then another question is Kauki is saying, I have a question for the presenter. Are you planning to extend the study on other sources across Africa, like on the North African countries? So I don't know if Kauki, is this directed to a specific presenter? So maybe I can, Kauki, are you there? I can just make you a co-host so you can maybe ask your question in. You can unmute now. You can ask your question verbally if you may.
Maybe while we wait for him or her, I wanted to ask Janto a question. Yeah.
Please carry on, Kauki. Okay, it looks like we lost Kauki. Adedapo was asking on that question. I think this is he said for the Alata presenter. So the question is, what is the implication of the upregulated genes within your work? What is the implication of yam breeding? So can the presenter please respond to this? You can unmute yourself.
Roland, are you still on this call?
Hello, sorry. The question for Pekadu?
No,
for
it's
Alata. Roland.
No, thank you, sorry, because my internet is unstable.
Yeah, this is for Roland, which I think because he doesn't have much connection, so he's kind of stepped out. But I think we can go on site now, because we don't have additional questions. So
well,
Genevieve,
I would have just quickly asked John one question concerning Moringa, especially when you said that there's one, I don't know, the population you said was a bit distant, but the question I wanted to ask is, since it's the phytochem that is of importance to that plant, did you take time to do a phytochemistry of the one that was deviant from other ones?
So I think that is to Jando.
Yes, yes. Thank you so much for that question. And yes, we have been trying to do nutritional analysis and also chemical component, and that is still unpublished. But we are willing to go beyond that through a genome-wide associated studies, and that will be to do with functional genomics.
Because the concern here is because it is a plant that people have claimed that can treat as many diseases as possible, and the efficacy of the plant is because of the phytochem. So if you do a robust analysis of the phytochemistry, okay, to be able to unravel the real difference, that may make sense. Why GWAX is good, but what would be the benefit to the industry would be of importance to the population.
Yes, what is happening is that we have done phytochemistry by one of our students in this, a PhD student in this lab, and we really want to relate the nutritional component and the medicinal component with the genetic information. That way we are able to do proper selection.
It's okay. It's all right. Yeah.
Okay. Yeah, so I have the presentation from Onyeka. I'm just trying to pull it up just now. If you give me a minute, I could just do that.
Can we take questions from on-site if there
Yes,
are?
please. Yes, please.
Please, any questions from the floor? Questions, please. Feel free to ask your questions.
So I think we're ready for.
Yeah, the online, they can carry on, please.
Okay, all right, we can take Onika's presentation now. So I'm just kind of bringing it up just now. Just one moment.
Okay, can you?
Yeah, I'm trying to share it.
All right.
I'm trying to share it just now. Okay, I am sharing the screen. All right, here we go. So
yeah,
I would do this slide view. Okay, here you go.
Great. Thank you so much. I am Onyeka Nwosu. I'm the technical advisor to the Director General Chief Executive of National Biosafety Management Agency. I want to make a presentation on the policy and governance implications for digital sequence information. That is a DSI-enabled synthetic biology applications. We want to look at the policy and governance implications for DSI that enables synthetic biology applications. Next. So let's set the scene. You can now imagine that DNA strands are being transformed into digital codes and then making them into binary sequences of AI. And considering that if our DNA strands are now going digital, you also consider the global community being interconnected by data networks. So they can also be able to access these digital codes. So based on that, what then happens when biology becomes data and data becomes the power? So I think the best approach to it is by balancing the innovation of digital biology, balancing the innovation of equity for those that will access it, increase the biosafety process, and then create a global access for all these. I think this is the key. So we are talking about synthetic biology being digital and how people can access it. What are the policy challenges and the implications? Next. So what is DSI and why does it matter? Digital sequence information refers to genetic sequence data that are stored digitally. So our genetic sequence data that are stored digitally, and this data can include our DNA or RNA sequences, our protein sequences, our bioinformatic databases, and metadata linked to organisms. So these are part of the genetic sequences that can be stored digitally. And when they are stored digitally, we refer to them as digital sequence information. Next. So why does DSI matter? Today, genes can be downloaded, edited, and then synthesized. It is in that process of synthesizing gene that you get digitally is what we refer to most of the times as synthetic biology. And then you can redesign the genes you have downloaded without accessing a biological material. That means you are not even accessing the biological material to extract the DNA, to sequence and so. You are now taking the sequences that are available digitally to do your work. And what is the real world impact? It helps in disease surveillance and vaccine design. It can help in crop improvement and conservation, and it can help us to develop synthetic microbes for several industrial activities. Next. So the question I should ask now, should genetic data be treated like physical resources? We have our physical biological materials. Now we are talking of DSI, digital sequences. Are we supposed to treat the way we handle the way we handle our physical biological materials the same way we are going to handle our digital sequences? This is a question for us to ask and understand the impact and policy implications of DSI. Next. So what is synthetic biology? Synthetic biology is a further development and new dimension of modern biotechnology that combines science, technology, and engineering to facilitate and accelerate the understanding, the design, the redesign, the manufacture or modification of genetic materials, living organisms, or biological systems. So what does this synthetic biology in redesigning our biological system? It's usually aided by artificial intelligence, machine learning, or digital sequence information. So synthetic biology are aided by most of these sequences that we have stored digitally, and then improved it using AI structures and so. So these are how AI and digital sequence information helps synthetic biology. Now, DSI aids synthetic biology. We have understood what DSI is and what synthetic biology is. What are now the policy implications? Remembering that I asked a question, do we supposed to handle our physical, the digital genetic materials the way we handle the physical biological materials? So next. Next, please. So DSI plus synthetic biology, no doubt, is a new bioeconomy. Because if you look at it, our traditional biology, what you will do is you collect your organisms, you go to the lab and study it. Now, modern biology is talking about downloading the sequences that are already digital. You download, you design them digitally with the aid of AI, and then use them to build biologically, to build biological systems. That is synthetic biology. Building on the sequences you got online and using it to design or redesign a new organism on itself. So what are the key applications where this is applied? Just like I've mentioned before, you can do it in mRNA, you can apply it for mRNA vaccines, biofuels, engineer bacteria for medicine and agriculture, climate smart agriculture, and then produce synthetic enzymes for our industry. Next. So why do the government care? Because whenever you are talking of policies, is where governments come in. So why do governments supposed to care for a DSI that enables synthetic biology? It is because DSI is now a strategic asset influencing our health, our food security, our economy, and nationally, our national competitiveness. What does it mean? From the nature, from our environment, you can get a material that you can sequence. And then this material, you can put it into the cloud into the cloud database. And from the cloud database, you can use it to design a new biological organisms, which can be done in our bio and can be reserved in our biofoundry. So it revolves around our environment, which the government takes care of. And based on that, the government must take care of the issues of DSI-enabled synthetic biology. Next. So what are the major policy and challenge implications for DSI-enabled synthetic biology? Next. I will briefly run on them, about 5 of them. One is asset and benefit sharing. So if a community or a country's biodiversity is sequenced and then you upload it globally, who benefits from it? What developing countries like ours in Nigeria and Africa argue that genetic resources come from a biodiversity-rich nations like Africa, and then companies in wealthy countries use this DSI, this information that has been uploaded commercially, and then use it for synthetic biology applications, and then use it to produce materials, use it to produce industrial organisms, climate smart agriculture. And the question is, who benefits are not fairly shared? Because we picked those materials from our environment, from our biodiversity, you sequence and put it globally, who benefits? So governance needs in this is that we will effectively domesticate the Convention on Biological Diversity's Access and Benefit Sharing Protocol. So all nations are expected to domesticate the access and benefit sharing so that it will help them to regulate the way our organisms, our biodiversity are being picked up and sequenced and put into digital biology. So again, government should create a fair mechanism so that DSI use will be shared, that the DSI, that things that are generated from our DSI, the resources generated, will be shared equally so that it will benefit from the community and the origin where it comes from. Next. So the second policy implication is on intellectual property rights. The question is, can digital gene sequence for synthetic biology applications be patented? What are the risks involved? It can lead to monopoly over biological innovations. It can lead to high cost for synthesized products, and it can lead to reduced open science collaboration. And for this reason, there is a need for balance. So we need to protect our innovation incentives while ensuring public access, research freedom, and having affordable synthesized products. And for us to balance these needs, we need to come up with policy options. We can either narrow our patenting, open source bring about open source biology models, or have a compulsory licensing in cases of emergencies. So next, the next policy implication, the third policy implication is, next please. The third policy implication is issues of biosafety and biosecurity. Here we are talking of the issues of dual risk, the dual risk process of synthetic biology. So DSI makes synthetic biology application faster, but also riskier. What are the potential risks we are talking about? There can be accidental release of engineered organisms. There can be dual use risk, that is the misuse of harmful pathogens. A particular pathogen can be used to produce a resourceful or productive material for societal use, but at the same time, it can be used to cause harm. So this is a major risk, a dual risk. And then we have our cyber biosecurity breaches. Now you are storing sequences online, digital. So it means that if you store a sequence of a high pathogenic organism online, people can breach. There is possibility of breaching the securities where it is stored and accessing these gene sequences from online and use it, applying synthetic biology and produce a new organisms that can be dangerous to the nation. So these are the risks. And the governance questions is, who screens the DNA synthesis order? This is a very important question that biosecurity and biosecurity community are looking at. Having how you can, if you order a sequence from the companies that has these genes or digitally, who screens it to know who is requesting for this screening? So, and how do we regulate a
Lemos, are you okay?
Yeah, it looks like we lost Kingsley there. I think the internet though.
Global, there is a need response for global early warning systems, risk assessment frameworks, and responsible research oversight. Please, next slide. So the fourth policy implication, okay, I think back, please back. So we have another policy implication, which is equity and capacity gaps. So we have the current global divide is seriously deafening because high-income countries often have the sequence centers, they have the AI tools, they have their skilled workforce, they have already existing biofoundries. But many low and middle-income countries, especially in Africa, have limited labs, they have weak regulation, they have poor internet and data systems, and there are a lot of brain drain. So this is a very big, there is no equity and capacity gap in terms of digital sequence information. So what are the governing implications? So without capacity building, DSI may widen the inequality. And the solutions include technology transfer, South-South collaboration, increasing our training programs, and having a shared regional labs. whereby the high-income countries and low and middle-income countries can share labs so that we can bridge the capacity gaps between the global north and the global south. Next. So the governance model, what are the options that exist? We have 3 options that can be recommended. We can have our open access model, that is having a free global sharing of DSI. But what is the good there? The good in this option is that it will accelerate science. Everybody can access and accelerate science. But what is the bad? The bad there is that it may create an unfair commercial gains, because the people that has the sequence that you are already accessing, they may not be able to benefit from the commercial gains that come from it. Now you have the second option, which is a controlled access model. Here, we can approve licensing, permission, and traceability. The good there is that it will bring fairness. We will be able to understand wherever the research is coming from. We'll be able to bring everybody on board, even those low middle-income countries like us in Africa, to be able to have a controlled access model to this DSI. But the bad is that it will lessen lower research, because the time and the cost for regulation in terms of licensing, they delay research. Now, the third option, which is currently existing, which is what we call the Multilateral Benefit Sharing Fund. This is currently in place under the Convention on Biological Diversity. So here, all the users will contribute globally and fund support conservation and equity. So I'm a little bit biased on this, because we see that I have only good there. The good there is that there will be practical compromise. So everybody will be accessing from the fund you are contributing. So even if you pick from my own country, from my own community, all I know is that I will find a way to access it, because we contribute a fund into a global fund that will be used to deal with issues of conservation and equity in my country, and also growing international support. You will see a lot of international people supporting the global south, because they know that when they contribute to the multilateral fund, they will use it to support many countries in the global south. So these are the 3 options. Next. So in concluding, what should governments do now? And I break it down into 3. Government can take immediate national actions based on policy, institution, and international diplomacy. In terms of policy, it is advised that many African countries should start developing their national synthetic biology strategies. And they should be able to update their law to include synthetic biology and digital biology. I want to tell you currently that Nigeria is the first in Africa to update their biosafety law to include the regulation of synthetic biology, which is now regulated by the National Biosafety Management Agency of Nigeria. So countries in Africa should also develop policy measures that clarify IP and data rules. In terms of institutions, there is a need for government to strengthen biosafety regulators. All biosafety regulators within Africa, government should strengthen them with all the necessary resources to carry out the regulation of these DSI and synthetic biology. And there should also build efficient bioethics committee. I can tell you currently in Nigeria, there is a National Bioethics Committee, which is saddled under the National Biotechnology Research and Development Agency, which are doing everything possible to look into these bioethics issues. So these are the things the government should do to improve institutions. And then they should support tertiary institutions and startups in these areas. Now, for international diplomacy, government must actively engage in the Convention on Biological Diversity. They should engage serious discussion, because discussions of DSI and synthetic biology is centered within the Convention on Biological Diversity. So any country that is not engaging in the CBD discussions may be losing out on latest information on issues of DSI and synthetic biology. And also, furthermore, engage in discussions with WTO, WHO, and FAO, because all of them has interrelated discussions as regards issues of biological diversity, where DSI and synthetic biology fall into. As for education, Government must prioritize training of next generation bioengineers.
I have 1 minute, please, so we can go for lunch. Yeah,
yes, I'm almost concluding. Next slide. Next slide. So the future trends that we are going to watch from 2026 to 2025, scientists, we should look out for AI-generated genomes. It is spreading and it will come. Massive DNA storage centers. That is the new thing that will start coming up. We have synthetic food systems and decentralized manufacturing. So, but as these things are coming, I now throw a policy questions for all of us here today. Can regulation move faster as biotechnology? I may not answer, but let it be the answer for a question, an answer that will come up at the end of this conference. So next slide. So from all I've said, our key takeaways is that, which are what I've used to conclude my presentation. DSI is enabling synthetic biology into a rich bioeconomy. There are great opportunities in DSI-enabled synthetic biology, which are in the areas of health, agriculture, innovation. And there is also great responsibilities in fairness, in safety, in inclusion, and global cooperation. So I will conclude by saying that we must all know that the future of synthetic biology will be written in codes, but must be governed by policy. Thank you so much for listening. Thank you.
Thank you.
Yeah, thank you, Dr. Nwosu, please, Wanyika Nwosu, for that presentation. There are so many, my stomach is cracking already because, but I will not have the time to ask you some questions on here because of our time. Maybe I'll get a contact and interact with you. You know, sometimes things are good in paper, but not in reality. That's the problem. But I like your presentation. Yeah, Dr. Thank God, please. What nice, because do we have questions? Say again?
I think there are questions on the chat box, but we need to head for lunch. So can I please ask Dr. Nwosu to respond to the questions on chat? And those on site, if you have any question, please paste it on the Zoom chat box. Then
yes,
Dr. Nwosu will respond to it. Because we need to head for lunch and be back for the next session.
Yeah, but we have a couple of questions here. Do we also take, or do we?
Yeah, I think we should. Yeah, let's if you can collect the questions and post it in the chat box, then Dr. Nwosu will stay behind to respond to them. Already I can see some questions for him, so please respond to them on the chat box.
I think it's good to take it live instead of chat box.
All right, that's fine. Okay.
Yeah, let's just start from Dr. Fortune here.
Thank you very much, Dr. Nwosu, for your nice presentation concerning DSI. I know I did a work some time ago where I did molecular breeding in Arachis hypogea. And one of the challenge I encountered in course of that particular research was the issue of its applicability in plant breeding. I was able to access databases and extracted these sequences, analyzed them, did a lot of permutation. And at the end of the day, I sat down and I was asking myself, what is the practicability of actually bioengineering, putting in back these designs we have done there in using our systems? How do we bring it back into real time, into real life? And that comes in with the idea of bioengineering, training of skilled workers in the area of bioengineering, so that after I've looked at these beautiful resources we are putting out there, because why I started that particular research was because I was thinking of this chunk of data we are bringing out and then pushing out, and nobody is actually looking at certain areas of this data. How do we harness it? How do we, because they are funded, you know, heavily funded to be able to get this data. So I want to ask the practicability of this in breeding. in plant breeding. Yeah.
But am I thinking that Dr. Nwosu, Dr. Nwosu, you talk about the policy surrounding what we are doing about DSI, but I'm not thinking that Dr. Nwosu is into breeding, so I don't know if we allow that question to pull through. Dr. Nwosu is in policy framework. I'm not thinking. Nwosu, do you have anything to talk about in that?
Just like you mentioned, it may not directly come to me, but what I can mention, what I can drop here in views of policies in line with policy from what she mentioned, yes, these are coming up, these data are coming up, and a lot of modern biotechnology is being involved in the agri sector. But the question is, all these must be regulated. And that is why there is a biosafety regulator across global nations to regulate the activities of these modern biotechnology that we are applying. Like, you know, there are new breeding techniques. We are talking of genome editing, which is now going on. These are new breeding techniques. And a lot of countries are adopting it. But the question is, there must be a policy to guide all these processes. And that is why where biosafety regulation comes in line with the Cartagena Protocol on biosafety. So I think this is what I can mention, because the policies and guidelines and regulation, it helps streamline the activities within these technologies, modern biotechnology sector.
Yeah. So thank you, Dr. Nwosu. So I think
Thank
tomorrow.
you very much.
Tomorrow
we have an.
Okay. I said tomorrow we have a session on these policies.
It's
So
going to be hot tomorrow.
So let's not. I wouldn't. Which is why I don't want us to ask so many policy questions today. Let's leave it tomorrow. So, Professor Julian, I note that the next session needs to start in 35 minutes time. So, can we take one question, very short, and then we go for lunch,
just one,
and then we return back? Yeah.
Yeah. Okay. Thank you.
All right. Thank you very much, Dr. Nwosu. That was a very lovely presentation. My question is more or less about, not really a question, but just to hear, have your own perspective. You made mention of the 3 different models that we could possibly adopt for, equal benefit sharing across different stakeholders. But I want to have your perspective around the 3rd model, which seems to be much more, I would say, practical and then an easy hanging fruit for us to, implement. So from the Global Fund, which countries would you say should have the largest or the larger share of the fund that we're pulling together globally? Is it the countries that are, generate a lot of data sets, or the countries with, highly improved AI or infrastructure in place to, translate those data into products? So from your perspective, which side do you think should get more share from the Global Fund? Thank you.
Yeah, sorry, along the line, I didn't get you very well. So, but I know you are asking a comparison of 2. Please, can you briefly mention it again? I didn't get you well.
Yeah, so my question is, who should get more share from the global pool of funds in terms of the benefit that are being derived from maybe products developed from the data sets we have from different genetic resources globally? Who should get more of the benefit? Is it countries that are generating the data, or countries that have advanced infrastructure in place that are translating the data into products? Yeah, that's just my question.
Okay, thank you very much. And this question is a question that is currently being debated in CBD, because for most of us in the global south, these are the questions we are putting on hold. But I know we also have a presentation of experts that has been into the multilateral fund, who will also make some comments in this area. But we within the global south are agitating that this funding, most of course, should come most to the global south. One, there's that most of the biodiversity-rich nations are within in Africa, and this is where they get most of their genomes that they sequence in Africa. And secondly, the capacity is not there, the infrastructure is not there, the technical know-how are not there. So we are agreeing within the international level and negotiation under the multilateral fund that most of this funding should be prioritized to the global south. So this is the standard, and this is a global argument, and the negotiation is currently ongoing. So your questions and your perspectives are germane, and we in the global south has also been putting up this perspective in order. So we hope that.
But almost, you know, that assumption is just for me, it doesn't hold water. The truth is Nigeria contributing to the fund, and if Nigeria contributing to the fund, to what amount are they contributing? Are you telling me that, for instance, if US or UK sends, let's say, millions of pounds into the account, and you that is sending 1,000 naira or so will get the same money? It doesn't work the way you're thinking, because the bureaucracy and the requirement for you to access the fund is not what you're thinking about. So you will only be begging for you to get it, but it doesn't work like that.
So sorry guys, I would really want us to, so we have tomorrow, the whole of tomorrow, to discuss this. So it looks like all the discussions for tomorrow are spilling into today's science tech talk. So can we just, Professor Odense, let's mellow down. Tomorrow we'll have experts from the CBD who will be in the meeting. He's going to be opening the session, and we'll have people, experts that have written papers and policies, briefs on this discussion. So let's not eat into the time for the science of today, if that's okay.
Yeah, for sure.
So, all right.
All right. Thank you. I will be there tomorrow too.
Okay, thank you. Yeah, I think you are going to. So it looks like a lot of discussion is kind of coming up, and I'm really pleased that this is provoking some discussion. So, Olarewaju, I'm hoping you'll be here tomorrow, so you can also get more clarifications on this particularly. So, Professor Julian, I think we can go on break now and be back in 30 minutes time so that we can meet up with the next session. The next session starts at 2:30.
Yes, I think that's
so that ultimately we can finish at 5 o'clock or 10 past 5, because we'll have several speakers from the US and in Africa who will need to finish up on time. So thanks everyone. We would go on lunch break now and be back in 30 minutes time, and 30 minutes time will be at 2:30 GMT +1. That will be 30 minutes past the hour. Thank you so much. Over to you, Professor Julian.
Yeah, thank you. And I think I want to also say that there are so many people here who want to ask questions, but to reserve your questions. Tomorrow we'll have enough ground for the questions. All right. So we are going to have our lunch break now. 30 minutes time, I think, will be good enough for us to take care of our tummies and then come back here. Please, we have very important presentations coming up after the lunch break. There are many key papers coming up, especially from the US. The papers were fixed to be this afternoon because of the time difference. The time we started was still night in the US, and it's now that is their early hours of the day. So we'll come back prepared to take more presentations. Thank you all. I'll thank you for your patience. We'll go for lunch break to come back in 30 minutes. I think this is the end of the session. Thank you.
Yeah, so see you all in 30 minutes time.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabytes of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have the bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our Incoterms include DAP and DDP, so we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budget. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genelab.ng or tgl_ont@genelab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the Gene Lab of Science. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequencing simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabyte of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budgets. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genelab.ng or tgl_ont@genelab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the General Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabyte of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budgets. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genlab.ng or tgl_ont@genlab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the Gene Lab of Science. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabytes of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have the bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our Incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budgets. You can place an order via our store. The link is on your screen. And also you can send a request to this email address, ont@genelab.ng or tgl_ont@genelab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.
All right. Thank you, everyone, for joining the session, the next session of the workshop. And this session would focus on the AI in conservation of endemic and endangered species. I understand that the on-site speakers or presenters are still, trying to return back to the hall, but we're just going to start anyway, and then they can join particularly. So our speaker for today, a keynote, will be Dr. Zara Munglo Dihomud. So Dr. Zara will speak to us about harnessing digital technologies to inform and strengthen agricultural and conservation policies. Over to you, Zara. You have the co-host rights, so please feel free to share your screen.
Okay, good afternoon, everyone.
Afternoon.
Okay, let me just see if you can see my screen. You can hear me, right?
Yeah, we can hear you.
Okay,
Carry on.
great.
Okay,
And
now we
you
are
can
trying to see your screen now. It's coming up slowly.
Okay. Is it
Yes, it's
okay?
up now. Yeah, it's up now. Carry on, please.
Good. Okay, so hello everyone. I'm Zara Mamloudil Mohammed. I'm an associate professor at the University of Mauritius, and I'm also part of the African BioGenome Project since 2021. So today I'm going to try to convince you of how AI can really help, how digital technologies through the use of AI can help with agricultural and conservation policies. So the title of my talk is Harnessing Digital Technologies to Inform and Strengthen Agricultural and Conservation Policies. So I reckon most of you already know this, but in case we've got different kind of audience today, I'd like to remind everyone of why it's important, why endemic and endangered species are important for Africa. First of all, we are lucky. We've got a unique biodiversity on the African continent. We've got the rare species, which are only found in Africa. These are important also because of ecosystem stability. So they play vital roles in maintaining ecological balance. And last but not least, for cultural heritage. So these endemic and endangered species, they also are sacred symbols and they have traditional knowledge. So these are very important for these reasons. But there are threats to these endemic and endangered species. First of all, habitat loss due to agricultural expansion and constructions, climate change and ecosystem disruption, and also, very importantly, unsustainable agriculture. Now, policies can help with the conservation, but often they lack real-time ecological intelligence and they come way too late. Now, AI can enable timely and data-driven conservation and actionable insights for agricultural and conservation policy decisions. But before we can see how policies can be impacted by this, let's see what we can do with AI in terms of endemic and endangered species. So why do we need digital technologies and indirectly AI? First of all, through digital technologies, we can have real-time data. So through digital technologies, we've got real-time data and it can help us take informed decisions. Then we've got enhanced forecasting. AI enhances forecasting for accurate predictions. We do have data currently, but the data is not always real-time. If we have digital solutions, all the data will be real time. Of course, there are many aspects to it. We've got connection and so on and so forth. But if everything is in place, then we can make use of those real time data. And through analytics, through AI, we are able to enhance forecasting. And we're going to look at examples on the coming slides. And then through digital technologies, we can have improved coordination. So this facilitates improved coordination and seamless collaboration. And finally, rather than having policies being reactive after an event occurring, then we have the policies through digital technologies and AI, we can have a proactive policy rather than reactive. Now, how can AI help in conservation? We've got 3 images in front of us. So these images, they show how AI can help with wildlife or species identification through the use of image recognition and acoustic monitoring. So the first image here is an image where we are using YOLO. In this instance, YOLO V8 was used, and YOLO is used for image recognition and object detection. And in this instance, it is able to detect that these animals are giraffes and is able to count how many giraffes there are on this image, as well as how many zebras there are. In the image below, it again uses object detection, and you can see that each image has a boundary around it. So again, object detection. And finally, here in this image on the right, it's making use of acoustic monitoring together with camera. And if you want to learn more about this, you can check out the links that I've provided here. So this is how, for example, we can use AI in conservation for image recognition and acoustic monitoring. Secondly, we can have habitat mapping through the use of drones and satellite imagery. And I've got an example here, which is an image which is provided by VITO, which is the Flemish Institute for Technological Research, which is in collaboration with the European Environment Agency and the Wageningen Environmental Research, where AI and machine learning have been used to classify habitat maps for regions including Netherlands, Austria, and South Portugal. Well, unfortunately, this image is not for Africa, but this is a good example where AI with drones and satellite imagery has been used for habitat mapping. We can make use of AI as well for deforestation and land use change prediction. So I repeat, here it's being used to predict when there's going to be deforestation or when there's going to be land use for other cases. And there is an example, which is the Forest Foresight, which is a WWF initiative that uses AI to predict illegal logging before it happens. How does it do this? It identifies early indicators like the construction of new access roads. So if there are new access roads which are being constructed to forest, the AI is able to guess that probably there's going to be some kind of illegal logging or some kind of deforestation which is going to take place or a change in land use. We can also use AI for population monitoring and to get migration patterns. So by analyzing existing data, the AI is able to track and analyze animal movement and behavior. We've spoken about animal tracking, so we can also track animals through their tracks. So the example here shows tracks of animals, different animals. So we've got bear, cat, coyote, fox, horse, and lion in different environments, in snow, in sand, in mud. And here the second image below, it shows tracks of animals on snow. And using AI, and deep learning in this case, we are able to track animals, in this case tracking, but also through their droppings and their rubbings. And again, through AI and object detection and image recognition, we are able to detect when people are going to poach. So for anti-poaching efforts. And again, the links have been provided here. Now, these are examples where AI can be used in conservation, but AI and digital technologies can also be for digital agriculture. So here on this slide, we are talking about, first of all, resource optimization. So AI irrigation systems can make adjustments in real time based on data from soil moisture sensors, from satellite imaging, and even from weather forecast. We all know that water is very important in Africa, and not only in Africa, actually in many, many countries in the world. And through the use of AI, we are able to optimize the use of water. And of course, this does not only apply to water, it can apply to any other resource, as well as we have the relevant real-time data. Then we've got data-driven decision-making. AI can combine the traditional observation and intuition that we've already been using to elements such as real-time sensor readings and data analytics, and therefore enables a more data-driven decision-making. AI can also be used in precision farming. So
So I think we just lost Zaria. Maybe perhaps she lost connection. So let's give her 2 seconds or so she can join back. I think Zara just lost her internet connection.
Yeah, while we wait for Zara, is there a possibility of calling for someone else so that Zara will take the next?
So maybe let's try and give her 20 minutes because she's like in the middle of the presentation and halfway through. So yeah, so she's just let's give her like about a minute or so, because I think she has internet or power challenge.
Yeah, it's okay.
So I think we might have to, because Zara hasn't joined back. I believe she has like internet issues. Okay, she just said she's trying to join again, so we just give her half a second or so. Okay, Zara is here. Zara, please carry on with your presentation.
yeah, okay, sorry about this. We have a power cut in
Please unmute yourself, Zara. I share your slide. You're a co-host now.
Can you not hear me?
We weren't hearing you before, but we can hear you now.
Oh, great. So I think maybe I got disconnected. Around. Was it?
Okay, so it looks like we still can't hear Zara, so we might have to move to the next presenter. Zara, are you still there?
Okay, let me just see if you can be better. Can you hear me
Yeah,
now?
okay.
Thank God.
Okay, all right. Carry
Yeah,
on, please.
I'm here.
From where you stopped, yeah.
I think I stopped here.
Yeah, because the
So
internet
I'm just going to start here
breaking.
again in case. Oh, is it still breaking?
Yeah, you are lagging behind, but it's fine. You can. Why don't you turn off your camera? I think that might be a good thing. Thanks, George.
Okay. Yeah. Is it better now?
Yes. That might help. Yeah, please go ahead.
Okay, thank you. So yes, I was talking about how AI can be used in digital agriculture. So we were talking about how we can have decision making, which is data driven, where AI can combine the traditional observation and intuition that we have been using currently in agriculture to elements such as real-time sensor reading and data analytics. And this makes the decision that we take even more powerful than the ones that we were taking with only traditional and intuition. AI can also be used in precision farming, where it enhances species conservation by optimizing resources such as water, fertilizer, pesticides, again for the use of sensors, drones, and machine learning, and hence reduce environmental impact. We can link agriculture with conservation data. We can we can write code so that we can have an agriculture which is more adapting to the climate. Through the use of AI and existing climate data, we are able to link, to find links that were previously unknown or which we could not find because we could not analyze such a large amount of data without the use of machine learning. And also, by making use of existing historical data, we are able to predict disease outbreak which can potentially happen, but which has not yet happened. Of course, for this, we need historical data on whether outbreaks have happened in the past, but also on weather data, on temperature, and on water, and so on and so forth. Through the use of digital technologies, we can also have biodiversity-friendly practices. So we can use AI models to analyze data, which can help suggest varied, resilient crop selections and farming patterns, breaking the resilience of monocultures. Because monoculture is very bad for biodiversity. Now, what are the risks and challenges attached to using AI for digital agriculture and for conservation? First of all, the major challenge is the data gap. Currently, there is a lot of data, but a lot of data is not necessarily for the African continent. Secondly, we've got the digital divide. We do not necessarily have the knowledge, and therefore we may be, if we do not have the necessary knowledge, we may not be able to implement this. This is why it's very, very important to ensure that there is training which is done, and this is why this workshop is so important. Then in terms of infrastructure, we are aware that infrastructure is not so good. And even in Mauritius, you just saw we had a power cut. So infrastructure is not always helping for AI. Then we've got governance issues. Do we have the regulations in place to help with AI use in agriculture and in conservation? And then we talk about human-centric AI. Now, what is human-centric AI? Human-centric AI is when we want our AI to work in collaboration with humans. Of course, when we are going to make use of AI, it will replace some jobs, maybe. But it shouldn't be that some people are left behind. When we replace jobs, it's very important that either these people are reskilled or we have an AI which works in collaboration with humans. We shouldn't have an AI which takes over human jobs and the humans are left behind. So this is a very important part. And then, of course, we all know about data bias. Data is not always, so when we have data, it's not always fair. So what does it mean to have data bias or data not being fair? It's when our data is either imbalanced, one part is overly represented and the other part is not well represented, or it for the other populations. And also data privacy. Many people will be afraid to give their data if they are not secure in the knowledge that this data will not be used wrongly and will not be used against them. So the regulations should be in place so that people know that when they are providing their data, okay, this is not going to play against them in the future. Now, how can all this be used for policy? How can that use of digital technologies and AI impact on policy? First of all, the strategies will shift from being reactive and intuition-based to being proactive and evidence-based. We know, based on numbers, based on outcomes, that this can happen, and therefore we can be proactive in coming up with new policies. And then AI can help with regulatory compliance and enforcement, because monitoring compliance with environmental regulations is a very complex task. And it can also help with cross-sector collaboration, and it can help with alignment with SDGs and CBD, which is in line with the Kunming-Montreal Global Biodiversity Framework targets. Policymakers must develop strategies to manage labor transitions effectively, ensuring that displaced workers have opportunities for retraining and alternative employment. It requires having policies in place also requires collaboration among governments, labor unions, industry stakeholders, and educational institutions to facilitate workforce adaptation to an increasingly automated agricultural sector. And we have the legal and regulatory frameworks which must also evolve to address the complexities of autonomous agricultural robots. Now, how do we go about doing this? So the roadmap for action is, first of all, capacity building, as we are currently doing this week. So we have to enhance the skill and knowledge of professionals so that they can know what is available and so they can take a state-of-the-art decision based on state-of-the-art knowledge. Secondly, infrastructure investment. Government institutions should be aware that they need to have the relevant infrastructure in place. Otherwise, they will not be able to keep up to date. Data governance. We should have the framework to manage data and for security. We should have an open biodiversity data, making biodiversity data accessible to the public. Policy pilots. So we should test and refine policies. It's not we come up with a policy and that's the final one, and then we find it doesn't work, but we should have pilots in place. And then we come up with policies, we test it, we refine it, and this is anyway a continuous process. And finally, we should adopt national and even continental AI strategies where we integrate AI into biodiversity conservation efforts. To conclude, the convergence of digital technologies with agriculture and conservation is no longer optional. It's foundational to resilient, evidence-based policy. By integrating data, analytics, and local knowledge, we can move from reactive decision-making to proactive, adaptive governance. However, technology alone is not sufficient. Impact depends on inclusive design, strong institutional capacity, data governance, and sustained collaboration among researchers, policymakers, practitioners, and communities. The opportunity is clear to build policies that are not only smarter, but fairer and more future ready. And I wish to thank you all for your attention. And as we end, I'd like to know if anybody has any questions. I don't know if you have time to take questions. And the QR code is for my Google Scholar profile.
Thank you,
very
Zara.
much. And again, apologies for the internet issue.
No problem, Sarah. Thank you so much. So, Zara, if you don't mind, please hang on the Zoom call if that's okay with you, because we're going to have like a panel style Q&A where we would, so anyone that has questions, please kindly put your question in the chat box, and I'm going to read it out basically for the online participants, or you can raise your virtual Zoom hand. And for the on-site participant, the on-site moderator would collect the question. So Zara, please hang on the call for the next
sure.
Yeah,
sure, no problem.
So. Okay, our next speaker will be George Tiley from North Carolina State University. I'm just going to give George the sharing rights to share his presentation. Okay, George, you have the sharing rights. George will speak to us about the prospects of AI in exploring polyploidy as a pathway to niche diversification and the expansion of Madagascar's Greenland. So I think we're also going to East Africa this time around as well. So yeah. Okay. Can you hear me okay? Yeah, over to you, George. It looks like can you speak? It looks like we weren't hearing you clearly. As if you were distant from the
Can try like
okay,
this.
I think this is better now.
Good.
Yeah.
Yeah, and so it will we'll focus a little narrowly on Madagascar today, but I think the topics and issues are really Africa wide, so hopefully there's a little bit here for everybody. So I research grasses and grassy ecosystems, and I've been working in Madagascar since around 2017. But before we jump into any specific system, right, I want to touch base a little bit on polyploidy in case some people here are unfamiliar. So polyploidy is a phenomenon where organisms might have, say, more than 2 copies of their genome like we do. And plants do this a lot. They're really good at it. And this comes up in a lot of our important crops, like, say, wheat or sweet potatoes. But here on this slide, on the right is a tree with about 10,000 tips. They're just colored by some different plant orders. And on that outer ring, you see the distribution of diploids and polyploids. The only thing you really need to get from this figure is that polyploidy has arisen many, many times in the plant tree of life. And then if we look at the panel on the left where we break down the frequency by order, it occurs in different frequencies in different plant groups. The number 1 group being Coales, which includes the grasses we'll talk about today. And you don't have to just trust my numbers. Botanists have been thinking about polyploidy for as long as they could observe chromosomes under microscopes, and this has been a recurrent theme. And we'll stop and think about that for a second. Maybe there's 12,000 species of grass. And if we say, well, a little less than half of them are polyploids, that's still several thousand species. And polyploids are really difficult to sequence and analyze and address with genomic data, but those are areas where we're having a lot of advances these days. And a lot of the story today, we're going to focus on, say, this one species, are a representative of a lineage, Lutetia simplex, and most people in the room there have probably seen this, or you know it, even if you don't think about it a lot every day. And this represents a tribe of predominantly African grasses known as the Tristacadiae, and their early diverging panacoid group, so related to a lot of other things we care about, such as corn and sugar cane and forage grasses. And I ended up studying grasses because they're really difficult ecosystems to understand. So a lot of the great work that's come out of African savanna ecology is that these are really dynamic ecosystems driven by disturbance, and that can come in the form of grazing and fire. It's not purely about precipitation and temperature. But this also makes them very difficult to tell apart from what's a natural grassland versus an anthropogenic grassland. And this is an increasing issue when we think about climate change and how that affects habitat loss. And these are ongoing challenges in Madagascar, especially where the origin and extent of their grasslands are really contested compared to, say, other parts of East Africa or tropical African savannas. Because people will say a lot, well, where are the grazers? And maybe it gets a little bit more rain than some other savannas. So we want to understand to what extent this is anthropogenic versus natural. And this generates really difficult applied questions in conservation biology. So in some of the figures on the right are the Ankafo Bay protected area. It's about a 1 kilometer square forest in the Central Highlands, and you could find, for example, species of mouse lemurs in that forest. Now, there was a bad fire there a few years ago, and that kind of makes land managers have to make difficult decisions about, is this now a reforestation project, or is this part of a natural cycle, regardless of if the source of the fire was anthropogenic. And other reasons understanding these grasses can be tricky. They're just very common. When you're in, say, these natural habitats, you'll see them as far as one of the herbarium labels said, as far as the eyes can see. But you'll find them really adjacent to recent agriculture as well, and even mixed in with agricultural systems like these terraced rice paddies and planted next to corn. So that can drive a lot of, say, temperate NGO and conservation groups to look at grasses and say, well, clearly these don't belong here. This should all be forest, but the story can be a bit more complicated. So some things that I've researched is how different grassland and plant communities assemble, and what are the hallmarks of native flora and natural communities where you might find Lutetia simplex co-occurring with these endemic bulbostylus sedges in Madagascar. But then when we look at sites where there were recent anthropogenic fires and these were recently forested areas, we don't find Lutetia simplex or some of these other community members. And a lot of this is to say that we want to really understand grasslands for both the sake of the grasslands themselves, but also as a way to understand other types of endemic and endangered flora and fauna. So this was kind of the flagship paper from a mouse lemur group I worked with for many years, and ultimately a better understanding of the ecology and the vegetative history led us to make recommendations for changing the taxonomy of some species, which would ultimately lead to some different risk assessments for them. So I'm not a policy person. I just follow the science and do that aspect on the molecular side. But there can be really big shifts in how we approach conservation biology based on these results. So I want to highlight a little bit of work from a former master's student of mine. She's now working on a PhD at the University of Toulouse. And what we found was that in the 70s, a Malagasy species was synonymized under Lutetia simplex, which is kind of the widespread African species. But even if we just look at these two herbarium specimens here, they look quite different. And that kind of got us thinking, well, maybe polyploidy is an important part of the story here, because a really common effect that polyploidy can have in grasses is the plant gets bigger. And she's done some nice morphological work and ecological modeling on this group that we hope to have published soon. And this is, again, just to highlight the distribution and the habitat suitability of Lutetia simplex across Africa. So we're really focusing on a narrow subset of the variation, what I'm talking about here today. And one of the important reasons we're interested in polyploidy is that it can often be a pathway for rapid local adaptation, especially to, say, drought and poor soil conditions in grasses. And there can be a lot of really nice examples of other African grasses that have these types of adaptations that are more applied. If we think about teff, if we think about fonio, these are allotetraploids that have a lot of these features that we want crops to have in general, and we want to understand those genetics. And Lutetia gives us another model system for understanding those mechanisms. So what Chana found from her distribution models was that there were novel areas in Madagascar that Lutetia simplex had moved into, and these were not predicted based on the projections from the environmental models of other African distributions. And I think that's where the polyploidy will come back into play a little bit later. And so a few things we've done to advance this research. We've generated a reference genome for a closely related species. I think if it's not on NCBI yet, it will be soon. And a few things we do to study polyploidy is looking at chromosome squashes. So we actually have to take a root tip, prepare some chromosomes and stain it, and we'll try to make an observation. So we wanted to make sure we had at least some diploid basis for studying this system, and we found that with a more widespread species, Lutetia flavida. And just kind of went at this with the single PacBio library and some Hi-C data. I won't cover too much about the methods of how we do all this, because I think the people there are going to get a lot of this in the workshops ahead. But it's really amazing how now we can get a reference genome for some of these groups that allows us to do the population genomic work that we're interested in much faster than it used to be. And this is just to give an idea of the reference genome. We're able to recover the expected number of chromosomes. It has some of the nice features of grass genomes when we compare to some other species such as rice and panicum, and we're able to recover the two haplotype phase genomes, even though there might be a few errors in there that can be corrected over time. Right. So now back to Lutetia simplex in Madagascar. And we also have a sample from some other parts of Africa, just a lot less dense. And this was all going to be using RAD-seq or GBS data. So it's not like you need to have whole genomes to do a lot of this work. What we did find was a single origin of Lutetia simplex in Madagascar. And some other bits of information that we know is that the South African samples were diploid. From recent collections, and here were some of mine, we found that this one population appeared to be tetraploid, and that's really in kind of the higher altitude regions of the Central Highlands. And we knew that another group in one of the novel areas that Chana found in her distribution models, those were hexaploid. But we honestly didn't know a lot of these other species. And when we want to talk about ploidy and polyploidy in plants, it can be really hard to do that when the specimens get a little old, in the sense that we go to the field, we collect specimens, we'll press them, we'll collect some tissue and preserve it in silica, and that can be very useful for DNA sequencing. And we've gotten really good at sequencing kind of older specimens out of herbaria. But to get the ploidy information, you really kind of need fresh tissue to do that. With grasses, we might have a 6-month window to work with them, but the signal erodes pretty quickly. And here's the primary way we're able to get ploidy information out of plants, is with flow cytometry. So that's a procedure where we have an instrument called a flow cytometer. We'll chop up a little bit of plant tissue with the razor blade in a nuclei extraction buffer. And we can stain it with a fluorescent dye like DAPI. And that just goes through a sensor that measures, well, how big are the particles going through? How big are the cells? And when we have, say, some calibration points, and here was that diploid reference genome where we had a chromosome count for it, we can start to differentiate, well, here's a diploid, and so maybe this is going to be our tetraploid population because it's roughly twice as big. And this is how we started teasing apart where the tetraploids and hexaploids were in Madagascar from the recent collections. We would get a peak around there that would represent the tetraploids, and if you're kind of 2/3 the size of that one, it could be a hexaploid to tetraploid ratio. So that's really the gold standard on understanding ploidy. But if you don't have that information, it can really complicate a lot of your downstream population genetics work, in the sense a lot of what we do in population genetics and evolutionary biology would rely on estimating genetic diversity. And you can think of that as something we do using the site frequency spectrum. So the nice thing about diploids is if you go through the process of genotyping and calling your variants, if you know you have a heterozygote, you know the dosage of the allele. And you can come up with these site frequency spectra that let you do things like estimate genetic diversity, look at loci under selection, reconstruct demographic histories. But if you don't know the ploidy, you don't really know the dosage of the alternate alleles, and that can lead to a lot of ambiguity in your analyses. And this is just to say that collections are really valuable. We can use them for understanding really broad evolutionary questions. In the case of the grasses I work on in Tristacadiae, like these are closely related species. And we want to understand the genetics of that huge phenotypic jump. Here's a narrow endemic from Sierra Leone. And then here's going to be a much more widespread species. Would that be a difference between ploidy in addition to the genetic variation? And these days, I work in a much smaller herbarium, and I spend quite a lot of time in between these two cabinets. Our collection is, comparatively small to the big biodiversity groups, but it's really applied and regional in the sense that we want to leverage these historical specimens to understand the spread of herbicide resistance in different weedy populations. We want to understand where we are losing genetic diversity in some other critical plant groups that we're concerned about. So it's important to leverage collections for regional work as well. It doesn't always have to be about the big biodiversity stories. So in order to kind of better leverage these collections, something I had been working on for a while was how can we estimate ploidy from sequence data directly when we don't have that flow cytometry information? And around the same time that I was publishing some of this stuff, there was also another scientist that had done some similar things, and we're going to look at her figure here because it's really pretty. So you would get your VCF from genotyping your specimens. And you say, well, if it's a diploid, a lot of the variants should end up with half of the reads supporting the reference allele, half the reads supporting the alternate allele, with a little bit of sample error there. But then as you go up to a triploid, say if you have one allele supporting the reference and two supporting the alternate, or the other way around, you would end up with two peaks, and you can start to develop some expectations about what the ratios of reads supporting reference and alternative alleles would be under different ploidy scenarios. And I applied this a little bit in this 2024 paper, and my co-authors and I have continued to work on these approaches, but not with a lot of satisfaction, but that's where I'd say machine learning or artificial intelligence has been really impactful for the work I do in our approach to some of these systems. So I'll say part of this shift was driven by a master's student in computer science with me, and then some collaborative work. And if you are interested in these methods, they are online and publicly available. And before we talk about Ludicia again, I'll mention that we did a fair amount of testing in various agricultural and applied systems, one of them being blueberry, where we've tried to work on the hybridization and polyploid histories of this group. And I'll just go through this quickly, but what we're looking at here are some confusion matrices for our classification accuracy for diploids, tetraploids, and hexaploids. And one of the important things was that we found there was a big reference genome effect, but that I think most people can make sure they're sampling smartly. So just make sure you do that, whatever your design in your project is, that you're thinking about the outgroups as well. But we got near perfect classification accuracy in terms of the ploidy in this group. Aside from the case where we had maybe an inappropriate reference genome. And we did have one disagreement in terms of the classification accuracy, but it turns out this was probably an error on the flow cytometer and that result rather than the computational method. So yeah, we'll reiterate that the machine learning method is working pretty well for classifying ploidy in blueberries. And even with the confusion that might arise from an inappropriate outgroup, it actually did really good at classifying diploids versus polyploids. It just kind of struggled with differentiating between the higher ploidy levels. Now I'm seeing I'm a little short on time, so I'll skip through a little bit there, and we'll get back to Ludicia simplex. So we had a little bit of flow data that I had generated, and then there were some prior expectations, at least for some overlapping samples from earlier microsatellite work. So anybody that maybe has used microsatellites in plants, it wasn't uncommon to have this approach where you would infer ploidy by looking at the maximum number of observed alleles from microsatellite data. And it seems like actually like quite a reasonable thing to do, but there's a lot of reasons we're not generating microsats anymore. So we had some a priori expectations about ploidy in Ludicia simplex based on the older literature. And what we did find, though, was that we were getting really good agreement between the flow cytometry and the machine learning methods. Really perfect accuracy in that regard. And then the microsat data was a little less preferable. It worked really well in the sense that the microsat data was always able to get to classify diploids correctly, but it was less certain when differentiating between higher ploidy levels. Yeah, and we'll scoot through that a little quickly too. And so here's a breakdown of what happens when we apply a flow cytometry trained model for classifying ploidy based on sequence data, as well as the microsat trained model. We do get a little bit of disagreement among a few samples, especially in these kind of novel regions of Madagascar where Lutetia simplex was not really expected. But based on the performance of our training, like test sets, and then what we're seeing with Lutetia simplex, we're feeling really good about this machine learning model and making some determinations. So with that, it's really an interesting story where we seem to have, and we certainly believe there's more ploidy variation in Lutetia simplex across Africa, right? This is just a very narrow slice of the diversity. But there does seem to be this diploid South African population, a single common ancestor of Lutetia simplex in Madagascar, and the tetraploids are sister to a single origin of hexaploids that have radiated over the island. And we can apply a lot of population genomic analyses to try and understand, well, what does that demographic history look like? So what we're seeing here on the left is an analysis called space mix, where the underlying model is you're assuming isolation by distance explains the geographic location of the individuals or populations you're looking at, and you want to try and find, well, what is the geographic orientation of them that minimizes that residual error between the observation and the expectation. And what we do find is that whether we look at the polyploid-based allele frequencies or the diploid-based allele frequencies, qualitatively, we end up with the story of some radiation from the central highlands, certainly in the higher altitude regions, that would be environmentally more similar to that diploid population from South Africa, that they have then radiated from there into these novel regions. And that's recaptured a little bit if you just look at the PCAs as well. You can imagine rotating those PCAs onto a map, and that will show you maybe the center of variation. And some other things that the lab is doing going forward, we've been able to do things like selection scans, look at association with different climate parameters, including precipitation seasonality. And independently in the population we call Miandravazo and Ishal, I know there's a lot of colors going on here. These don't represent like significant outliers in terms of FST. The colors are correlations with environmental parameters. We find one of these spermidine synthesis genes that are shown to be associated with drought resistance in corn that really stand out as a candidate to look at in Lutetia simplex as far as local adaptation driving the expansion of these grassy biomes. And yeah, this is just really great because it's integrating a lot of the knowledge that Chana had put together for us with the novel genomic methods and the machine learning and just really standard population genetics. And with that, we've been able to develop a demographic model that's giving us a kind of a new perspective on the evolution and distribution of at least Lutetia simplex dominated grassy biomes in Madagascar. These are not primarily driven by anthropogenic disturbance, but that we think, right, these are at least several hundred thousands of years old, driven by paleoclimate change. And this is corroborated
George,
a little bit.
I think you have about 1 or 2 more minutes then.
Yep, I'm almost done. Thank you. Yeah, and this is corroborated a little bit by the paleoecological data as well. So as we move to more and more, say, digital sequence information and machine learning and AI, we shouldn't undervalue a lot of this previous work that's been done in terms of validating our predictions from these types of data. Right, so the demographic models that we applied, we found a lot of agreement with the increases and decreases in grass pollen through time up to the limitations of these paleoecological records. So yeah, I'll just reiterate two points where we're combining a lot of these approaches to understand what is a model of polyploid dominance during paleoclimate change, and that this likely isn't unique to Madagascar, but it's a story about grasses all over the world, and especially in tropical Africa, where we want to understand how some species rise to be quite dominant, and what are some of the drivers of grassy biomes. And we're certainly still taking a look at some of Chana's original work as well, where we're trying to understand species diversity within these understudied African genera and groups as well. There's 26 synonyms under Lutetia simplex. It's probably a lot more complicated than we appreciate, but that's ongoing work. And with that, I'll just say, thanks for letting me speak to everybody. There were a lot of people that contributed to this work across multiple institutions, and maybe there's time for one question, or I'll stick around for the panel.
Yeah, thank you, George, for presenting. So I think please stick around for the panel. We have one more presenter, and we'll just kind of collect all the questions in one panel, if that's okay.
Sounds good.
All right. Our next speaker is just gonna go to the agenda. Professor James Liebex-Mack from the University of Georgia. So I'm just gonna make James a co-host. Okay, Jim, you can share your screen now and unmute, please.
All right. Hello, everybody. And thanks so much for giving me an opportunity to chat in this symposium and workshop today. I'm at the University of Georgia in the US, although until July, I'm based in South Africa at the University of Pretoria in FABI, and this is enabled with a Fulbright program fellowship. So we're going to talk about using phylogenetic approaches to learn things about plant genome, gene, and trait evolution. And I'm going to be talking, I'll mention as we go through how AI tools are being used for each of the steps I'll be talking about. So my work is centered around phylogenomics in two senses. On the one hand, I'm using genome scale data to resolve phylogenetic relationships across groups of organisms and various groups I'll talk about. And then after I just briefly introduce that, I'll talk a little bit about how we can then use our understanding of relationships among species to make inference about genome, gene, and trait evolution. And this is really in line with thoughts that have been around for a long time in biology. Dobzhansky back in '73 wrote a paper to life science educators, nothing in biology makes sense except for in the light of evolution. And more recently, folks working with doing phylogenetic analyses would say nothing in evolution makes sense without, except in the light of phylogeny. And this is a note from, this is a sketch from Darwin, actually, maybe not so recently. So he was trying to think through his understanding of the variation he was seeing in his voyage in the Beagle by counting for shared ancestry and making the inference that things that look similar might be more might have shared ancestry and then have diversified from those ancestors. So using this approach, we've used transcriptome data to resolve relationships across, in this case, all Archaeplastida. So that would include red algae, green algae, glaucophytes, and then most of this is shown. Most of what I'm showing here are green plants, including land plants and algae. And so this analysis about almost 1,200 species with 410 low copy genes, really gave us a big view of relationships across the green tree of life. Following that work, folks took at Kew Gardens, with help from others, took 353 of those 410 loci that we had used and developed a bait set to be able to capture the sequences of these 353 low copy genes, often single copy genes, and reconstruct relationships for the majority of genera, and they're still working on this. They're wanting to get to 100% of genera, data sets for 100% of the genera in flowering plants, and resolve relationships across the flowering plant phylogeny. And one of the things that they've done with that, the phylogeny then, is tracked diversification through the history of flowering plants. And a key finding from their work is that most of the orders had arisen across the flowering plant phylogeny by 120 million years, about 30 million years after the estimated origin of flowering plants. And then families shown here in this purple have been accumulating more gradually over time. So that's phylogenomics in the first sense. We can again use genome scale data, whether it's from transcriptomes or genomes or gene capture analyses, to understand relationships. But then the second meaning is basically the flip side of this definition is to then take our understanding of organismal relationships, and then organize our genomic data into gene families, cluster all of our coding sequences, for instance, in the gene families, make multiple sequence alignments, do our gene tree estimations, and then refine our estimates of those gene tree estimations and interpret those gene trees that we're finding to look at things like gene duplications, maybe in some instances whole genome duplications, as George was just talking about. By combining our analyses of our gene tree estimates and our species tree estimates, and then to make some biological inferences, like maybe AI aided inference to inform downstream analyses to contribute to a growing bioeconomy. Now, as I think many of you all know now, that genomes are being generated at an increasing pace. This is a website that is available. I consult it frequently by folks in Germany, and they are keeping track of the published plant genomes. And you can see that as of the first quarter of 2026, we're up to about 2,600 genomes, many a single species per a single genome per species. But in some instances, we've got 2 or even more than 3 or more, and we're kind of entering for crop species and so on, and model species, the era of pangenomes, where we're looking at whole genome assemblies for lots of genotypes within the genome to do some of the analyses that we've talked about, that we've heard about earlier today, to understand diversity within species. Of course, the African BioGenome Project is contributing to this increase in genome availability. Darwin Tree of Life is doing quite a bit of work. Both of these kind of within the larger frame of the Earth Biogenome Project. And some of you may know this database here, this GOAT database that's kind of keeping track of where we are in terms of the number or the percentages of sequences that have been phyla, classes, orders, families, genera, and species that have been sequenced. Clearly, as we go to the higher or the lower taxonomic ranks, we have lower percentages of genomes that have been sequenced, and we're all working to try to close that gap and increase the number of genomes that we have for species that are well distributed across the tree of life. And so an initiative that I've been involved with, and it's an open initiative, so everyone's invited to be part of this, is Open Green Genomes Initiative with some folks at the Joint Genomes Institute in the US. We're generating genome sequences for species across the land plant tree of life, and the focus of this work is to fill in gaps in the tree of life. So I'm showing in blue the species that we are sequencing right now that we've identified as to sequence and are sequencing for this Open Green Genomes Project. And one thing you may or may not see is that we are really putting a lot of our effort in sequencing parts of the tree of life for which there have not been genomes available. And I'm going to talk today about work we're doing in the poales and in the legumes, two very important plant groups from a lot of perspectives, including agriculture. This Open Green Genomes project was launched with 100 co-PIs, but since it's been initiated in 2018, we've just been accumulating folks that would like to participate. And so I'd encourage anyone who would like to participate. I'll put my name and my email in the chat box to contact me and let me know. A big shout out to the folks at JGI, Hudson Alpha, and the University of Arizona who have been doing a lot of the heavy lifting here. So the two examples I'm going to talk about is work that we've been doing in the poales to understand, to generate genomes, and then use those genomes to understand important biochemical processes, including the production of lignin, biosynthesis of lignin. Most plants are making lignin, which is a key component in cell walls and other important structures in plants. By taking phenylalanine and converting phenylalanine to cinnamonic acid and then on to comuric acid and lignin. What the grasses have been able to do is they're able to use tyrosine as a precursor and convert tyrosine then to comuric acid, then feed that into lignin. Grasses are able to use both these pathways to make lignin and related compounds. And so we were curious, when did this ability arise? What happened? What gene? It was a gene duplication or genome duplication responsible for this new function that had been characterized in the grasses, but not in other plants. And it turns out that that not only grasses, which are shown up here in blue, but also other graminids, Ectiocloe and Joinvillea, which are non-grasses, but they look a lot like grasses. We call them graminids. Are also able to use tyrosine as a precursor for lignin biosynthesis. And so, where we at first thought that all grasses share a genome duplication event in their ancestor, and we thought, well, maybe that genome duplication event is what gave rise to this ability to use both phenylalanine and tyrosine for production of lignin. But no, it turned out that it was a tandem duplication event in the last common ancestor of all the species above this star here that had that gave rise to one copy doing using being able to use both tyrosine and phenylalanine. So here we see this is a gene tree showing the relationships of these those genes derived from that duplication event. In light blue are these non-grass graminaids, and dark blue are the grasses, and we see that these species that are shown here have the ability. They have one copy, this p-tal copy, that is able to again process tyrosine in order to make phenylalanine. These are the other genes, the PAL gene. So one of the duplicates one of the genes that came out of this duplication event just maintained the ability to process phenylalanine and not tyrosine. So using molecular evolutionary analyses, looking at our multiple sequence alignments, we were able to identify conserved sequences that in the phenylalanine, in the PALs, the phenylalanine liases, that had changed in the p-tals, and we hypothesized those might be responsible for this new ability of this gene to process tyrosine. And then work by Yuri Takada Kamura and Hiroshi Maeda's lab, they did site-directed mutagenesis and showed that actually, if we do one mutation, and it turns out to be in the active site here, right here in this AlphaFold picture, you are able to really increase the ability of this phenylalanine liase to process tyrosine. And a second mutation that could be one of a couple second mutations really makes it so this gene is able to work both with tyrosine and phenylalanine. So again, the genome sequence gave us a sense of when the gene duplication events occurred for these species, when the gene duplication events occurred, and then digging into those sequences, we were to see, well, what changes following duplication events gave rise to this new function. Similarly, there are other functions. So I'm just going to highlight one other here. So in grasses, their endosperm, they are making a lot of starch. And the reason they're able to make so much starch and provision it to their endosperm, among other things, is that they have this dual starch production ability, analogous to what we saw with the p-tal being able to process two paths to ligand biosynthesis. Here we've got genes, actually a whole pathway of genes that are able to work both in the cytoplasm and in the plastids where starch production is typically occurring. And so we see that this isn't happening in Joinvillea and Ecdyocloa. It's only happening within the grasses. And so we hypothesize, well, maybe the gene duplication events that allowed this starch biosynthesis to occur in the cytoplasm may have had something to do with this whole genome duplication event. And the last common ancestor of all grasses, about 100 million years ago, plus or minus 10 or 15 million years. And so that would include this so-called basal group, Ferris, and then Pluidae. Pluidae is a subfamily within the grasses that only exists, that is only found, is endemic to Africa. And I'm showing down here the distributions of Gudella and Puella, and they both include multiple species, but again, all centered in Africa. And we do not have any genome sequences for any of this. The community does not have any genome sequences for any of these. So just to, this sequencing this genome would really, could be really elucidate, it could really help us elucidate, well, what was the success of this core, why do we have this core POAC group really much more diverse than these other so-called basal subfamilies of the POAC, the grass family, and how what can it tell us about the things that make crop grasses like maize and wheat and rice so successful in serving our needs. I'm going to end just with a couple of slides on this one last, on another system involving legumes. So legumes, again, is a big family, including many economically important and agriculturally important species. The Leguminaceae, we think its ancestry dates back to about 60 to 65 million years ago. Again, it's a big family with multiple subfamilies. And so we generated in a collaboration with these folks at Iowa State and the University of Saskatchewan and others, genome sequences for Chamaecrista up here and Cercis down here. And it turns out that this analysis of these genomes indicated to us that whereas most legume lineages have undergone whole genome duplications or polyploidy events, like we were hearing about from George, Cercis has not. So it has got a haploid chromosome number of 7, and we estimate, we infer that might have been the ancestral chromosome number for all legumes and perhaps all core eudicots. And we see that in these other species that have had genome duplication events in their ancestry, if we look at any one color, we're seeing that they're showing up in multiple chromosomes, suggesting that there's been a genome doubling, and other analyses are showing us this. So for instance, we see blue here, and we see blue over here, down there. So it's suggesting that this ancestral chromosome 1 has been duplicated into multiple chromosomes. And so this is a picture that shows us this. Again, the papilionoids, including Medicago and Glycine, soy, lots of other important species. It has in its ancestry a genome duplication event. The sesapinoids, including Chamaecrista and Mimosa, other things, has a genome duplication event in its ancestry, very interestingly, maybe involving an ancestor of the papilionoidea. And then Cercis does not have a duplication event, but Behainia, it also in the Cercidae does. And then this other subfamily has yet another genome duplication event. What I'm showing here is Dipicartia is a single species in this subfamily, Dipicartoideae, and we don't have a genome for it. We don't know what its ploidy is, and having a genome sequence for that could really inform us about the evolution of genomes early in the history of legumes, and ultimately be of importance for better improving legume crops. So here's a beautiful picture of Dipicartia. It's a vine that's growing in West Africa here, and it's just a beautiful species. And again, the Africa BioGenome Project is really the group that should be sequencing these two taxa that I mentioned. So I'll leave it there, and I'm very happy to take any questions either now or during the panel discussion. Thank you.
Yeah, thank you, Jim. So I think we'll just take some questions. We have about 10 minutes for question marks before we move to the next session. So I'm just going to bring up, maybe it's Jim, if you stop sharing, then I will bring up Zara, and yeah. So, do we still have Zara? Yeah. So I think George and Zara, I say you guys are already responding to questions. So, yeah. I don't know if there are any questions on site or online. Juliet or Genevieve, is there a question on site for either George, Zara, and Jim before I get the questions from Zoom asked?
Is any question? Okay. Yeah, there is.
Okay. We can't see the on-site team. So IT, if you can just remove the banner so we can see the hall or something. Okay, please ask the question while you're doing that.
Okay, thank you. My question goes to Zara. AI in agriculture, of course, we know that would depend on data, sharing data on farm records, soil data, weather patterns, production figures, and maybe also personal information. So I have a question. So who now owns the data? Okay, this data that will be shared on AI platforms, who owns this data? Now, in your presentation, I saw a very beautiful, one of your slides where we saw drones, and I think I did see smart sensors. So for a young manager who is trying to tell the board on the need to integrate AI into their farming, and how cost effective is it to have these gadgets, drones, smart sensors for a farm that is not so large? And I also have another worry. I don't know how we look at this. I want to believe that there are still local farmers who could look into the sky and tell you the rain is coming and it's going to rain for days. Now, when we turn to technology and we depend on technology, are we not going to put away indigenous knowledge? And then gradually we'll have generations that are not able to interpret what we have. Now, we also know that AI hallucinates. So how sure are we that what we have is accurate without bias? Also knowing that some of these models are Eurocentric, and maybe have been trained with European data. And there's also an issue of cybersecurity. So these are concerns I've raised. I don't know what you have to say to them. Thank you.
Okay, so those are many questions. I'll try to address all of them. Okay, the first one pertains to the data. Your data belongs to you as long as you don't put it in a public domain. If tomorrow you want to use AI with your data, you don't have to put it in a public domain. You can easily use existing data together with your data through something known as transfer learning. And in that case, you are training the model using existing data and your data, and the data still belongs to you because you're using transfer learning and it's on a local machine. So in that case, the data belongs to you. If we are going to put the data in a central repository, then obviously there are other issues that come into play. But this is why on my slide, I said there should be regulations in place, data governance, and there should be data privacy as well, regulations pertaining to this. This is for the first part of the question. Now, how cost effective is it to use AI? And sensors and so on. Now, those sensors, some of them, they are not that expensive. Drones, yes, they can be more expensive, but I suppose it all depends on how big your farm is. If it's going to be a small farm, then maybe it's not worth using too much technology or investing too much in technology. But again, the person who wants to do this has to see how much it's going to cost to implement sensors and to have drones and to collect data and so on, and then compare to, for example, what is the profit, what is the possible profit that may be obtained. So that's for the part for the costing. You've also asked a question about farmers being able to look at the sky and being able to predict whether it's going to rain. And I completely agree with you. This is why on one of my slides, I said AI will need to work with intuition and experience. And I said, we don't want an AI which uses only sensor data and drone data, but we want a combination of both. And this is not an easy task. Doing one which uses only sensor data is quite straightforward, to be honest. But if you're going to use one which uses intuition and experience, that's more challenging. But it can be done. And this is why I have this on one of my slides. And your last question pertains to hallucination. But remember, all of you, when we speak about AI, we are not only talking about LLM. We are talking about deep learning. We're talking about machine learning. Hallucination is mostly an LLM problem. Hallucination is mostly when you deal with ChatGPT, Copilot, and so on and so forth. When we talk about AI, we don't necessarily mean LLM. And when we talk about AI for agriculture, it's a combination. Yes, you can potentially use LLM, but it's also a lot of computer vision, image processing, object detection, which is YOLO, and deep learning and machine learning. And those, they do not hallucinate because they work differently. They've been trained differently.
Yeah.
And I hope I've answered all the questions.
Yeah,
Thank God.
thank
Have
you.
I missed anything?
No, you haven't. I just wanted to add something just to stir up the conversation as well. In the direction of cost, there is a report that just came out today. Maybe a report, maybe a briefing or something. But I also read it last week, where some AI platforms are now increasing the cost for you to use their model or run things, or maybe their token. Token is more like around, when you have a sentence, you break it up, that's kind of in a very simple definition. So now the costs are going up, whether it be in Copilot. So I think Copilot did say that last week, and now several other organizations are kind of saying that. So, and one of the points in the commentary that some people are making now, including one published today, is that that when LLM came out, when I mean came out, I mean when there's this buzz around LLM around 2022, 2023. So the question is, it's going to reduce, it's going to make us code faster, for example. It's going to, if you have 10 software engineers, then you only just need one, things like that. But now with all of these tokens subscription increasing and all of the cost on AI, because it uses a lot of energy, the cost of hiring those 10 engineers versus the cost of also paying for the tokens and everything is now getting equivalent, is now getting at the same level, or even, in fact, the cost of even using the token is now more. So in the report, one of the questions is, what is now the net gain? You get my point. So there's that, and I think that might also be it might be tangential to the point the gentleman also was making in terms of what is the cost and how do you kind of look at it. But I suppose the net gain perhaps could come from, maybe the way I see it is that on small tasks, and as you mentioned, Zara, AI it's like using a sledgehammer to kill an ant, right? But as the task becomes so large, you have large data set, then the cost now makes sense, whether or not it's, kind of, I think that's just what I wanted to add to that. So I have a few questions. I don't know, on the on-site participant, do you still have more questions? Otherwise, I just ask two questions to Jim and George.
More questions from the floor? Any more questions?
None, right? Okay.
None.
Okay, thanks. So I just have, George, I see you've been answering some of the questions, so I'll just ask you both one question each, so we can move to the next session. I did put it in the chat box. Now I can't find it. Okay, I think, George, I see one of the questions. So in your presentation, you talked about polyploidy estimation, right? I suppose this is new, right? This is a new project, or has it been around for a long time?
I think our approach is new, but also not like that groundbreaking in the sense the type of machine learning I'm doing, the statistics are based in the 80s for the most part.
I know. I'm just talking about the actual tool itself.
Yeah,
So,
but But I think the goal has been a long-standing one, and the
Yeah.
idea that we can actually unlock that type of information where if you have in your local herbarium populations from that same place collected 50 years ago,
Mm.
you're like, I want to look at, was there a shift in polyploidy? Was there a gain in, say, herbicide resistance genes in these populations that are potentially crop pests or weeds? Those are things we can do now and try to understand what's the mechanism and how polyploidy affects those selection tests and so on.
Yeah. But I think, but for me now, the question would be, how does this, because in the presentation, I was trying to see how this differs, maybe I might be wrong, but how this differs from other polyploidy estimation tools. Maybe perhaps they don't use the same technique, but it would be nice to look at both in the content. An example, if I'm correct, like Genome Scope does similar estimation or smudge plot and all of those things.
Yes.
So how does this differ, and how did you do a benchmarking with both of them and things? And is this also being used already in the community? So it's to be nice to kind of have those juxtaposition.
Yeah, so I skipped a lot of that just for the sake of time. One, yes, there's things like smudge plot or other K-mer-based methods that will work really well if you have whole genomes. The thing I kind of didn't mention about the data sets we were looking at, they were either target enrichment data, so like 350 loci where you don't get those nice K-mer distributions, or RAD-seq data. So we're trying to make this a process that fits with biodiversity data a lot more easily, especially for these types of early exploratory analyses. And in the tests we had with some other methods, we did get better classification accuracy. So we did improve upon the previous probabilistic tests. So this is all brand new. I kind of polished it a little bit today just for the talk, but it's going through the process of getting reviewed now, but it's online and available. And we've done some benchmarking tests in like sweet potato and other crop systems where we know the distribution really well.
So what would be of interest to us in the Africa BioGenome Project is, so first of all, you're saying that this cannot be propagated to the genome level, right? Is that the...
So it certainly can. We can use it with whole genomes. I think it's if you have really nice whole, like deep coverage whole genome sequence data, you do have other options available to you. But so I'll say from my perspective, studying grasses, whether we want to talk about their ecological importance, a lot of the forage grasses come out of Africa. When you talk about Eurocloa, that is East African grass, and now it's the biggest one in Brazil. And we'll probably see it in the US eventually.
Yeah, so what would happen? Do you have this paper in, because I want to get my colleagues in the Africa BioGenome Project to have a look to kind of try to, because we use smudge plot, genome scope, and other people used other familiar tools. So it might be what if you share it with me?
Yeah, I can send you a link to the 2024 paper where a lot of this was in a supplemental document. And the new one is coming out. So
yeah, I mean, if you also have the new one, which is, if it's in preprint, then please feel free to share it. Okay, thanks, George. Jim, I think we have just one question before we move to the next session. It looks like you've answered the question that I have, Jim. So essentially, I actually like the link you have in the, that talks about the plants. I know in 2021, 2022, the number of plant sequence at that time, because I know I wrote about it, it's around 800. So it's good to see that that number has increased up to 2,600. So for me, what would be of interest is to see how things have evolved from that time up till now, and see, where does the Africa continent also sit. So that, okay, I see you sent the website, but I need to then check whether it's categorized where the data is coming from versus who is submitting things, and if one taxa is overrepresented versus the other. So I'll just go through I've just sent the link now, probably just go through it as well. So except you want to add,
it's scraping publications. So it's just grabbing these publications and posting them. So it really doesn't have, there's not a database that would allow you to break it down by source. It is broken down. You'll notice that there's a tree view. So it is breaking things down by taxon. That could be of interest. But yeah, I mean, Africa BioGenome Project plant genomes are now coming, and so they're contributing to this increase.
And in that case, then I mean, also just answered my second question that it's not goat, because when you talked about it, I was like, okay, how does this differ from goat? Is this now taxa specific goat, kind of in Germany? But I think it appears it doesn't do what the goat platform does. Okay, I think I'm noting that we have 5-7 minutes off. So does anyone have any final question for Jim, Zara, and George? Otherwise, we'll move to the next session. Thank you, Jim and George, and also Zara for presenting. And we look forward to having you all next time as well. Okay.
Thank you.
All right. So we'll move to the next session. The next session is on AI for crops and livestock improvement and health. So do we have Professor Vincent on the call? Okay, we have him. I'm just going to make him co-host. So Professor Vincent, please feel free to share your screen.
Yes, let me do that right now. You able to see it?
It's coming up. Now it's up. So you need to put it in a slide mode, please.
Okay, let me do that.
Oh,
okay.
I see. Sorry, I see Tobias has his hand raised up. Tobias,
who's got his hand raised up?
I think Tobias. Tobias, is this for this current session or the previous section? So I must note that. Or do you want to leave your answer for the next session? Your question for the next session?
Mute me so that you hear me.
Yeah.
Thank you very much. I've been raising hand because during the last session before the launch, one of the speakers who said is the technical advisor to the director of biogenome, whatever, Nigerian biosafety. So I had a question, and incidentally, the whole questions were set to be put on hold until
tomorrow.
Okay, I thought you said until after lunch.
Yeah. So no, it's on the tomorrow because those questions are more policy oriented. Tomorrow we are focusing on policy. Today we are focusing on the science and technology, because otherwise we will discuss, what we are supposed to discuss tomorrow. We are discussing it today. Then it doesn't. It affects the flow of the activity. If that's okay, can we take this tomorrow, please? We'll have a whole day for policy related questions.
It's okay.
Yeah, except you have a question for Jim and George and Zara.
No. It is that question was for that man. It
okay.
Just relates to policy.
All right, thanks. So I can see Mamoni. Please unmute. Okay, now Mamoni. All right, let's go ahead, Professor Vincent.
Okay. Greetings, everybody. I'm speaking from the US here, but I'm part of the University of Port Harcourt, faculty. So let me thank the organizers to start with for putting this together, and it's quite innovative that we are doing this. So let us start, from where the science begins. Can you see the slides okay?
Yes, please carry on.
Okay, wonderful, yes. So my talk is about the intersection of digital sequence information and pharmacogenomics in the age of artificial intelligence. And I come from the clinical background, but I've been doing quite a bit of work in terms of research with my basic science colleague. because translation, which is pretty much from bench to bedside, bedside back to bench. And there is no way you're going to be doing these, doing translation without working with your basic science colleagues. So that's why I'm quite happy that, even though I'm from the clinical background, I was invited, to participate, participate in this. So the central idea is that the genetic data are no longer static laboratory findings. They are digital assets that can power AI models, clinical decisions, and drug development. For Africa, this creates opportunity and some challenges because the continent's genomic richness can advance global science if African institutions help govern how those data are used. So that's why I'm so glad we're doing this. So the scientific community actually begins with the basic facts. Because African genomic diversity offers a powerful basis for discovery. So to understand why this matters, we must understand the genetic variation, which of course I stated earlier. So this particular slide here talked about the paradox. The paradox here is this, that Africa contains the greatest human genomic diversity in the world, yet African genetic sequences remain poorly represented in the databases that support AI research and modern drug development. So that's why I call this, the paradox. The most diverse source of human variation is not proportionally shaping the tools increasingly to understand disease and design treatment. So as a result, scientific models may be incomplete, and the populations with the richest diversity may receive the least benefit from discovery. You already all know, because I've been hearing the few times I clicked in to listen to this, you've been talking about digital sequencing informations and on and on. So that's an area where you're quite familiar with. So I'm not going to go over what you've already discussed. I'm just going to go straight to the main facts. So now the next slide, which is about the scientific community in terms of what needs to be done, is that the African digital sequence information can reveal population-specific differences in drug development, in drug metabolism, risk of adverse drug reactions, a therapeutic response, and precision dosing, which is quite key, which is what I've been involved with over the years in terms of precision medicine. So these are not minor scientific curiosities.
Sorry, Professor Vincent, do you want to speak to the mic? Because you're breaking off in between. So if you speak to the laptop or mic, that would be helpful. Thank you.
Okay, yeah. So can you hear me okay now?
Yes, please carry on. Yeah, carry on.
We can, volume? Okay.
We can hear you now. Just speak to the mic, and that would be very helpful. Thanks. Yeah.
All right, no problem. Yes. So talking about the scientific committees, I did mention about the precision dosing requirements, which is in this era that we are looking at this, looking at the genomics and plants and humans. That's where precision medicine plays a big role. So definitely what you're doing today and then translating it to what's happening in humans definitely will help the concept, precision medicine. So the next slide, then I talked about when genomic diversity is ignored, lots of things do happen that are not good. For example, the adverse drug reactions and ineffective treatment that we've seen across the African population, not just the African population, but the world as a whole. And a good example is the HLA-B 5701 allele that was involved in terms of toxicity with a drug called abacavir, which I was involved with when I was in a phase 3 clinical trials. So that becomes an issue where when we ignore those kind of things, then becomes quite difficult for us to handle. The next one is about pain management. A lot of the world now are involved with these pain management decisions in terms of opioids, where opioids use is probably used globally all around the place. And there are variations in the cytochrome P450 system, especially the CYP2 6 differences, across the African population. So which of course is going to lead to inadequate pain management or increased side effects. So those are the things that happen when the genomic diversity is ignored. Again, the misdiagnosis of inherited diseases, like I've just mentioned, had the hypertrophic cardiomyopathy, which of course we've also seen in the African population. And then missed opportunities for disease control, like sickle cells, modifiers, and on. So those are the things in terms of the clinical. I just wanted to cite some few examples, since I know I have about 28 minutes or so to discuss this. And just to give you, because normally a keynote is to enable you to pontificate so that we can put all the issues together that will now lead to what we're doing or where we need to be headed. So I've talked quite a bit about the genomic diversity, which is actually the foundation, precision medicine. Most precision medicine tools were developed using populations that are predominantly of European ancestry. So because we were developed based on that, the databases came from European ancestry. So it's a little bit difficult then to perform reliable studies or reliable stuff in other parts of the world with different allelic frequencies. So that's why it's very important that African genomic diversity is therefore essential to improving the validity of precision medicine. So the goal is not simply inclusion of fairness alone, but the development of therapies and dosing that are genuinely precise and all over the place. If you have this data going on and it's not used in a particular population, then for me it's really not, you're not really moving forward to handle the issues that is supposed to be. So which of course, dovetail into the concept of global health. So there is a scientific basis asset of global significance when it comes down to that. So now, what's going to be the role for the African BioGenome Project? A lot to do here, because the platform for building a continent-wide genomic ecosystem is very, very important, because that means that we are creating regional nodes, collaborative scientific network, and training opportunities, infrastructure. So the project, which of course you're involved with now, is not limited to gathering data. Its larger purpose is to build the capacity needed for African institutions to generate knowledge, govern genomic resources, and participate from a position of strength. That's the key. That's what I wanted to point out in this particular case. In that sense, African BioGenome Project is a strategy for scientific coordination, self-determination, and long-term leadership. So let's not work out if we're thinking that we're just generating data. We need to actually take a much stronger role in terms of leadership. And I think it's key. And I want to make this clear, where I put this slide here, that the strategic scientific infrastructure that protects Africa while enabling responsible global science, it is not a charity. It is not a charity. You got to put together your strategy. African genome science should not be treated as a charitable inclusion project. It is a strategic investment in scientific infrastructure and negotiating power. So building African capacity in sequencing, analysis, governance, and collaboration allows African institutions to help define research priorities and share in the value created from African data. That approach protects Africans' interests while still supporting responsible global science and equitable international partnership. So that point needs to be very clear-cut, because sometimes people walk in there thinking that they're just there to collect data. I was part of the University of Illinois a medical faculty for a long time, over 20 years before I moved to the University of Port Harcourt to start getting involved in what is happening. So, and I've noticed that over the years that sometimes we just think that we're just there to collect data. Now, I did divide the talk into 3 parts, and then the other part, the second part, is about policy and governance, which of course we also need as scientists. We also need to realize that we have a role to play when it comes down to policy and governance. Because sometimes we just feel like we are so busy and then we should leave policy and governance in the hands of other people because we don't think it concerns us. It definitely is something we need to be very involved with. We did that in the era of HIV/AIDS when I was involved with a lot of work in terms of clinical trials with HIV/AIDS, with the antiretroviral therapy. And we did that a lot. In fact, it was because of policy and governance that the concept of PEPFAR, came into place, because there was a very important part of advocacy. So that's why I put this particular stuff there. So the genomic material becomes a digital sequence information. The key issue is no longer only scientific discovery, but also who controls the access, who benefits, and what rules govern use. So policy and governance create the framework for consent, benefit sharing, and fair collaboration. Because these questions cross borders, African nations need a shared approach that allows them not just to participate in global science, but to lead within. And that's the key, the key. And of course, there are opportunities. The opportunity is quite whole because the biological observation is there. It is a resource with scientific, clinical, and strategic value. So calling it a global scientific asset means it's got implications for how future knowledge, diagnostics, and therapies are built. And I've had the few times I logged in, I've seen some presentations that discuss some of these things, which of course I will definitely advocate that should be carried on. Now, how do we move forward? Well, there've been some international framework, so it's not like it's completely new, whether we can use to at least base our data and then start exploring. And the two ones I picked out here that supports African interest was pretty much the Convention of Biological Diversity and then the Kimmel-Montreal framework, which of course I'm sure, I don't know if you don't talk about it today, I'm sure perhaps somebody's going to be talking about it, tomorrow during the policy sessions. So the Convention of Biological Diversity recognizes the sovereign rights over biological resource and supports the principle that benefits should return to the country of origin. And that's why I think this framework is quite important. While the Kimmel-Montreal framework extends the logic into the era of digital sequence information by linking that to fair benefit sharing obligations through 2030. So together, these arrangements, agreements show that the legal and policy groundwork is already taking shape, even though there are no legal backings yet. But the fact that we are doing something about it is a good sign, which I believe that we should carry on from there. Then which of course dovetails into the Nagoya Protocol, which is the operational mechanism for access and benefit sharing under the Convention of Biological Diversity. So, which I'm sure is going to be discussed during the session too. And the biodiversity governance framework is key here because it's because primarily the Convention of Biological Framework and Nagoya Protocol helps to put this together because they are increasingly relevant to pharmacogenomics. So if in case you're wondering, what does that SCADA tie in with pharmacogenomics? Yeah, they're relevant to it to AI, okay? Because the diversity governance framework should dictate how genetic data are shared and how access to this. And because the key here, the most important thing is that it creates a policy gap where providers of biological resources may not receive equitable benefits. So once we put this stuff together, that gap, we can now shrink the gap. So the core issue is that technology allows researchers to develop new drugs without access to the original plant and animal and biological specimens. But with this policy, now you could put this together so that some people are not left out. So that's why the concept of good governance, becomes a key. Because the goal of modern diversity governance is to keep data open while sharing, to ensure that scientific progress does not come at the expense of equitable partnership. That's the key. So these three goals I've listed here in terms of sovereignty, benefit sharing, and open exchange should be things that we should be discussing all through this period that this workshop, is going on. I know as scientists, we tend to focus a lot, on the science, but definitely all through this period, we should be discussing these because we all need to be on the same table. So Africa needs to be at the table and shaping what comes next. And that's why I'm a big proponent of what's happening here today, and we'll continue to get it going. And the West African Regional Node, University of Port Harcourt, which of course is hosting this, is already taking charge. And I think that's what we want other institutions to do because they're already doing that. So I'm sure you're going to be interacting with a lot of folks there who are part of the planning of University of Port Harcourt and have a chance to interact with them as far as how this local action will now lead to a continental impact. So every center, no matter where you are around the world, that you're listening in or you're participating directly, at the University of Port Harcourt, we must have to have regional nodes. East Africans probably will also have their own node. The same thing with South Africa. The same thing with already West Africa is doing thing, and then Central Africa and on and on. So having said that, so now let's get into implementation, because implementation happens to be quite a big challenge, especially for those of us who are scientists. I did mention that part of my earlier work, while I was in the United States, was about translational research, which is pretty much from bench to bedside back to the bench, and then using the concept of implementation science. So implementation science, which is quite a new area here, a new concept, because it's so contextual that sometimes we don't seem to understand it well. Because scientific discovery and governance framework are necessary, but they do not automatically improve patient care. So implementation is a phase where evidence must be translated into systems, workflows, institutions that can use it reliably. So the phrase from discovery to practice captures this movement. So the purpose of implementation is to make sure that genomic insights, AI tools, policy frameworks result in real clinical change rather than remaining only in publication databases or conference discussions like what we're doing today. It must result in real clinical change. So I wanted to emphasize that. Of course, there's going to be challenges when it comes down to this, but we don't have to let our challenges discourage us. And because AI may identify meaningful genomic patterns, but without a deliberate implementation strategy, those patterns never affect diagnosis, prescribing, or patient outcomes. So this is why implementation matters. The challenge is not only to produce new knowledge, but also to make health systems capable of using it. So without that step, scientific progress remains conceptually impressive, but clinically irrelevant. And I think we want to make sure this is relevant. That's why I had this part in discussing implementation science, which is the framework that translates evidence into clinical reality. Well, this is no news to this group because you've been doing quite a bit of computational work, listening to what some of the few sessions I listened to today. So this is not news to you in terms of digital infrastructure. So I'm not going to be spending time, talking about this because you're already familiar with this. But I just wanted to. The few slides left talked about some few things in terms of how you're going to harmonize these. AI models require consistent inputs, and then the clinical decision support, which of course means that real-time pharmacogenomic guidance embedded in workflow. And that's the part, those of us who are in the clinical sciences, this is where it becomes quite important to us, because the real clinical value of pharmacogenomics appears when genomic information can guide treatment decisions at the point of care. which is what we're doing now in most of the developed world. At the point of care, decisions have been made. And then the clinical decision support will do this by embedding relevant guidance into provider workflow. So instead of requiring clinicians to search separately for genomic interpretation, the system presents useful recommendations in real time during prescribing or treatment planning. And then this turns genomic knowledge into actionable tools. And this is not just to admire genomic complexity, but to improve safety, effectiveness, and precision in everyday care. Because at the end of the day, what matters based on all the stuff we're doing now, we want to prolong life. We want people to live long and do the wonderful things you're doing so far, okay? And not Africa be a place where everything about bad data regarding healthcare is a place where people are giving examples to. I can bet you, like the country where I come from, which is Nigeria, the average life expectancy of a male, both male and females, they all between the age of between 55 and 58. Okay, so you could see that we still have a long way to go when it comes to that. While the average life expectancy in the United States, where I'm speaking from right now, at the moment, you could see that it's in the 80s. When you look at the entire population. So that's why I think I might say wanted to take some time to discuss this clinical decision support, not just because it is my own area of interest, but because I think it's quite vital. So capacity building, you're already doing that based on what is happening today at University of Port Harcourt and those now who are calling in, which is key. The deliverables here is that the end goal is not simply more research, output, or more sequence files stored in databases. The deliverable is the implementation structure that converts African genomic data into actual patient care. Okay, that means systems, skills, governance tools, all working together. The phrase built by Africa, benefiting all, captures both authorship and reach. Africa is not only contributing raw materials, it is helping build the structure through genomic medicine becomes useful. That I wanted to get that clear in terms of deliverable. Of course, we do have multi-state collaboration, which I did put here, called the triple helix model and beyond. The triple helix model here is academia, industry, and government, working together, not working in silos, which of course we see quite a bit here because these groups are working in silos and they are not talking to each other. And then the beyond here, because I've added the civil society, because the civil society provides the ethical oversight and keeps equity in view. So none of these actors is sufficient on its own. So the point of collaboration model is to connect scientific capacity, product development, public accountability, and governance to that of genomic innovation becomes both effective and legitimate. So yeah, no matter where you are or you're practicing from, please try to see where you can adopt this triple helix model. Because what I've noticed around the world, when I travel around and engage, is this that everybody's working in silos. The academic people are working on doing their own thing. Industry is doing their own thing. Government the same thing. So until we adopt this, then we know for sure that things are going to be moving forward. For the African government, we need to ratify and implement some of this framework, invest in national genomic strategies, which means that we need to educate the government in terms of how important these things are, then establish data governance authorities, and then fund infrastructure, not just a research output. So that will be for government. And then for academic institutions, we need to continue what we're doing, participate actively in the African BioGenome networks, engage as co-developers in international partnership. So those are the things. These slides that I'm presenting here, I've already sent them to the organizers, so they can actually share the slides, with you, after my presentation. Then for industry partners, they need to engage institutions like what they've done today, because I've seen some of the people who were involved in terms of funding. They need to commit to benefit sharing agreements. They need to support infrastructure development and capacity and respect African leadership in priority settings. That's very, very important, for the industry partners. Then for the international funders, international funders shape scientific system by deciding what kinds of work are rewarded. So if they focus only on short-term projects and rapid outputs, they reinforce fragile capacity, which is what I've seen over the years. So if they fund long-term institutions, infrastructure, and human development, they help create durable leadership. Okay, so that is going to be the actionable recommendations, for the international funders. Then, in terms of the AI deployment pathways, it must be scientifically rigorous. I've seen one of the fellows here who asked the question was talking about AI hallucinating and things of that sort. And then the speaker basically answered the question. And also, Dr. Ebenezer did chip in and then answered some of the questions and did narrate how these things should be done. It should be ethically grounded and then oriented towards public health. Then the way forward is that the future described here is that African authorship rather than just passive inclusion. So saying that Africa is building the table means that African institutions intend to shape the rules, priorities, and infrastructure of genomics and precision medicine rather than simply seek a place within systems designed elsewhere. So the broader point is that scientific leadership grows from coordinated institutions, policy clarity, and strategic investment, which is quite key. Then for Vision 2030, I've already mentioned the regional nodes, and then in terms of what in African the DSI should be integrated ethically into global AI training, clinical support team, and One Health genomic ecosystem should be fully active. It must be very fully active. The vision combines scientific governance, infrastructure, and care delivery. It offers a measurable picture of success in which African leadership produces both continental capacity and global relevance. So the commitment must be there because the core commitment is to ensure that African genomic assets and digital sequence resources lead to tangible and equitable health gains for African populations. The science is already available. The governance framework is emerging, and the institutional foundation is developing. Because look at what we've seen today from today's program. Well put together, and things you could see that things that institutions are rising. So that means that the remaining task is action. The emphasis here is on responsibility, valuable data, and promising ideas are not enough unless they are converted into systems and policies that improve health in real communities. And I think that's my last slide. And I wanted to thank the presenters. I wanted to thank the organizers for inviting me to participate in this, even though I'm not a bench research, and I'm coming from the clinical research angle, but I'm so glad that I was invited that I can share some of my own views and the way forward when it comes down to this stuff in the age of artificial intelligence and pharmacogenomics. Thank you very much. I'll be glad to entertain questions or suggestions based on what you've had this afternoon, Nigerian time, or this morning, American time. Thank you very much.
Yeah, thank you so much, Professor Vincent Idemo. Just hang on the call. We are going to have one more speaker, and I think that is the last speaker for the day, and then we'll wrap up. And then after his presentation, we would have a panel session Q&A. So do we have choosing Ekene Obi on the call, please? Choosing, okay, we have, okay, I'm gonna ask him to can be a co-host now. So choosing, please unmute yourself, share your screen, and make your presentation.
Thank you, thank God, and good day, everyone. Can you all hear me?
Yes, please carry on.
Oh, great. Share my slide. So my name is Chosen. I'm a doctoral researcher at the University of Arizona, and just want to say thank you to the organizers for having me today. In my talk, really, I just want to highlight some of the opportunities that I see on how we can leverage AI in advancing African biodiversity, and then some of the challenges that we need to be thinking about. So as some of the previous speaker already highlighted, in Africa, we have, a paradox, which is that we, hold roughly a quarter of the world's biodiversity, but a lot of this still remains uncharacterized. And we need to begin to change this narrative, which the African biogenome is already doing. And one of the things really that I think about sometimes, in bridging the sequencing gap, is really targeting some of the varieties and some of the species and varieties, that everyday, Africans depend on. And for example, the sleeper fish, which some people from the Port Harcourt region might know. I grew up, eating this fish. And it's still surprising that currently we have very little genomic data on this. And this also is applicable across a lot of different plant species. So really, if we are to begin to breed resilient varieties and monitor genetic erosions, there is need to sequence this data. And some speakers before me has already, highlighted that, the cost of sequencing is really, really dropping. And as you can see from the figure on my right-hand side, which is comparing the drop of the cost of sequencing the human genome since the inception in the early 2000s, and right now that Ultima Genomics in the last couple of years is now has proposed that you can sequence the human genome for $100, and some sequencing companies are currently even targeting $80. So the issue really, it's not on generating sequences, but how can we gain biological insights into this data that we're generating? And it's one of the things that I know that this Congress is really thinking about. And so here I want to highlight some areas that I think that we can apply AI. And basically, it is in step 3 to 6, because a lot of the African endemic species are often crops, and they have very few related species that have been sequenced at this time. So I think that AI will be very applicable in genome annotation and sequence and also assembly, and also in doing some comparative genomics. And I'll highlight some of these tools that I'm currently using in my own research, because I work with Arizona Genomic Institute, and I'm currently working on some genomes in the Tumamoka genomes that are very endemic. They're endemic in the Sonora Desert, but then have very few related species that have been sequenced. So there are a couple of foundation models that are out there already that I think would be handy. One of them is EVO 1 and 2. EVO 2 was recently published. And one of the things, again, that I'd like to highlight, because somebody talked about hallucination and all of that, is that foundational models helps to reduce the chances of this. But again, you have some issues where some models are predicting like one nucleotide exons and all of that. So my recommendation is that as we adopt some of these models in our work, we do not throw away the older methods, but that this, adopting these models would really facilitate, one, how we annotate genomes and bridging the gaps of predicting proteins and other functional genes. So we have a challenge, like I previously mentioned, that the traditional tools that we have at the moment often require related sequences in their annotation, and this is often missing in thousands of often species that are found in Africa. So what's the solution? The solutions are a couple of foundational models that I've mentioned. Specifically, right now, I'm using Helixer in my research, and I find it to be amazingly accurate. And I think this is one model that we can readily adopt in the African BioGenome Project to annotate genomes that have been sequenced and also possibly reduce the cost of investing in generating RNA-seq or proteome data. And so the opportunity here is that we can train or fine-tune these models, using data that is focused on crops that are sequenced that are very related to the African species. And so instead of spending a lot of time and waiting to generate some of these other data that I've mentioned, we can go ahead and use these AI models. And when these other data are available, for example, we can compare the accuracy of both approach. Another opportunity I see that I wish was there, for example, a decade ago when I was an undergraduate in Nigeria doing DNA barcoding, is some multi-model models that are there. For example, BioClip, that is becoming really, really handy in monitoring species migration and also identifying herbarium samples. And as I previously mentioned, a couple of years ago, worked in a number of DNA barcoding projects on plants and mushrooms. And one of the gaps, that was there that made me come to the US is the fact that there was always a gap of those analyzing the sequence and gaining insight into them. But beyond that, there was also the gap of identification. Getting a lot of trained taxonomists was a big issue. And right now, I think even if you look at the data, there are very few university programs that are offering programs on plant taxonomy and animal taxonomy, because a lot of these universities are now rebranding in order to get more students. So I think that AI will be very handy in these areas of research in identification of species and tracking how the species migrate over time. And then there is also some applications in environmental DNA that some models are able to predict the distributions of different species in environmental DNA, just by feeding the sequence data that are generated from the metagenomics. And I think these are things that really we can do, leveraging existing models, first of all, and then moving over to the level of fine-tuning existing models. And then also there are deep learning models and also multi-models that are focused on acoustic monitoring. So some specific things that I think about, and I was really thinking about when I was given this topic to speak, is that we know that the biodiversity that we have still remain really largely uncharacterized. And I think that there is a potential that there are a lot of novel proteins in this biodiversity that haven't been characterized. And that's why I really liked the talk of Jim and George, where they're going beyond identifying this species and sequencing the genome and asking the questions, what kind of genes, what has made this species to adapt to this region of Africa. So I think there is really an opportunity to explore these genomes as we sequence them with reference to predicting novel proteins and also getting insight to genes that has enabled the adaptation of these different crop species to different regions in Africa. And then as the previous presenter also mentioned, there is also an opportunity to develop new drug targets and also develop antimicrobial targets from all of these uncharacterized genomes that we have. Okay, so what are the challenges? A lot of people already mentioned we have a data challenge. We're bridging this gap currently. We also have infrastructure challenge. I know we're also bridging this gap currently. And then some people already mentioned about ethics and AI, so I wouldn't go over that. But the part I really want to talk about, because this is really close to my heart, is how do we bridge the readiness gap? Like I mentioned before moving to the US 5 years ago, I worked in Nigeria for 5 years, where mostly my work, I was leading a local laboratory and was really involved in a lot of capacity building in molecular biology and bioinformatics through the Biotechnology Society of Nigeria. And over the years, one of the things I see is that we have workshops, 2-3 days workshops, which are really good, but I think in terms of impact, we need to really begin to reevaluate this approach. And the reason for that is that, as I think about the previous approach or the current approach we're using, we train 50 people, 100 people, but are we really following up on these people? And are these people applying what they are learning in what they're doing? And by that I mean, if you got a group of people and you train them in genome assembly and genome annotation, and they have no data really to work with, they will just go home and learn all of this theory, and nothing really, in terms of application. So, my challenge to us, and as we think about this really, is that how do we develop a system, right? Where people can have access to existing data, play around with this data, but also, really, lobby and speak with more government and different organizations to generate more data that people can really work with. Because right now I'm finishing my PhD, and I talk with some colleagues back home, and they have such brilliant ideas about what they want to do. But you see, the issue of funding is still a constraint. The issue of opportunities for training is still a constraint. So, this is one thing that I hope that we can also really think about. How do we reevaluate and improve on the current training pipeline that we have in order to bridge this gap? And, again, just talking about readiness gap, a recent preprint that I was reading from the African CDC, where they surveyed over 250 participants across 46 of the African Union states. Basically, only 4% of genomics researchers say that they are very familiar with AI application in genomics. And when they ask them, why is this? What are some of the key barriers to this? 78% says lack of training programs. 60% said there is a high cost of training programs. And even if you look at the other data out there, Africa is spending like 1/3 of what the rest of the world is spending on R&D. So there's really a need for us to change this narrative. And my hope is that this Congress would go beyond the rooms we are in right now, and hopefully inform government policies to change some of these key barriers. So in conclusion, I just want to say that I believe that Africa is at a critical inflection point, and that AI and genomics can really transform our agriculture, conservation, and bioeconomy. But this would depend on capacity building, on equity and collaboration, which we're already doing. And if there's one thing I would like to leave us with today, it's the fact that I strongly believed that we should not just be a source of data alone, but we must really be a source of insight. And so whether you're a graduate student, a professor, whatever level you are in your training, these things are possible. And I believed that in no time we're really going to catch up with the rest of the world. Yeah, thank you all for giving me an audience. I'll take questions if you have any.
Yeah, thank you, Chizin. I think we're gonna have like a panel style Q&A. Do we have any question for Vincent and Chizin? So maybe we'll go to the participant on site. So Julian or Genevieve, is there any question on site for Vincent and Chizin before we come to the online participants? Is there any question on site? A comment? A contribution?
Digital gap from Africa.
Sorry, could you please repeat your question? We didn't hear you earlier on, so start from afresh.
Okay, sir. Thank you for the wonderful presentation.
And also, can the IT person remove the background so we can see the the person asking the question? If you remove the poster on the background, that would be great. That's the IT. Okay, carry on, please. Carry on with your question, please. Okay, carry on, please. We can't hear him again. We can't hear him. We can't hear this person asking question. We can't hear him online.
No, nobody can hear him.
Yeah, we can't hear you online. So maybe we can just, while you're trying to fix it, let's take some online questions and then we can come back on site if that's okay. So Vincent, I think I see some questions for you.
Yeah, I saw one from Dr. Isaiah Ubu, but go ahead and
Okay, all right. So Isaiah Ubu is asking, I would like to seek your insight. Oh, I don't know if you want to answer this question because this is more clinical. Anyway, we're going to ask it and you decide.
I would like to answer it. Even though it's a clinical question, I think all of us will gain from it because we can always extrapolate. Yes, exactly.
What we're trying to avoid is anyway, I said he would like your insight regarding the use of AI-driven tools such as ChatGPT in the context of drug prescription. Specifically, when these tools are unable to provide an exact drug prescription, they often suggest alternative sources. However, some of these alternatives may be contrary to prescribed treatment or may not be appropriate. In light of this, do you consider the use of AI tools advisable for prescription purposes? What are the potential risks with relying on AI for such clinical decisions?
Yeah. Excellent question. And I think let me just break it down so that everybody, whether you're in the clinical area or the non-clinical area, at least I know it's going to be useful. These AI tools are supposed to assist you. They are not supposed to replace your knowledge. Because people are so scared that AI is going to take their job, take their job. That's no. AI, people that understands AI and how to use it and things of that sort will be the ones to take your job, not AI. So it's good for you to understand these tools. So the question Dr. Isaiah Ubu is asking is that you want AI to do all the work for you. No, AI can provide you the information necessary, and then you will still have to go back in to decide exactly what do you think is the actual way to do it based on your own knowledge. Because there is no way. I'll give up one example. When I was active here in the United States, we had several cases of endocarditis, okay, caused by some organisms. And then we noticed that some of the patients who had endocarditis from a particular city, the organisms that were responsible for endocarditis were different from the organisms in another city, okay, based on lifestyle. So AI cannot tell you these things. It's your own knowledge that will now help, to decide exactly what to do using the AI tools. So don't walk out of here thinking that AI tools is going to replace what you're supposed to do. You still need to go there and acquire the knowledge and then use the AI tools to make your work faster. Isaiah, are you?
Thank you, Vincent.
He did not unmute, so maybe I don't. Hopefully, he had me. Okay.
Yeah. So I
Another
think
question.
in the interest of time, we have one more question for choosing. Then we go to the site, and then we wrap up for today.
Okay.
Choosing, this is for you. Francis is asking, which of the AI-enabled structure modeling platforms would you recommend as being more accurate and reliable for crystalline stabilized peptide structure modeling as opposed to more complex proteins? I think is there like a benchmark or something that has been done? Is this
yeah, I would say he can try both two, which a number of people have found to like be a little bit better than AlphaFold, depending on the kind of structures you're working on. And I'll reply to him. Mostly I've used AlphaFold and BOS2 in my research, but I'm a plant genomics person, and most of the genes I work on are a little bit different than this 16, but you can try BOS2, I think, and then see how that works. Gus, you're muted.
Say, please go to the chat box and respond to the questions. In the interest of time, we can't go through all of them, so feel free to do that. We'll just take one question from Tobias, and then we go to the on-site participant, partly because he has raised his hand before and he wanted to ask more questions, so he doesn't feel like I don't want him to ask questions. So Tobias, please, very quickly, I know that we are 3 minutes past the hour and would have finished 3 minutes ago. So let's try to, over to you, Tobias, please. Just very briefly.
Okay, thank you. I thank you for the presentation. It's just that I want to, because you have presented something on this AI relative to drug discovery and all whatnot, I seem to have a challenge. I have one of my PhD students. Okay, I've been working on a particular plant which shows a positive indication for so many things, from phytochemistry and to using them. Then I wanted to find out which actual phytochemistry responsible for positive results I get with the crude. And then we now try to apply, he now tried to do, what do you call it now, AI related something where some Lepsius rule of 5 was applied to find out whether the candidate fits into the picture. And I now find out that that particular drug seemed not to fit in because of toxicity factor. Now, if you now resolve the positive results gotten on without the AI related or without the AI related, vis-a-vis the result gotten from non-AI, how do we move forward to resolve this problem? Because it seems I am placed on hold on where to move forward.
I think, Vincent, this is more for you because it's more tilted to clinical.
Okay. Well, yeah. Well, in case he had a question, he's involved with investigational drug discovery, and then the stuff was the chemical came from plants, which of course they are trying to move into different phases. So basically, they've not even got into the phase one yet. They're still in the preclinical phase. So if with the Lipinski rule of 5 you use, which of course did fit in, but there were toxicity issues. So now it's not going to move forward to the next phase. So we need to now go back to solve, to look at the chemicals again, see exactly what needs to be changed or adjusted so that you can eliminate, so at least the toxicity, the challenges you're having can be resolved before you can move to the next phase. Because nobody is going to be moving a chemical from one stage to the other, when already you've outlined these toxicity issues. Mind you, assuming that if you are successful and things work out and you go from different phases, from phase to phase to phase to phase, the world will be spending minimum $800 million before that particular compound is going to be used in patients in terms of the end users. So these things got to be taken care of. That's why for every one product you see that's available on the market, there are 100,000 of similar products that did not make it. So my suggestion is that we go back to the lab again, and then we get some more work done.
Thank you, Vincent.
Sorry, thank you for your insight. But please, can you suggest exactly one or two things that could be done as I go back to the lab or that preliminary stage before coming back again?
You get
privately.
You get your scientists together, and then you see what the issues are.
Professor Vincent, can you please put your email in the chat box so then he can reach out to you? I think this conversation needs to carry on outside the meeting because in the interest of time.
Yeah, so, my email is on the last slide. Yes.
Okay. All right. Thank you so much. People are asking for you to share your slide, so please feel free to share it. Thank you, Choosing. Thank you, Vincent. We're going to go to, just hang on the call. We're going to go back to on-site. So on-site, 1 or 2 questions, and then a brief wrap up ahead of tomorrow.
Yes, here we have one person that was actually asking a question, but we couldn't really get him. Now,
okay, now site is having internet issues. And they were waiting for us all through.
Part of the infrastructure.
We talked a lot about infrastructure. So these basic things got to be taken care of.
Yeah. Okay, so I think we will just give on-site maximum 30 seconds or 45 seconds. Then if they don't come online, I'll just wrap off or 1 minute.
Well, for those on the line, I don't think they can hear me.
We're hearing you.
Okay. For those on the line, you can feel free to reach out to me. And then if you want to reach out to me in person, I should be, if you're going to be in Port Harcourt during the convocation of the university, we can see face to face and discuss these issues. Once I get back to Port Harcourt, the convocation is in the first week of June. So just to let you know, so we can, and I'll be there. Be heading back sometime this month. So we can.
Okay.
We can take up some of the discussions from there.
Yeah. So it looks like they are not back yet. So I think I can actually wrap this up. because we couldn't get back. They couldn't reconnect.
You couldn't get
again.
back to the, okay.
Yeah, they couldn't
Okay.
reconnect again. So basically, we're almost wrapping up the
Yeah,
meeting.
can you?
Okay, we can hear you. Please go ahead.
Yeah, all right. The last person asked his question. I don't know if you got him. If you had answered that question, probably we might wrap it up because
No,
there's.
we didn't hear the question because we lost you guys like as soon as you wanted to speak.
Yeah, okay, please go ahead. Yeah, okay, I think I take this one.
Yeah, so you talked about the data gap on African data sets, and you said so much is being done to close the data. So I just wanted to say in terms of GWAS, that most of the African data is basically more on African of data on continental Africans. So what is being done to close this data gap on continental Africans, especially on GWAS and also polygenic records? Thank you.
Did you get his question? Because I think I struggled to hear him. Just choosing Vincent, did you get his question? Otherwise,
Yes, I did. I did, but I think the question was probably directed to the last speaker.
Choosing. So choosing, did you get his question? Feel free to ask me to ask him
Partly, but I was
Okay.
actually thinking it was directed to Vincent.
Okay.
So, yeah, okay, I can tackle it then from the clinical grounds. Where what happens here is this, that to simplify it, it's about getting more data for stuff that Africans can use to get to able to tease out. I'll give you an example. In America, when I go to, just a very common example, in America, when I go to the store to buy a shirt, I buy a small, because a small is what's going to fit. Because the data here, the small shirt here is based on the size, because the average American is at male is about 5'10". Okay, so that's what fits. But if I go to China to buy that same shirt, and I buy a small, it's not going to fit. I can barely put it on. So in China, I'm going to be using a large, because that's what the data there stipulates in terms of that. So we need to now basically collect our own data, okay? And then after we've collected our own data, and now it's going to be useful, because we cannot extrapolate data from elsewhere to the African population. So we are still in the stage of collecting data, and it's not going to stop. Even in the advanced world, they are still collecting data. Data collection and storage.
Thank you, Vincent. Julian, do we still have any more questions on site?
Oh, no, nobody. But I guess you're satisfied. Yeah, I think the last person that asked question is satisfied with the answer.
All right.
And here again, there's no other person that wants to ask a question. So I think at this point, we may have to announce that we are about to wrap up today's sessions. Tomorrow we are meeting again, and the fourth session tomorrow is starting at 9 o'clock GMT +1. Hello, can you hear me?
Yes, we can hear you. Go ahead. We can hear you perfectly well. Okay, now we can hear him again. So, Julian? We can't hear you if you are speaking. So I think you are trying to wrap up the meeting. I'm sure we can also wrap up the one online. Okay, so I think we will just wrap it up. As you heard previously, we'll be meeting tomorrow for the Congress, kind of to focus more on the policy and the infrastructure on the DSI side of the conversation, which many people wanted to have today unintentionally, but wanted to leave tomorrow for the policy-related discussions. So I would like to thank all the speakers for speaking today, and those that also joined the panelists, through the Q&A, and the oral presenters as well, and the moderators. So we'll see you all tomorrow, and yeah, hopefully we'll have additional conversations tomorrow. Bye.
Bye-bye.
What if we could anticipate the future of disease and prepare for it today? Genomics, epigenomics, transcriptomics, proteomics. Question, innovation, insight, impact. With the Illumina Insight ecosystem, you can define a new era of omic connectivity where predictive, preventative, and personalized medicine are within reach. As we uncover the questions and explore the solutions together, we come to understand the promise of our search. Your question is the innovation. Your innovation is the insight. Your insight is the impact. In this new ecosystem, at the nexus of innovation, solution, and community, with expansive multimodal data sets and AI-powered discovery, with the highest quality insights at the lowest end-to-end cost, we can accelerate impact and improve human health together.
Welcome to the Journal of Bioscience. We are located in Lagos, Nigeria, and we also operate in Cameroon and other West African countries. Our mission is simple: to unleash the potentials embedded in life science to make a healthier and safer ecosystem. And this is why we have partnered with Oxford Nanopore Technologies, a technology that has made sequences simple, and the mission is for anyone to be able to sequence anywhere. This technology has been used in the space, under the ocean, and even anywhere, small labs to big labs.
Oxford Nanopore Technology offers long-read DNA and RNA sequencing, and the Oxford Nanopore Technology has 3 varieties of devices. We have the mini-ion device, which enables you to have a data output of about 48 gig. So depending on your projects, maybe you want to do animal fungi genome, you want to do metagenomics, you're able to use the mini-ion category of devices. Then we'll move on to the Promethean. Promethean offers about 7 terabyte of data output. So you want to do whole human genome, you want to do population studies, you want to do biodiversity studies. Promethean will enable you do that.
The Oxford Nanopore Technology produces long read, which helps cover complex genomic region. And with this long reads, you don't have to struggle with genome assembly or genome alignment. And because the long reads has covered complex genomic region. And of course, these long reads are very essential for structural variant detection. And beautifully, the ONT platform has created an application for the downstream bioinformatics analysis. Because without the bioinformatics analysis, you only have chunks of letters, A, T, C, G. You don't know what that means. You can't make clinical relevance from them until you have done the downstream bioinformatics analysis. And the ONT platform has made an epitome, an application for this downstream bioinformatics analysis.
We have a reliable team to support you in every aspect. You do not need a special expertise to be able to use this technology. We have technical team to guide you through the wet lab process. We have a bioinformatics team, and we have the sales team to help you with all your requests and quotations. We have a standard logistic team, inventory team to supply you. Our incoterms include DAP and DDP. So we are able to supply to your doorstep. You do not have to worry about custom clearance. You do not have to worry about logistics. And these are reliable teams that are with you all through the way, from wet lab to dry lab to logistics to even planning your budget. You can place an order via our store. The link is on your screen. And also you can send a request to this email address ont@genelab.ng or tgl_onc@genelab.ng.
Meet the new Vega system from PacBio. Trusted Hi-Fi long-read data is now more accessible and affordable than ever. From genomes to transcriptomes to epigenomes, Vega gives you the control, read length, and accuracy you need to explore with confidence. With its fast and intuitive setup, just touch and go. You can focus on discovery without delay. Your process, your pace. From ideas to results, we're here with you every step of the way. Vega by PacBio. Hi-Fi sequencing within reach.